Chapter 1 Preface The Colloid Chemistry of Protoplasm by L.V. Heilbrunn, Protoplasma Monographian, Volume 1. Published in Berlin by Gebrüder Bornträger, 1928. This book was begun at the Kaiser Wilhelm Institut für Physikalische Chemie in Berlin-Dahlem. It was continued at the Pflanzenphysiologisches Institut of the University of Graz, and it was completed at the Stazione Zoologica at Naples. Finally, it was completely rewritten at Graz. I have enjoyed the writing of it, partly because of the kindly help of Professor Weber and the pleasant discussions we have had together, partly because of the pleasure I have had in looking up old references to forgotten literature. In present day biology, there is a tendency to avoid criticism. One does not like to hurt the feelings of one's fellow workers, and biologists seem particularly sensitive to criticism. When I began to write, I thought first that I would make no mention of work that I knew to be incorrect. It was Prof. Weber who urged me to discuss all papers, good and bad. This in general I have done, although I have usually not attempted to consider the writings of those speculative authors whose work has been limited to an expression of opinion or the development of theory. The modern biologist often reads only summaries or abstracts of papers. He scarcely has time to do more. As a result, he is apt to take an author's statements at their face value. Accurate, trustworthy data receive no more attention than conclusions based on the flimsiest sort of evidence. Both are cited equally. The result is that our biological knowledge, and especially our knowledge of the physiology of the cell, is an unfortunate mixture of the true and the false. Perhaps, as some optimistic scientists insist, the true will eventually be sifted from the false. But why not hasten the sifting process? An incorrect observation or a wrong conclusion may start many workers on false trails and may involve a huge waste of scientific time. As one reads the literature of cellular physiology, as one runs across some incorrect statements cited over and over again, one often wonders if there might not be some means of preventing obviously false statements from finding their way into the literature. Perhaps now and then, it might be possible for isolated workers to send their manuscripts away for criticism. I, for one, have frequently benefited by the critical help of others, and I should be only too glad to extend any critical help in my power to authors who might wish to send their manuscripts to me. The science of the colloid chemistry of protoplasm is barely beginning. One could scarcely hope to develop the subject on a thoroughly logical basis. For this reason, I have for the most part considered the experimental data piece by piece. Thus, the book contains chapters on the effect of temperature, on the effect of acids, etc. later when the colloid chemistry of protoplasm is better understood. One may perhaps be able to write a book in which the subject is divided on the basis of the fundamental properties of the protoplasmic colloid, rather than on the basis of the type of treatment to which it is subjected. Wherever possible, I have tried to indicate lines of research that might prove profitable. It is my hope that these hurts may prove useful, even in those cases in which I state that experiments of my own have been started in one direction or another. I do not feel that this should stand in the way of other workers who might wish to begin investigation in the same field. The book was written during the tenure of a Guggenheim Fellowship. The author wishes to thank the donors of the fellowship for the opportunity of spending a year away from the demands of teaching, away too from the disturbances and distractions of American university life. Thanks are also due to Professor Weber for the use of his splendid collection of reprints on the physical chemistry of protoplasm, to Professor Linsbauer of Graz for the use of the library of the Pflanzenphysiologisches Institut, to Professor Boemig of Graz for the privilege of using the library of the Zoologisches Institut, a library which is remarkably rich in the older literature, and to Professor Freundlich and Dr. Ettisch of the Kaiser Wilhelm Institute of Berlin for answering questions of a physico-chemical nature. Finally, thanks are due to Frau Prof. Weber for the conscientious care with which she redrew those figures which were copied from other authors. For aid in correcting proof, I am especially indebted to Professor J.W. Wilson of Brown University and to Mr. L.G. Barth. Chapter 2, Introduction. During the 19th century, it became customary to think of living things as being made up of a special sort of material, the protoplasm. This might differ in various organisms or in various parts of the same organism, but it was thought that all living substance, no matter where it was found, had certain characteristic properties not shared by the non-living. These characteristics were not usually stated in terms of the protoplasm itself, but rather in terms of vital function. Thus, protoplasm could reproduce, it could grow, it breathed, that is to say, it gave off carbon dioxide. In this way, the living properties of the organism as a whole were transferred to the stuff of which the organism was made. Although in some respects this is a useful point of view, it is far more important to be able to describe protoplasm directly in terms of what it is, rather than in terms of what it can do. In books on biology, protoplasm is usually described as a viscous hyaline liquid. Probably it is a liquid, at least typically, although many recent authors have contended otherwise. To say protoplasm is viscous means little or nothing. Water is viscous in comparison with many other fluids, but in comparison with glycerin, it is highly fluid. Speaking broadly, most protoplasm is quite hyaline, although the work of Keelan 25 indicates a wide distribution of pigments in living cells. The microscopic appearance of protoplasm has been the subject of countless studies, and the knowledge that has been built UID has been gathered together in various books on cytology. Protoplasm is typically in the form of cellular units, each with a nucleus. The structures found within these cells are of different sizes and shapes, but on the whole there is a thoroughgoing similarity in the cells of very different types of living material. In the next chapter, some of the more essential facts regarding cellular morphology will be briefly enumerated. For the present, let it suffice to say that morphological study has clearly demonstrated that living material, although never quite the same in different animals and plants, nevertheless presents a remarkable constancy of appearance when viewed under the microscope. Thus, two animals of extremely different form and size are frequently found to have cells which are surprisingly similar. Chemical study has shown this similarity to be more than a matter of form. Analyses of different animals and plants have yielded the same elements and the same types of compounds. There are differences to be sure. In some cases, specific types of chemical compounds are found only in some organisms and not in others. But all living things are alike in that they are a saltwater solution of proteins with an admixture of fats or lipids. It was in the 19th century that the underlying unity of life was first stressed. The cell theory and the evolution theory are only two aspects of such a point of view. More than anything else, it was this emphasis on the fundamental similarity of living organisms that made the development of theoretical biology possible. If each type of organism and each type of cell were totally unique, there would be 1,000,000 biologies instead of a single science of biology, and the human mind could hardly attempt to solve such a vast myriad of enigmas. The knowledge that living things are remarkably similar has given the biologist courage to proceed with his investigations. Fortunately, problems that are completely insoluble for certain types of organisms are relatively simple for others, and the results that have been gained for one type of living thing are often generally applicable. The laws of inheritance, which were established for the garden pea, are laws which apply to the inheritance of human characteristics. Ether, which was found to be anesthetic for human cells, is an anesthetic for most of the simpler organisms as well. In stressing the similarity of living mechanism, it must not be forgotten that there are innumerable differences in various types of living material. It is true that there is one science of biology, but it is a science with infinite variations. Certain biological truths are universal, others are relatively specific. It is never safe to assume that what is true for one sort of protoplasm is true for another. Only experiment can decide. Sometimes what we might be inclined to regard as a general truth is extremely limited in its application, whereas another fact, apparently specific, may be found to apply to many different types of living substance. It is a natural tendency for a student of one particular sort of protoplasm to imagine that his results are of importance for every sort of protoplasm. The history of biological thought has shown the necessity of continual guard against such a tendency. Often enough, what is true for the cells of one animal is not true even for the corresponding cells of a closely related animal. There are facts about protoplasm which hold good for practically every type of living substance. And these are the facts which are most worth knowing. If it is true that all living cells manufacture carbon dioxide, in making this statement, it is possible in a single sentence to give information regarding millions protoplasm. This is far more important than to know that some special sort of cell is able to produce a specific chemical compound. The human mind cannot hope to be all-knowing. We must cling to the facts which are broadly and generally true and leave the specific details to gods and encyclopedias. But it is not merely a question of memory. Those characteristics of protoplasm which are universal are almost certain to be intimately related to the machinery of the living process, whereas a characteristic peculiar to a few cells must be related rather to the life of these particular cells than to life in general. The biologist whose primary interest is to gain an insight MTO, the mechanism of life, can well afford to neglect specific properties of certain types of protoplasm, in focusing his attention on those properties, which are universal. Certain types of protoplasm are far more favorable for study than are others. Animals high in the evolutionary scale are made up of vast numbers of cells, for the most part closely bound together. To be seen under the microscope, these cells must usually be isolated, or at any rate cut up into smaller masses. Under these conditions, abnormality or death frequently results. True. The newer methods of tissue culture have done much to make possible the maintenance of life in cells cut out from large masses of tissue, and perhaps in the future, cells in tissue culture will more frequently be investigated by the student of living protoplasm. There are problems in the physiology of the cell for which the tissue culture method seems particularly suitable. Some types of cells are isolated in nature. Protozoan cells, yeast cells, the eggs of marine invertebrates, all these are familiar examples. An isolated cell is relatively easy to study. It must not be too small. With our present methods of study, no one could hope to learn much about the inner machinery of bacteria cells. On the other hand, not all large cells are favorable material. A bird's egg is made up mostly of yolk and investing membranes. Only a small fraction of it is protoplasm. And what protoplasm there is can only be examined microscopically under conditions which involve the death of the cell. Protozoa have often been used in the study of protoplasm, easy to obtain in all seasons and all places. The cells are often of a very convenient size for microscopic examination. Unfortunately, many protozoan cells are not as simple as was formerly thought. They may be provided with neuromuscular systems or with various other miniature organs, which give the protoplasm a high degree of specialization. Except for its food vacuoles and its contractile vacuoles, such organs are lacking in amoeba, and it is thus easy to understand why this organism has so frequently been chosen for study by the student of protoplasm. The egg cells of marine invertebrates have many advantages. They are large enough for a thorough-going microscopic investigation, and yet often not so large as to be opaque. Many of them are quite transparent. Most invertebrate eggs are spherical. It is thus easy to estimate their volume. This is an important matter, for in many experiments, it is essential that the volume of the cell remain constant. In any given species, the egg cells are usually of very uniform size. This is an advantage such material has in comparison with protozoan material, in which there is usually great variation in the size of individual cells. It is, of course, true that marine eggs can only be studied at certain stations, and often only at certain times of year. But when the material is available, it is generally present in great abundance. Obviously, not all marine eggs are equally valuable for purposes of study. Some worm and mollusk eggs are shed when they are in the metaphase of the FHS maturation division. If such eggs are subjected to almost any chemical or physical treatment, they immediately react HJ, throwing off a polar body. It is therefore difficult to study the effect of chemicals on these eggs, for the experiment is usually complicated by the onset of the maturation process. For many purposes, the sea urchin egg is ideal. When shed or cut out of the ripe ovary, it is fully mature. Sea urchin eggs can usually be obtained in large quantities, and the breeding season is very long, often lasting for many months. Because of their many advantages, sea urchin eggs have been widely used in protoplasm studies. although these cells are devoid of all specialized organs and organelles. Nevertheless, it must be remembered that an egg cell is only one type of living substance, and that any results gained with this sort of protoplasm can only provisionally be applied to other sorts of protoplasm. It is unfortunate that so few types of cells lend themselves to experimental study. The student of protoplasm should be ever on the lookout for new types of material. For some purposes, the protoplasm of slime molds is especially useful. It occurs in large aggregates, and for this reason it offers unique opportunities for experiments on protoplasm in mass. Some authors have found the cells of various algae convenient for study. Algae often have very large cells, but in these instances, most of the content of the cell is usually vacuole. This introduces complications, as does also the presence of a chromatophore like that of Spirogyra. Nevertheless, the Spirogyra cell has frequently been used to advantage in protoplasm studies. The cells of higher plants are also useful for many purposes. On the average, these cells are larger than the cells of animal tissues, and most of them are relatively undifferentiated. In many plant cells, the protoplasm is normally in a state of motion, and this has often proven very helpful to the students of plant protoplasm. The speed of movement of the protoplasm is readily measured and may frequently serve as an index of changes in the physical properties of the living substance. The many studies of protoplasmic streaming which have been made in the past will be even more useful when a satisfactory mechanical explanation of the phenomenon is arrived at. The first cytologists studied and described the morphology of protoplasm. Later, with the development of the science of biochemistry, conclusions could be drawn regarding the chemical composition of the living substance. Until recently, the physical properties of protoplasm have been field for speculation rather than for exact study. This is in part due to the intrinsic difficulty of making a physical study of such tiny objects as living cells. Chemical study is in some respects easier. One can analyze large masses of cells and be certain that the chemical compounds found in the entire ensemble are also present in the individual units. But many physical properties are not additive. The surface tension of a mass of cells is not the sum of the surface tensions of the single cells. The electric potential difference across a tissue can certainly not be calculated from the electrical potential difference across an individual cell, or vice versa. Generally, if one is to obtain information concerning the physical properties of protoplasm, one must study the individual cellular units. This is a broad statement and is not to be taken too exactly. Nevertheless, in the main, it is true. To the biophysicist, protoplasm is chiefly interesting from the standpoint of mechanics. True, living cells give off heat and electricity, and some or perhaps all give off light radiations. But this energy is more in the nature of a byproduct. Fundamentally, the living cell is a machine which functions mechanically. The movement of a cell is essentially a problem in mechanics. When a cell grows, the primary physical factors, as far as we know, are such processes as diffusion and osmosis. A cell divides in a complicated mechanical fashion. Thus, from the standpoint of physics, it is the mechanics of protoplasm that is of the first importance. In the physical study of the living cell, there have been two chief lines of investigation. Following the lead of Perfei, many authors have studied the osmotic properties of protoplasm. Cells are osmometers or osmotic machines, and they behave as such. In any osmometer, the nature of the osmotic membrane is of primary importance. Within relatively recent years, cellular physiologists have given a great deal of attention to the study of the permeability of the osmotic membrane of the living cell. There can be no question but that this is a subject that deserves full and careful study. Unfortunately, as is now generally recognized, many of the results that have been published do not stand critical examination. At present, it seems just as important to attempt an evaluation of the older experiments as to make new ones. From the large mass of papers that have been published, certain facts stand out fairly clearly. The relative ease with which different types of chemical compounds pass through the osmotic membrane of the cell seems to be quite well understood. On the other hand, there is still a great difference of opinion as to the relation of permeability of the osmotic membrane of the cell to the activity of the protoplasm. Important as are the osmotic properties of the cell and the cell membrane, They represent but one aspect of the physical study of protoplasm. The living substance is not a homogeneous material. Beyond any question, it is a colloid. In the study of colloids, the physicist and the physical chemist have gathered together a vast body of information, which forms a definite branch of physics and is called colloid chemistry. It is possible to study the colloidal behavior of protoplasm as well as its osmotic behavior. We are thus led to a colloid chemistry of protoplasm. What is colloid chemistry and what are colloids? The definitions are somewhat arbitrary. When one substance is mixed with another, its molecules may be scattered in such a fashion that each molecule is distinct, or the molecules may form larger aggregates. Generally speaking, when one component or substance is scattered through another so that the individual particles are larger than 1 at GT 0.000 millimeters and smaller than 200, the particles are said to be colloidal particles, and the entire mixture is spoken of as a colloidal solution. Colloid chemistry is the science of colloidal solutions. Solutions whose dispersed particles have dimensions between 1c and 200. Some substances of high molecular weight, for example, proteins, have molecules so large that the individual molecules may be regarded as colloidal particles. The upper limit of size of colloidal particles, 200, is a purely arbitrary one. Ordinarily, particles smaller than 200 pLGIG diameter cannot be seen with the microscope, and it is for this reason that this particular dimension has been chosen. But colloid chemists do not take the definition of their science too seriously. In general, when a material is dispersed so that its particles are larger than molecules or smaller than 200 EWW, the resultant solution has certain typical properties which are not found in true solutions or in very group. But there is nothing magical about these size limits. A suspension with particles just barely visible under the microscope differs in no way from one with particles just too small to be visible. Broadly speaking, a colloidal solution has dispersed particles large enough so that they show the properties of matter and mass rather than the properties of individual molecules. Frequently, the colloidal particles of any one chemical substance tend to adsorb at their surfaces traces of other materials, and these added materials often have a decided effect on the physical behavior of the entire particle. The entire particle with its adsorbed substances is sometimes referred to as a micella. From the time that colloid chemistry first appeared as a science, no one has doubted that protoplasm is a colloid. The very fact that it is largely composed of proteins indicates that it must have colloidal properties. Meat and blood are colloid, so too is a solution of egg albumin gelatin, so often studied by colloid chemists in recent years, is a derivative of living tissue. Many biologists have stressed the importance of developing a science of the colloid chemistry of protoplasm. But progress has not been rapid. For a long time, ideas with regard to the colloid chemistry of protoplasm were of two sorts. In the first place, it has been common practice among physiologists to explain every biological process in every sort of tissue by some postulated colloidal change in the protoplasm of the tissue cells. Such philosophical speculation was indulged in by the best and most prominent physiologists of the latter half of the 19th century. And as a matter of fact, this type of explanation still remains very popular in certain branches of physiology. A more scientific, though hardly a more successful method of attack, was the method of analogy. Various inanimate colloids were, for one reason or another, chosen as being similar to protoplasm. The colloidal behavior of these relatively simple colloids was then studied as though it were the behavior of protoplasm itself. In the absence of other methods of study, there is some justification for this type of work. Unfortunately, the colloids chosen as models of protoplasm were extremely unlike the living substance. Consider gelatin, for example. More conclusions have been drawn about the colloidal properties of protoplasm from a study of gelatin than from a study of any other colloid. And yet, in its fundamental behavior, gelatin is entirely different from animate material. From the first, it has seemed certain that protoplasm is coagulated by heat, and recent direct evidence supports this general notion. On the other hand, gelatin is liquefied by heat. Moreover, gelatin solutions are gels at room temperature, whereas the types of protoplasm about which we have most certain knowledge are not gels. If there is to be a colloid chemistry of protoplasm, it must be based on a direct study of the living substance itself. Life is undoubtedly dependent on the colloidal properties of the materials of which living things are composed. No amount of study of dead inanimate substances will ever reveal these properties. Such study may eventually prove helpful. In the future, it doubtless will. But as the study of the colloid chemistry of protoplasm makes its first crude beginnings, it is hardly possible to decide just which inanimate colloids are similar to the colloids of living cells. That is to say, similar in their physical properties. And all we can hope for at best is a remote resemblance. The simplest protoplasm is vastly more complex than any known inanimate colloid or mixture of colloids. Only in the past 10 or 12 years has there been any systematic attempt to study the colloid chemistry of protoplasm. Before this time, there were a few sporadic observations, byproducts of studies in other directions. And of course, there were plenty of theories, but the investigation of the colloidal properties of protoplasm could not make progress until methods of work were established. The great difficulty in the way of studying the colloid chemistry of living cells lies in the fact that visual observation usually gives no certain information with regard to colloidal change. When gelatin in a test tube liquefies, this is immediately obvious even without measurement. The coagulation of egg albumin or the flocculation of a colloidal solution of ferric hydroxide can easily be followed with the naked eye. But when one looks at protoplasm, even with the highest powers of the microscope, it is usually impossible to detect even the most thoroughgoing change of state. True, authors in the past have described coagulations and liquefactions in living cells on the basis of simple microscopic observation. But the criteria employed have often been purely arbitrary, and the results are frequently of little value. That the physical state of the living colloid does undergo marked changes has been amply proven by recent studies. Moreover, it seems certain that these changes in state are closely related to the fundamental machinery of the living process. From this standpoint alone, the colloidal study of protoplasm has made an auspicious beginning. But this is not all. Given the proper methods, the student of protoplasm can study it just as though it were an inanimate colloid. And in this way, he can learn to know the physical proprieties of the most interesting of all fluids. How can one study the colloidal properties of protoplasm if microscopic observation does not show when the protoplasmic colloid changes from solid to gel? How is it possible to find out? In a test tube, when a colloid changes from solid to gel, it undergoes a great increase in viscosity, and at the same time, it becomes elastic. If then, we were able to measure either the viscosity or mation with regard to colloidal change. At the present time, it is possible to make measurements of the viscosity of protoplasm, and these measurements can indeed be made with a fair degree of accuracy. Given such a possibility of measurement, it is no longer necessary to limit ourselves to speculation, or to confine our studies to inanimate colloids, which are assumed to resemble protoplasm. We can actually begin a direct colloidal study of the protoplasm itself. In a subsequent chapter, the methods of viscosity measurement of protoplasm will be critically discussed. For the present, let it suffice to state that such methods have been developed. In studying the colloid chemistry of protoplasm, it must not be forgotten that living cells are very small, and that the conditions are thus different from those to which the colloid chemist is accustomed. When a small amount of electrolyte is added to a metallic sol, or when a protein is salted out, there is a precipitation of the colloid, and this is easily observable. But hot precipitation could hardly occur often in protoplasm, for a living cell may be much smaller than a single flake of a colloidal precipitate. The speed with which a precipitate settles depends on the size of the particles. For spherical particles, it increases as the square of the radius. Obviously, in tiny cells, there can be no rapid settling out of a precipitate, for the particles of a X3 precipitate could never be large enough to settle rapidly. Thus, in protoplasm, one of the commonest types of colloidal change cannot ordinarily be realized. It is not only in their small size that living cells differ from ordinary colloidal systems. Some of the peculiar characteristics of living colloids will be discussed in subsequent chapters. Because of the fact that the conditions in living systems are so different from those of non-living systems, it is rather difficult to use the ordinary terminology of colloid chemistry. In general, it seems proper to refer to a very pronounced increase in viscosity, that is to say, an increase of several 100% or more, as a gelation. In those cases in which the viscosity increase is certainly due to the appearance of a new phase, it seems logical to say that there is a coagulation. But it must be remembered that the suggested use of these terms does not exactly correspond to the manner in which they are used by the colloid chemist. The colloidal study of protoplasm is not easy. Observations must be made on tiny droplets of a material incompletely defined from a chemical standpoint, and probably consisting of a mixture of highly complex compounds. The results that can be gained with such material must be regarded leniently. The biologist cannot be expected to obtain data of the same degree of accuracy as those which the physical chemist can obtain with chemically pure compounds, but one must not be too lenient. If the colloid chemistry of protoplasm is to be a science, it must be based on firm facts, and not on subjective IMX regressions. In the chapters which follow, an attempt will be made to present the facts as they are now known. Few as these facts are, they show that a rational science of the colloid chemistry of protoplasm is possible, and that it is now in the making. Chapter 3. Introduction continued. The morphology of protoplasm. Inanimate colloids may be studied in any container convenient for the experimenter. Protoplasm must be studied in cells. It will therefore be necessary to review briefly the elementary facts regarding the general form of cells, as well as the knowledge regarding the microscopical appearance of their contents. Colloid chemistry is directly concerned with the problem of form. In any study of a heterogeneous system, it is important to know the general arrangement of the various phases which compose it. In a colloidal solution, the size and shape of the dispersed particles are factors of importance. Inasmuch as colloidal particles are typically too small to be seen under the microscope, information concerning their size and shape can in most cases be obtained only by the use of some ultramicroscopic method. As was pointed out in the last chapter, systems whose dispersed phase is composed of particles barely visible with the microscope also show the properties of true colloidal solution. Protoplasm contains many particles so small as to be barely visible. It is therefore important from the standpoint of colloid chemistry to determine exactly what can be seen when living cells are examined under the higher powers of the microscope. The founders of the cell theory regarded the cell as primarily a receptacle with a wall. Later, when it was found that some cells were apparently naked, the interior of the cell was thought to be more important, and a cell came to be defined as a mass of protoplasm rather than as a receptacle. Every schoolboy now learns that a cell is a mass of protoplasm containing a nucleus, to be a discrete mass of protoplasm and not an indefinable part of a whole. A cell which forms part of a tissue must have boundaries. In plant tissues, there is typically a fairly thick wall around each cell. When a piece of leaf or other plant tissue is placed in a concentrated solution of salt or sugar, the individual coals or protoplasts shrink away from their walls. They are then said to be plasmolyzed. In general, animal cells do not plasmolyze. When they are placed in concentrated solutions of salt or sugar, the entire cell shrinks, and there is no wall outside of it. Apparently, the animal cell is comparable to the plant cell without its wall. The fact that living cells shrink in concentrated solutions and swell in very dilate solutions is regarded by most physiologists as evidence that the cell is surrounded by a semipermeable membrane. No other explanation seems to fit the facts, although a few unorthodox physiologists have contended that other explanations are possible. The semipermeable membrane, which surrounds a living cell, is called the plasma membrane. Physiologists often state that the plasma membrane is invisible, and this has become somewhat of a tradition. Actually, there is no reason to suppose the membranes which can be seen about animal cells are not plasma membranes. The egg of the sea urchin is surrounded by a hyaline membrane, which appears to have very nearly the same refractive index as seawater and is thus hard to see. This membrane is about 1 micron thick. It is a semi-rigid structure. At any rate, under certain conditions, it will retain its form as the egg contents are squeezed out. When placed in a hypertonic solution, the egg decreases in diameter, and the membrane is pulled in with the shrinking protoplasm. Because of its rigidity, the membrane can be thought of as being under an elastic tension when it is pulled back in this fashion. There would then be a tendency for the membrane to spring away from the rest of the cell, and perhaps this occurs under certain conditions. Many protozoan cells are surrounded by relatively thick membranes which are readily visible. In paramecium, for example, the outer membrane is a thick structure, cilia, that this rigid membrane is really the plasma membrane of the protozoan argument. If we suppose a cell to be surrounded by a relatively rigid membrane, a membrane which could bend but which could not decrease its surface, shrink, age of such a cell would not be possible unless the cell changed its shape in such a way as to have a smaller volume but yet the same extent of surface. This is exactly what happens when a paramecium is placed in hypertonic solutions of sodium chloride. Under these conditions, the paramecium immediately flattens. The volume of the cell thus decreases, whereas the surface remains relatively constant, just as we have postulated for the case of a cell surrounded by a rigid membrane. Thierry had thus reason to believe that the visible tough membrane of paramecium is its plasma membrane. It may therefore be regarded as probable that the membranes which can be seen to surround some animal cells are the osmotic membranes of these cells. Perhaps in every case, the visible membrane of the animal cell functions osmotic. In plant cells, on the other hand, it is generally impossible to distinguish a definite membrane inside the cell wall and separate from the main mass of the protoplasm. We conceive of a cell as a receptacle in which there is an outer membrane which functions osmotically. This membrane, the plasma membrane, may or may not be visible. It should perhaps be stated that there are types of protoplasm which are really not in the form of cells. Some algae and many fungi consist of long strands of protoplasm with many nuclei. A muscle fiber also contains many nuclei. If we define a cell as a mass of protoplasm containing a nucleus, then a muscle fiber is either a group of cells without cell boundaries, or it is a single CU which differs from the usual type in that it possesses many nuclei. Cytologists usually refer to such a cell or group of cells as a syncytium. Obviously, a syncytium is difficult to interpret in terms of the cell theory. Moreover, it is only by a stretch of the imagination that tiny bacteria can be conceived of as cells. Books on cytology usually make no mention of bacteria. Cells vary greatly in size and shape. Human red blood cells are discs, the diameter of which is 7 microns. Sea urchin eggs are spherical, and in some species are about 75 microns in diameter. Cells which form part of a tissue mass may be prisms or polyhedrids. Animal cells are usually larger than red blood cells and much smaller than sea urchin egg cells. Of course, there are exceptions. On the average, plant cells are larger than animal cells. It has become part of the cytologist's credo to insist that every cell has a nucleus. In those cells which have no nucleus, it is usually possible to distinguish granules which have the same affinity for dyes as do the granules of typical nuclei. Such granules are then referred to as a dispersed nucleus. Dispersed nuclei occur in a few protozoan cells, but are not ordinarily present in the cells of higher animals. Typically, the nucleus appears as a spherical or ovoid body, and it is generally in the center of the cell. The volume of the nucleus may be as much as 1/3 of the volume of the cell, or it may be only 1/100th of this volume, or even less. Concerning the form of the nucleus and the structures which can be observed in it, both in the living and after treatment with various reagents, the cytologists have built up a large literature. When a cell divides, chromosomes are formed out of the nuclear material. For each species of animal and plant, these have a typical number and often a particular shape or shapes. Volumes of description have been written about the chromosomes, and their study has proved one of the most interesting chapters of modern biology. Apparently, the nucleus is the most important part of the protoplasm. It has, however, only seldom been studied from a physical standpoint. This is largely due to the fact that it is more difficult to obtain information concerning the physical properties of the nucleus than it is to obtain similar information concerning the cytoplasm, began searching the living material for details of structure which might help them to understand its amazing properties. The microscopes of the 17th and 18th centuries were crude instruments with lenses uncorrected for chromatic aberration. The early microscopists saw every object as a series of rainbows. More than anything else, it was the development of achromatic lenses in the early part of the 19th century that made possible the establishment of the cell doctrine and the growth of the modern science of cytology. But even before the appearance of the modern microscope, it was clearly recognized that the living substance contained granules. And just as every modern textbook of cytology or histology emphasizes the cell just the unit of the organism, so did the textbooks of the early 19th century emphasize the granule as the fundamental unit of the living material. Compare Slav. 99. This point of view has never quite faded out. There has been an unbiased line of investigators who have clung to the idea that the genus of protoplasm are the essential units. Many of these workers conceived of the granules as elementary organisms in Latin. Years they have frequently been compared to bacteria. It has even been claimed from time to time that the granules are capable of an independent existence outside of the cell. Whereas to some authors, the granules of the cell have signed the only living part of it, others have held strongly to Tlio view that the granules were of secondary importance, and that the life of the cell centered in the hyaline ground substance. In the lack of definite experimental evidence, neither view is justifiable. The centrifuge experiments of various embryologists, that is, interested primarily in the organization of the egg in relation to development, indicate that various cells of developing 3-AECV, freight larvae, can exist normally without the presence of ionol, another specific type of granule. In these experiments, none of the cells is ever wholly granule-free, partly because Tli granules are usually not completely moved by Tliocentrifuges, but also because in most of these experiments, the time de-12 centrifugal treatment and cleavage is long enough to allow many of the smaller granules to return by Brownian movement to their original position in the egg. As a matter of fact, Tlierci 6 miss every reason to believe that both granules and ground substarusi have a definite part to play in the life of teleocell. CV and die, as far as our present knowledge goes, we have no right to consider either the one or the other constituent as more truly living matter. Interpretation aside, it is certain that teleoprotoplasm of practically all cells contains vast numbers of tiny granules. These may be as much as 3 or 4 microns in diameter, and usually they are smaller, and quite commonly they too almost at the limit of microscopic visibility. The granules and rogofuoli very nimius. There may be so many of them that the RS to the protoplasm is little more than a series of films surrounding the granules. The universal occurrence of granules in living calls is certainly a fact which must be of great importance in any interpretation of the mechanics of protoplasm. Some protoplasm contains fibrils or strands. In the 18th century, the physiologist Haller held the doctrine that all living things were made up of fibrils or fibrae. This is a view that has reappeared from time to time in cytological literature. There can be no question but that muscles are largely made up of fibrils. The protoplasm of nerve fibers may also be fibrillar. Following the development of modern cytological technique, in which cells are treated with a variety of reagents before being sectioned, fibrils have been described in a great many types of cells. Whether these fibrils occur in the living or is, of course, an open question. From time to time in the history of cytology, there have been arguments as to whether protoplasm was more truly granular or fibrillar. Whether granules or fibrils were the ultimate units of the living material. It would scarcely be worthwhile to review these old arguments. As long as cytology remained a purely morphological study, it seemed of importance to discover some form element which could then be regarded as a unit and in a certain sense as an explanation of protoplasm. There are types of living protoplasm which do contain fibrils, but ordinarily when living cells are examined under the microscope, no strands or fibrils are visible. On the other hand, A11 or almost all living cells contain granules. and these are readily demonstrable without the use of any reagents or stains. Whereas fibrils are usually found only in highly differentiated types of living material, granules are of practically universal occurrence. In many cells, the granules are extremely small, barely visible under the highest powers of the microscope. It is probable that in some protoplasm, the granules may be still smaller and may thus not be detectable under the microscope. At least this much seems certain. Protoplasm is a colloid in which typically large numbers of the dispersed particles are of microscopic dimensions. In the language of the colloid chemist, it is a suspension. In a given cell, the visible granules are not always of the same type. Obviously, in most instances, it is not a simple matter to distinguish differences between granules so small as to be barely visible. Sometimes the granules are of two or more sizes. Thus, in the egg of the sea urchin Arbacea, in addition to granules about a third of a micron in diameter, there are a micron in diameter. At the small granules O, egg are also of two varieties. Most of them are he-pound rest of the protoplasm, some are lighter. When the fused, the heavy granules go to one pole, the lig to the opposite pole. See figure 1p45. Many other be shown to possess granules of fatty material, while then the rest of the protoplasm. Perhaps in some granules are really fluid and should be called dro. A particle is only a third of a micron in diameter, and I matter to decide its physical state. The fact that miles of cells do not fuse when thrown to one side, fuse is evidence that they are solid. But it is not CC dense for the failure of the granules to fuse midget one presence of a solid film. Small granules are certainly of two sorts, but there may really be iron types of granules, which with J-Risen methods we distinguish in the living cell. Differences in size and specific gravity are an means of distinguishing granules in cells, acaciae, are obviously colored. But this seems relatively rare. Cytologists have been in the habit of identifying in cells by their staining reactions. In teleoclassical cytology, there was much discussion of the various newels which might be found in Livewag cells. This is easily accessible, and it requires only brief mention. In general, staining reactions are apt to be decopted, but in a few cases it is impossible to decide chemical peak by microscopic color tests. Same substance in conditions may or may not take a Kortenstein. The older cytological knowledge regarding the dipsy of granules to be found in cells depends for the observations made on dead, that is to say. Fixed metallic salts, alcohol, and various other reagents in order to make sections of cells possible. It is such reagents are capable of producing granular PR in solutions of protein. That they can also protein formation in living cells can be shown by a simple E. When a sea urchin egg is centrifuged, some granules pole, others to the opposite pole. And between the HI stereoductals are continued, the morphology at Center 19 of granules is a hyaline region free from granules. On treating centrifuged egg cells with mercuric chloride, one of the common reagents used to fix cells, new granules appear in the hyaline protoplasm. It is therefore unsafe to draw conclusions about the granules of living protoplasm from the study of fixed protoplasm. This has been frequently recognized by cytologists, and there has been an increasing tendency to compare fixed preparations with the living material wherever this is at all possible. In 1897, the histologist Bexta coined the word mitochondria for certain types of granules, which he found first in rat spermatocytes and later in various other types of tissue. These granules often showed a tendency to form threads. Benda developed a complicated fixing and staining technique, which he believed to be specific for mitochondria. Later, it was shown that the same granules could be demonstrated by much simpler methods, and that in many cases the mitochondria of Benda were indistinguishable from the granules so often described by the older cytologists. At the present time, there seems to be some slight uncertainty as to just what are to be termed mitochondria, and in what respect they differ from the other granules of the cell. Some modern authors regard the dye Janus green as a specific test for mitochondria in living cells, see count by 24. It is generally agreed that in most instances, at least mitochondria in section cells only appear when acetic acid and strong fat solvents have not been included in the fixing reagents with which the protoplasm was treated. Agad 8 and various other authors have expressed the view that the mitochondria contain a lipoid constituent and that they are dissolved away by fat solvents. It might be possible to define mitochondria as granules composed in part of lipoids. This definition appears to have the advantage of precise statement, but it has not been universally accepted. At any rate, lipoid constitution is not regarded as an absolute criterion of mitochondria. Cytologists have identified certain types of granules as mitochondria, mostly on the basis of their general behavior toward various fixing and staining reagents. But at best, the mitochondria concept is not a very precise one. In the Aracia egg, the pigment granules are chromobophore stained selectively with Janus green, Wilson 26. They also disappear when the egg is treated with fat solvents. They thus satisfy two criteria Wallachia through the past have been used to distinguish mitochondria. As a matter of fact, it might not be inappropriate to regard these pigment granules as mitochondria, for in other eggs, pigment granules have been regarded as mitochondria by mitochondria specialists. See D. Beak, 15 JB 19. Curiously enough, although the pigment granules of the Aracia egg disappear in the presence of fat solvent, this is not due to the fact that they are dissolved. In some recent studies, for the most part unpublished, the behavior of the pigment granules has been followed both inside and outside of the cells, CP 229. Disappearance of the pigment granule in the presence of fat solvent is the result of a series of reactions, the first of which appears to involve the liberation of free calcium ion. If pigment granules in Arbacea are true mitochondria, it seems probable that the disappearance of mitochondria in fat solvents is not a simple solubility phenomenon. In 1898, Golgi described certain structures which appeared in nerve cells after these had been treated with a silver impregnation process. Following Golgi, similar structures were found in various other types of cells, and many authors have described what is now generally known as a Golgi apparatus. This may take the form of a network, or it may be granular. In every instance, the Golgi apparatus is demonstrated either by silver impregnation or by an osmic acid treatment. Until recently, the numerous descriptions of Golgi apparatus were all for fixed material, and there was no certain knowledge as to whether or not it might be present in living protoplasm. Rate, Bramble, and Gatenby 25 and Kabova 25 describe Golgi apparatus in untreated cells. The structures which can be seen only in fixed cells are relatively unimportant to the student of the mechanics of living protoplasm. When cells that are alive are observed under the microscope, they are seen to contain large numbers of granules. These may be of different sizes and of different chemical composition, but granules about half a micron in diameter are a very common occurrence. Such granules are found both in the cytoplasm and in the nucleus, although the nucleus contains relatively fewer granules and may appear quite free from them. The fact that small granules are so constantly present in the living substance is an indication that such a fine suspension of material represents a co-colloidal condition favorable for the life process. It seems certain that as the physiology of the cell becomes more clearly understood, there will be shown to be a definite dependence of many vital phenomena on the granular nature of protoplasm, on the properties which it possesses by virtue of the fact that it is a suspension. It must not be forgotten, however, that there are outstanding differences between different types of living substance. When any particular type of cell is investigated from the standpoint of the colloid chemist, an effort should be made to determine what is visible in the cell under the microscope. Granules of one sort or another, fibrils if they are present, all would doubtless have an effect on the colloidal properties of the ensemble. Chapter 4. Introduction concluded. The chemistry of protoplasm. Colloid chemistry is a branch of pH6. Through most of this book, the discussion will center around the physical properties of the living substance. But before entering this field, it may be well to review briefly the main facts about the chemistry of protoplasm. It will be necessary to consider not only the chemical constitution of the protoplasm, but also the reactions which occur in it. Naturally, no exhaustive treatment is possible, Huge volumes have been written about certain aspects of the chemistry of protoplasm. In this chapter, it will only be possible to outline a few of the essential facts, emphasizing here and there a point of especial interest to the student of protoplasmic mechanics. Just as the colloidal properties of inanimate colloids are profoundly modified by traces of one substance or another, so beyond any question, the colloidal properties of protoplasm are dependent in large measure on the ions and molecules which form a part of it. Moreover, the intricate complex of chemical reactions constantly going on in the cell must both influence the colloidal condition of the protoplasm and be influenced by it. What these influences are, we can, for the present, only vaguely hint at, but we can at least present the problems involved as our modern knowledge shows them. The student of the colloid chemistry of protoplasm is handicapped by the small dimensions of the cells, which contain the material he is interested in studying. But this is not his only handicap. In ordinary colloid chemical investigation, the main mass of the material is a known substance or substances. True, traces of unknown constituents may occasionally play a role in the colloidal behavior of an inanimate colloid, but in general, the colloid chemist can be fairly certain of what his test tube contains. On the other hand, the chemical makeup of protoplasm is extremely uncertain. We know a few broad facts, and that is all. For the most part, chemical analysis of the living substance is only possible under conditions in which the protoplasm is killed and doubtless changed during the course of the investigation. In general, the ordinary analytical methods are too drastic. When such methods are used, the protoplasm dies, and as it dies, a number of chemical reactions are initiated, which may, for all we know, have a profound effect on the chemical compounds of which the cell is composed. To some extent, these reactions at dying protoplasm can be retarded by cold, but they cannot be completely stopped. Some years ago, it was not uncommon to regard protoplasm, or the essential living part of protoplasm, as a single chemical compound of extraordinary complexity. This view was thought to find support in the older experiments of Heilbrunn and Pellegeb, which seemed to indicate that the metabolism of the cell was bound up with very complicated systems, rather than with the chemical transformations of relatively simple compounds. The idea that a single chemical compound of extraordinary complexity was responsible for the life of the cell was elaborated by E.B. Waben III in his well-known biogen hypothesis. Webb maintained that the biogen molecules were present in the ground substance of the protoplasm rather than in formed elements such as granules, etc. It was not present in the nucleus, but the nucleus contained materials for the maintenance of biogen metabolism. There is very little evidence in favor of the biogen hypothesis. In 1867, Heilbrunn concluded that muscular action depended on the breakdown and building up of a complicated nitrogenous compound. This he thought of as a very labile substance, which only remains intact at 0°C, and which at room temperatures disintegrates into carbon dioxide, lactic acid, and myosin. H.B. Mann regarded this substance as of the same order of complexity as hemoglobin, that is to say, he thought of it as a conjugated protein. But the recent remarkable advances in our knowledge of muscle metabolism have all tended to show that relatively simple compounds may play a very important part in the chemical reactions which go on within the muscle. Thus, carbon dioxide and lactic acid are formed from glycogen rather than from some complicated hypothetical substance. Just as it is unwise to regard certain morphological elements of the protoplasm as more truly alive than others, so also it is unwise to maintain that certain of the chemical constituents of the protoplasm are more intimately concerned with the life process than others. Protoplasm contains many complex materials of high molecular weight, and there is always a tendency to ascribe the attributes of life to these less known and thus more mysterious compounds. But the simpler constituents are just as truly a part of the living system as are the complex. We cannot conceive of a protoplasm without water and salts, and certainly these simple compounds form an indispensable part of the living material. In order to have before us a picture of the chemical constitution of protoplasm, it will be necessary to review briefly the elementary facts concerning the chemical materials found in living cells. We will consider first the evidence obtained from the investigation of large masses of cells, and second, the observations that have been made on individual cells in the hope of localizing definite compounds in various morphological elements of the protoplasm. In many types of living cells, the water content is approximately 75%. Resting plant seeds have a much lower percentage of water, and certain animals, e.g. rotifers, can be dried until their water content is practically negligible. Such dried animals are scarcely alive. Their protoplasm is in a quiescent state. As far as is known, active protoplasm is always rich in water. Besides water, the only common inorganic constituents are the salts of the alkaline and alkaline earth metals. Below are given, first, an analysis of the salts of maize pollen cells, and second, a recently published analysis of the salts of the sea urchin egg. These are merely two examples of many that might have been chosen. They represent analyses of cells rather than of tissues. This is an advantage in that tissues often contain lifeless connective tissue fibers, doubtless a very different chemical in the above analysis of the sea urchin egg. Sit2 was found to be present, but was not determined quantitatively. No distinction was made between the phosphorus combined with lipoid and the inorganic phosphorus. But Page indicates that about interesting in that he has calculated the molar concentrations of the various ions. Apparently, there is a much higher concentration of cation than anion within the cells. This also appears to be true for the pollen ash. If, from Page's data, one calculates the osmotic concentration of the salts within the sea urchin egg, it is possible to show that this concentration is very much higher than the concentration of the salts in the surrounding seawater. The fact that some living cells, and perhaps living substance, generally contains more inorganic cation than anion is very important. In the sea urchin egg, Page found the molar concentration of cation to be 10 times as great as that of A. The equivalent concentration was 6 times as great. Such an excess of cation may very well have a bearing on the colloidal properties of the protoplasm. The subject is in need of further elucidation. Doubtless, some of the excess cation is combined with protein. Some is probably combined via lipoid or fat. In the sea urchin egg, it is noteworthy also that there is a larger amount of bivalent cation, Ca and Mg, within the egg than monovalent cation, and this in spite of the fact that the surrounding seawater has only a relatively small amount of bivalent cation. Analyses such as the above indicate the actual constitution of one type of cell or another. Obviously, the inorganic salts which are found in a cell may vary somewhat as the environment of the cell or organism changes. By experimentally varying this environment, it is possible to obtain data with regard to the salts which are indispensable for the protoplasm of a definite species of animal or plant. For the most part, experiments of this sort have been made only with plants. Seeds can be grown in nutrient solutions which contain some elements and not others. In such nutrient solutions, if all the necessary elements are present, large normal-looking plants are obtained. On the other hand, in the absence of some needed element, dwarf pathological tides appear. Details concerning these experiments can be found in most books on plant physiology. Flowering plants require the following ions: K, Mg, CFA, istos, sulfur, PO4. Normal growth can be obtained without NaAlC. In the studies that have been made with flowering plants, some salts are of course contained in the seed, and this is a possible source of error. It is doubtful also whether the older experimenters were careful enough of the purity of the salts they used to make up their nutrient solutions. Often mere traces of a substance are important. Thus Peters 21 found that the protozoan Ulpidium was able to live in solutions which contained no potassium only if glass vessels were used. In quartz vessels, the animals died. Peters took this to mean that enough potassium dissolved out of the glass to keep the animals alive. For bacteria, yeasts, and molds, it has been found that the only cations necessary are K, Mg, and Fa, and there seems to be some question as to whether iron is essential or not. In experiments with these organisms, there is no source of error from the presence of a large seed. However, there is always the danger of impurities. The study of the elements which are required for animal life is often and very difficult. The higher animals are easy to rear on a pure chemical diet, but they are large at birth, and it is quite impossible to decide whether or not an element is necessary unless large amounts of it are required. Sometimes this is the case, and many studies have been made on the need of such elements as calcium and iodine. In these instances, the calcium goes largely into the bones and the iodine into the thyroid gland. So that such experiments are of little interest to the student of protoplasm, who is concerned chiefly with the inorganic needs which are common to cells in general. It seems not at all unlikely that experiments with lower animals might yield results of value. A few studies have been made with protozoa, and some work has also been done on Drosophila. The organic materials found in animals and EEI3 are of three main tissues: proteins, fats, and carbohydrates. To the fats should be added a heterogeneous group of fat-like substances, fat-like in view of the fact that they are generally insoluble in and chloroform. These fat-like substances are called lipoids. Concerning the present-day knowledge regarding proteins, fats, lipoids, and carbohydrates, abundant information will be found in any one of numerous textbooks or handbooks of biochemistry. From a physico-chemical standpoint, it seems especially noteworthy that all protoplasm should contain, in addition to water-soluble substances, others that are typically water-insoluble. We should expect that the fats and lipoids of living cells would often be found in the form of a suspension or emulsion. This apparently is true. In the pollen, the carbohydrate starch, saccharose, and the pentosines represent about 30% of the dry weight, whereas the amount of protein calculated from the nitrogen content is roughly 27%. Thus, in the plant cells, there is more carbohydrate than protein, whereas in the animal cells, there is almost 14 times as much protein as carbohydrate. In myxomycetic protoplasm, there is also a high percentage of vagisal, shows almost as much carbohydrate as protein. The chemical study of large masses of animate tissue or of large numbers of cells is relatively simple compared to the study of individual cells, or the attempt to identify chemical compounds within the confines of a single cell. In general, insoluble constituents of protoplasm would tend to be more localized in their distribution. Obviously, if a substance is highly soluble, we should expect to find it dispersed throughout the entire cell. But this need not necessarily follow. A soluble substance may be very largely adsorbed at some of the protoplasmic surfaces. It has been pointed out that protoplasm is typically granular, and the surfaces of the granules no doubt adsorb many dissolved substances. Often a cell contains various types of granules, and it is quite possible that the adsorption at the surface of one kind of granule may be quite different from that at another. In the attempt to identify chemical compounds within a living cell, one encounters a host of difficulties. The small size of the cell is not the only troublesome factor. Microchemical analysis has developed many methods for the identification of extremely small quantities of material, but microchemical analysis depends very largely in its identifications on the formation of characteristic crystals, which are then recognized under the microscope. Within a cell, or rather within the protoplasm, crystal formation is usually impossible. Generally speaking, normal crystals do not form in the presence of large quantities of colloidal material, and living cells contain high concentrations of various colloids. A few microchemical tests are color tests, cells. Unfortunately, the number of such tests is not very great, and not all of the tests that have been used are valid. Cytologists often assume without adequate proof that certain dyes are specific for definite chemical substances. In addition to color reactions, the student of cell chemistry can also make use of solubility tests. Cells are treated with fat solvents, with salt solutions, or with various enzymes, and the disappearance of one structure or another is regarded as evidence of its solubility. Inferences are then drawn in regard to the chemical constitution of the structure which was found to disappear. Solubility tests on protoplasm are not as reliable as they might seem. In the last chapter, an example was cited of certain granules which always disappeared in the presence of fat solvents, and which were apparently neither fats nor lipoids. The disappearance of these granules was the end result of a series of reactions initiated by the fat solvent. In a system as complicated as protoplasm, the addition of a foreign chemical may produce results which are difficult to interpret. Doubtless, however, many of the results that have been obtained with solubility tests are valid. For further and more detailed information concerning microchemical teats, see Peratt 27, compare also Klein 28, and Patzpel 28. The microchemical study of the cell has, among other things, given information regarding the distribution of glycogen, of fats, and of nucleoproteins within the cell. It seems fairly certain that the nucleus consists largely of nucleoprotein. As a matter of fact, nucleoproteins were first isolated from the heads of spermatozoa. Pats and lipoids are usually scattered through the proto-23 and are not limited to the surface layer of the cell, nor even especially abundant there. Heilbrunn 26b has suggested that irotoplasmic granules are typically surrounded by a lipoid or fatty film. The evidence for this view rests both on the colloidal behavior of the protoplasm and also on the fact that when sea urchin eggs are centrifuged, the granules heavier than the rest of the cytoplasm give a reaction with osmic acid. This indicates the presence of fats or lipoids with an admixture of unsaturated fatty acid in the granular constituents of the protoplasm, and presumably at their surface. Further evidence that the granules of protoplasm are typically surrounded by a fatty or lipoid film is given by the fact that many types of protoplasm resist the action of pepsin and trypsin, but are soon digested if their fats are first extracted. For literature on this subject, see Bietman 24. According to Biedman, there are two possible explanations of this phenomenon. One might suppose that the lipoid was combined with protein in the form of a lipothioprotein, or one might regard the lipoid as held by adsorption. Biedman states that the first possible explanation cannot very well be the true one, because of the fact that lipoids can generally be completely removed from cells lipoid solvents. There is therefore evidence from several directions in support of the view that protoplasmic granules are typically surrounded by a film of fat or lipoid. In subsequent, it is possible that the protoplasm may contain lipoids with a relatively high content in phosphorus and a specific gravity greater than the specific gravity of the cytoplasm of the sea urchin egg. The specific gravity of the cytoplasm must be over 1.03. Moreover, the granules in question have a specific gravity of at least 1.1 c-chap. 5. And it seems certain that no protoplasmic lipoids have a specific gravity as great as this. Chapters, it will be shown that this conception of the protoplasmic granny fits in with certain of the known facts regarding the physical behavior of the living substance. From the standpoint of cell chemistry, as well as from the standpoint of the colloid chemistry of protoplasm, one of the most important questions to be solved is the question as to the degree of acidity or alkalinity of the protoplasm. This problem is by no means a new one, but in recent years it has taken on a new significance. Many authors have attempted to determine the hydrogen ion concentration of protoplasm. Their results have varied not only with the type of cell study, but also with the method. For relatively simple systems under known conditions, the most accurate method of measuring hydrogen ion concentration is the electrical method. The solution to be studied is connected with a hydrogen electrode on one side and some standard electrode, such as a calomel electrode, on the other. The EMF of the cell is then measured, and from this EMF, the hydrogen ion concentration, or its negative logarithm, the pH, can readily be calculated. In determining pH electrically, one must be very careful to exclude any factors which may have an influence on the EMF. For instance, a hydrogen electrode may frequently serve as an oxygen electrode, or various substances can poison the electrodes. The electrical measurement of the pH of protoplasm is complicated by the fact that it is very difficult to devise electrodes small enough to be inserted into a living cell. This technical difficulty has been overcome by several recent W. Merkers. Thus, Teju and Wiyatakeb 27 have succeeded in inserting electrodes into the protoplasm. Their results, however, are very improbable, and Taylor and Whitaker do not regard them as a proper measure of the protoplasmic EF. Beery Tenduck and Wertheimer 27 have devised an antimony electrode, which they have introduced into frog eggs and similar objects. The results of by Tenduck and Wertheimer are certainly more within the range of probability than those of Taylor and Whitaker, but there is no means of knowing whether or not they are accurate. Many factors might influence the EMF of a concentration cell, one end of which is immersed in protoplasm. When a cell is cut or torn, it frequently forms films or membranes at its free surface. See Chat 13. There is good reason to believe that such a film or membrane might be formed about any object inserted into a cell, and the presence of a membrane might have a considerable influence on the EMF of any current which had to pass through it. There are also certain difficulties connected with the use of the antimony electrode. See Vlaz and Valashchub 27. Some workers have ground up cells and have determined the pH of the resultant mass. In view of the many changes which accompany death, one would have to be rather optimistic to assume that the pH of living cells could be measured in this fashion. Most of the measurements of protoplasmic pH have been made with iodicators. If cells are placed in solutions of various indicators, the amount of dye which enters depends both on the J-permeability of the plasma membrane and on the readiness with which the protoplasm combines with the dye. An indicator may be either adsorbed or it may be bound chemically. Naturally, the concentration of the dye within the cell would be very different from its concentration in the surrounding solution. The intensity of its color will therefore be quite different, and one is apt to make errors if one attempts to estimate pH from color intensity. Some authors have tried to control the amount of indicator in the cell by injecting indicators with micropipettes. Perhaps more reliable results can be obtained in this way, but it is always difficult to control the amount of dye injected, and there is always the chance of violent injury. It seems obvious that the most reliable results can be obtained with indicators that undergo a complete change of color rather than with those that merely change their shade. An indicator may be light red or dark red either because of a difference in pH or because of a difference in concentration of the indicator. And the concentration of the indicator is not. But when an indicator changes from red to blue, the concentration does not so much matter. For this reason, some investigators have emphasized the importance of using indicators which change color, and it is obviously an advantage to use as many different indicators as possible. This point of view has been most often stressed by Small and his collaborators. See Small 26, Hay and Small 26, and various other papers in volumes I, Protoplasma. Unfortunately, Small and his co-workers have used alcoholic Inadachia concluded the chemistry, etc. 33 solutions of indicators, so that their results are valid for dead cells rather than for living cells. Small claims that his results can be duplicated with aqueous solutions of the indicators, but Nyene or most of the dyes he uses are strongly toxic, so that even in aqueous solution they kill the cell. Since the above was written, Ischiamal has recognized the error due to the presence of alcohol and has attempted to devise a new and improved technique. See in Gold and Small 28. In view of the uncertainty with regard to the various methods which have been used for the determination of the pH of living cells, it is difficult to decide which values are the more likely to represent the nearest aptioch to the truth. In their work on the starfish egg, Chambers and Pollack, 27 used a number of indicators and observed color changes rather than mere difference in intensity of shade. They found the pH to be 6-7. Indeed, many workers have found the pH of living cells to lie somewhat on the acid side of the neutral point. However, the results of various investigators are far from concordant, and we have as yet no means of knowing whether this discordance is due more to differences in the hydrogen ion concentration of different types of protoplasm, or to errors in method and observation. Or references to literature on intracellular hydrogen ion concentration. See Pleiad 20 Rias 26. An interesting recent paper is that of Velling 28. They used the natural pigment of Thierbasia egg as an indicator, and they were thus able to determine the pH without crushing the eggs and without injuring them by microdissection. Examine spectroscopically, the pigment Owa Aracia egg shows different absorption bands at different hydrogen ion concentrations. From their studies, Vlelas and Yelling EB concluded that the pH of the Aracia egg cytoplasm in the neighborhood of the pigment granules was between 5.2 and 5.9. In all types of protoplasm, there are many and complicated chemical reactions. Of these reactions, the ones most often studied are the oxidation-reduction reactions. From a physico-chemical standpoint, oxidation involves the loss of an electron or electrons from a molecule. By the study of electric oxidation reduction cells, it is possible to express the tendency for a substance to become oxidized or reduced in terms of electrical units. The measure of this tendency is the oxidation potential. It is to be hoped that protoplasma monographically in the future, accurate determinations of the oxidation potential of protoplasm will be possible. Thanks to the work of Clark, a beginning has already been made, as can be seen from the review of J. and B. M. Needham 26. But in the words of these authors, much investigation and much thought will be needed before the nature of the assumptions required in the application of oxidation reduction potential to living systems is accurately understood. An important recent contribution is that of Cohen, Chambers, and Resnikoff 28. Oxidation reduction reactions are not the only type of reactions which occur in protoplasm. Proteins are broken down or built up, fats are hydrolyzed, glycogen undergoes various transformations. Probably a cell contains at least 20 or 30 types of enzymes, capable of furthering particular chemical reaction. It is difficult to understand how so many different reactions could go on more or less simultaneously within the narrow confines of a droplet of protoplasm. As a formal explanation of this difficulty, Hofmeister 1 proposed the theory that the col was divided by partitions into many small chambers. Various enzymes were thus separated from each other, and each controlled its own type of reaction in its own allotted space, never diffusing into a neighboring compartment. Although the theory of Hofmeister has been accepted by many, it certainly cannot apply generally, as will be shown later. See. 5. Some types of protoplasm have a very low viscosity, and this would be quite inconceivable if the cell were divided up by partitions. On the basis of our actual knowledge concerning the morphology and the physical properties of protoplasm, it is much more logical to assume that the chemical reactions of living cells occur most frequently at the surfaces of protoplasmic granules. The conditions for chemical reaction are especially favorable at surfaces, and this is particularly true for enzyme reactions. The work of Warburg, see for example Warburg 21, offers definite evidence that certain reactions which occur in living systems are really surface reactions. Chapter 5. Methods of Study. Natural science depends on measurements. If the colloidal study of protoplasm is to prosper, methods must be developed which will enable the biologist to determine roughly, or if possible quantitatively, the colloidal properties of the living material. In a few cases, the ordinary technique of the colloid chemist can be applied to the living cell, but in order to answer many important questions, one must employ methods specially adapted to the peculiar form and delicacy of protoplasm. Of the methods that have been used, not all are good. Obviously, no scientific method is without its limitations. One could hardly expect any great degree of accuracy in the physical determinations made on living cells. An inaccurate determination is certainly better than no determination at all. But it must be remembered that subjective impressions are more apt to hinder scientific progress than to advance it. And in its beginnings, the colloid chemistry of protoplasm has had to suffer a great deal from the hastily published guesses of its pioneers. There are 3 types of methods that have been used in the colloidal study of protoplasm: optical methods, methods of viscosity determination, and electrical methods. The classification is not an exact one. Indeed, one type of viscosity determination is almost purely an optical method. In the last analysis, practically every colloidal investigation of the living cell involves oidicial study, for without a microscope, the cell would typically be invisible. Mere examination with a microscope can give some information about the properties of the cell as a colloidal system. The sizes of the various granules in the protoplasm are important. items which can be decided by jet microscopic study. Occasionally, the appearance of new granules or the disappearance of some already present may be seen with the ordinary microscopic lenses. Sometimes new films or membranes can be observed in process of formation. But as long as the biologist depended entirely on what he saw in the cell, his knowledge of its colloidal properties remained very slight. With a microscope alone, one can, in general, obtain only scant information regarding the physical properties of 2-DRO-2 Lazanda. Nevertheless, the many authors have not hesitated to express bold and certain 2 annuities on the basis of pure morphological observation. But what to one author looks like coagulation or gelation may to another seem like a chelation. Protoplasm is an extremely intricate system, and frequently one is led astray by its complexities. Great care must be taken in interpreting observations. At least one writer has regarded the increase in volume of a cell as evidence of its chelation or liquefaction. This may at first sight seem plausible, and yet frequently increase in volume of a cell is accompanied by a too renounced gelation. It is quite possible that in some types of living material, careful observation with an ordinary microscope may give very valuable information concerning colloid chemical processes. Thus, in cells relatively free from granular material, it may be possible to observe the formation of new granules or strands. It might also be too possible to note gross changes in the refractive index of the protoplasm. But certainly in the past, purely morphological study of living cells has yielded very little definite and indisputable evidence concerning the colloidal properties of the living substance. This may have been due in part to the inability of the older cytologists to interpret their observations too early from a physical standpoint. But the main difficulty lies in the fact that two physical changes in the protoplasm of a cell are frequently quite invisible with an ordinary microscope. Some cells may become completely coagulated or gelatinized without any obvious change in their appearance. In recent years, the colloid chemist has found good use for the polarization microscope. With it, he has often been able to demonstrate shotpiece and arrangements of colloidal micellar. Fortunately, the externalization microscope can also be used on biological material, and it frequently has been. Indeed, long before there was a science of colloid chemistry, biologists were well acquainted with the polarization microscope, and some of them contributed to the theory of its use. With the development of staining and sectioning methods, interest in the polarization microscope waned as biological workers became more and more involved in all the complicated technique of classical cytology. But even though stained and colored cells show many details which invite description, such cells are of course very much dead. And in recent years, there has been a pronounced reaction against the type of cytology which makes no attempt to investigate the cell as a living object. New instruments for examining the living cell have been sought for. In late years, stimulated perhaps by the discoveries of the colloid chemists, biologists have again started to examine animal and plant material under crossed nickels. A book by W.J. Schmidt, 24b, gives a resume of old and new work in this field. Most of the researches have been made on inanimate fibers, but some observers have examined living muscle and also other types of protoplasm. The polarization microscope is a complicated instrument, and its use could hardly be explained in a page or two. The recent book of Ambacon and Frey, 26, provides a Feral 3 complete manual. It gives details both as to theory and practice. There is a pamphlet written by Schmidt 24a, which is simpler but not so inclusive. In the main, a polarization microscope is an ordinary microscope provided with one nickel prism below the object and one above. There is also provision for inserting thin I3/8 of quartz, mica, etc., of various thicknesses and of various optical properties. When an object is viewed under crossed nickels, it appears black if it is isotropic, no matter in which position it lies on the stage of the microscope. Anisotropic or doubly refractive objects appear bright under crossed nickels, and as they are rotated on the stage of the microscope, there are two positions in which they appear brightest and two in which they appear darkest. To be studied profitably under a polarization microscope, an object must be anisotropic. This anisotrogy may depend on the presence of fibers or plates, or it may be due to the fact that the body is subjected to greater pressure in one plane than in another. The fact that fibrils show anisotropy explains why muscles and nerves are such excellent objects for study under the polarization microscope. Unfortunately, undifferentiated protoplasm is typically isotropic, CP97, and it is thus not possible to study it with the polarization microscope. In the development of the science of colloid chemistry, the ultramicroscope has played a very important part. This instrument makes possible the study of particles smaller than those which can be seen with the ordinary microscope. The principle of the ultramicroscope is quite simple. Ordinarily, light is sent in parallel rays through the tube of the microscope. But in ultramicroscopic study, the object is illuminated from the side, and the image which is seen by the observer is not formed by the absorption of light, but rather by the rays which are diffracted from the surface of the object. Various textbooks of colloid chemistry and of microscopy give information regarding the theory of the ultramicroscoid. Reference may also be made to the papers of Sidentopit 7AB89. In ultramicroscopic study of protoplasm, either a paraboloid or a cardioid condenser is ordinarily employed. In the use of the ultramicroscope for biological study, it must be remembered that the protoplasm is not free, but that it is surrounded by a membrane or membranes. This introduces one complication, and there are others. Apparently, only certain material is favorable for study. The following quotation from P. Bysum is instructive. Considerable difficulty is experienced in selecting material suitable for observation and experiments in various directions. Gajnik-Joukov showed that only in certain cases was good observation possible. The considerations affecting the suitability of material are roughly as follow. The tissues or structures must generally be only one cell in thickness. The walls of the cells must be optically homogeneous. The diameter of the cell or cell filament must not be so small that the diffraction image of the walls interferes with observation of the cell contents. And the cells must not contain a large number of bodies of the nature of chloroplasts, as these, by scattering the light, interfere with good observation of the more minute structures. Some authors, in using the ultramicroscope, have failed to recognize the limitations of the method when it is used on living material. And they have sometimes drawn too hasty conclusions regarding the presence or absence of colloidal particles, or the geophysical state of the protoplasm. When a cell is surrounded by a membrane or wall, which appears bright in the Darldield, great caution must be exercised in interpreting results obtained from a study of the interior. As in Exam 2, law of the inadequacy of the method under these conditions, it may be noted that in salamander blood cells, according to Agazzati 10, the nuclei of the cells are almost or completely invisible. In the cells are examined under Darlhield. In Agazzati's figures of these cells, he shows no nucleate. In this case, the light entering the field from the side is largely diffracted from the surface of the cell. Under conditions of this sort, the visibility of structures within the cell depends largely on the amount of light that enters. Changes in the physical structure of a cell wall or membrane may very readily have an effect on the amount of light that is able to enter the cell. In a recent Geiger, Estuago 26 warns against interpretations arrived at by use of the dark field. That is to say, the ultramicroscope. Especially is this true in the study of the nucleus. Frequently, owing to the brightness of the nuclear surface, the interior of the nucleus appears optically empty in Darldield. Although study under ordinary conditions of illumination clearly shows the presence of structures within it. In study among the colloid chemistry of protoplasm, it is of great importance to be able to detect physical changes in the living substance. No property of colloidal systems is of more importance than the viscosity, and it is very fortunate that in recent years methods have been developed which make it possible to estimate and even to measure the viscosity of the protoplasm. In most experiments, it is only necessary to determine the viscosity at a certain time or under a certain set of conditions. If, On the other hand, one wishes to compare the viscosity of protoplasm with the viscosity of inanimate fluids, it is essential to know the viscosity in absolute units. The measurement of the values that have been obtained have varied with the type of method employed. In the next chapter, the whole subject of absolute viscosity determination will be examined critically, and for the present we shall consider only the measurement of relative viscosity. When one crushes a cell under a cover slip, often the protoplasm emerges and flows into the surrounding medium. The speed of outflow depends on the viscosity of the protoplasm, and by observing cells subjected to pressure, several EAE observers were able to estimate protoplasmic viscosity. See Reike 98. Unfortunately, when protoplasm emerges from a cell, it is no longer in a normal state. It may become mixed with the surrounding medium. Frequently, as the protoplasm starts to emerge from the cell, it becomes surrounded by a film or membrane. See Chap. 13. The nature of this film or membrane varies under different conditions. Under certain conditions, it forms much more rapidly than under others. There are thus two factors which govern the speed of outflow of protoplasm from a cell. One is the viscosity of the protoplasm. The other is the reaction which the protoplasm undergoes when it comes in contact with the outer medium. This second factor can be very important. Thus, if one crushes Arbacia eggs in seawater and in isotonic sodium chloride solution, the speed of emergence of the protoplasm is much greater in the second case. This is clue not to a lower viscosity, but to the fact that in seawater, Arbacia egg gyrotoplasm forms a film or membrane as it emerges, and no such film forms in isotonic sodium chloride solution in the absence of calcium. CP 222. In recent years, many workers have studied the viscosity of protoplasm, BG inserting fine glass needles into living cells. This Majorata section method was introduced by Kite T. Pi employed a Barber 2 pipette holder for the mechanical manipulation of the needles. Following Kite, more perfect apparatus has been developed, and microdissection instruments are now to be found in most biological laboratories. The two most common types are the Chamby's microdissection apparatus and the Petri micromanipulator. The former is sold by Lights, the latter by Zeiss. Other types of apparatus have also been described. Bishop and Thaleson 21st, Taylor 25C. For a general discussion of method, see Chambers 22, Peterfee 2427 BI. When a cell is to be dissected, it is placed in a small hanging drogillet of fluid in a moist chamber, and is there operated upon. Beyond any question, the microdissection, microinjection methods have found many uses in biology, and will doubtless continue to find many more. For the measurement of viscosity, however, the microdissection method can at best give only indications of gross differences in viscosity, and even for these, it is more or less uncertain. Microdissectionists themselves now generally believe that the introduction of a needle or a GPET into a cell produces injury. See, for example, Peterfee 27, Chambers and Pollack 27, Howland and Pollack 27. Such injury might conceivably cause either increase or decrease of viscosity. It is sometimes a little difficult to understand just what criteria are employed for viscosity determination by the users of the microdissection method. In some instances, the experimenter seems to rely most on his general impressions as he watches the needle pass through a cell. In many cases, apparently, the viscosity is judged largely from the speed of flow of protojolasm from injured cells. Thus, for example, Spiabi's 26b states, owing to the extraordinary thinness of the cytoplasmic layer lining the wall of the amphibian red cell, it is quite impossible to obtain any direct evidence of the consistency of the protoplasm. The cytoplasm cannot be made to flow out of the corpuscle and retain its identity, as is possible in other types of cells, and thus permit a determination of the consistency with a fair degree of accuracy. For some microdissectionists, the fact that certain types of protoplasm can be cut into discrete fragments is taken as evidence of high viscosity. If the microdissection makes viscosity determinations on the basis of the speed of outflow of protoplasm from a cell, or on the basis of the fact that some cells can be cut into fragments and others not, then such determinations are subject to the same objections already considered for the case in which the protoplasm is squeezed out of the cells. Layer 2, just as in the case of the cells crushed under a cover slip, the reaction between the naked protoplasm and the surrounding medium certainly plays an important role. Consider, for example, the case I previously mentioned of Aracia eggs in isotonic sodium can be cut into discrete fragments, whereas in the isotonic sodium disperses as soon as the cell is cut. To a microdissectionist, this would mean that the protoplasm was more viscous in seawater, which is quite the oeithreposit of the truth, see chap 9. The fact that the cells can be cut into discrete fragments in the presence of calcium and not otherwise is due to the fact that the formation of a film or membrane around the naked protoplasm depends on the presence of calcium, see chap 13. In 1920, Spiekes made an atomizite to put the microdissection method of measuring viscosity on a semi-quantitative basis. He arranged a series of 10 viscosity values which he based on the viscosity of various known substances. These are shown regarding the gelatin he used in his comparison values. Spiekes says that it formed a soft jelly at 18C when the concentration was as low as 0. Presumably in concentrations lower than this, the gelatin remained fluid. In a recent paper, Heilbrunn 27A, the following statement was made: that anyone should be able to measure viscosity accurately by observing the passage of a mechanically controlled needle through protoplasm seems on the face of it unlikely. It would be easy to put the matter to a test if a microdissectionist were given certain unlion samples of fluids. It would be interesting to see how his estimates of their viscosity compared with the known values. When the above sentences were written, it was not realized solutions of gelatin so dilute that they do not gel have a viscosity only about twice of water. True, the viscosity varies at different rates of shear, see for example Pionlik and Abimson 27, and the rate of shear in Spiebes's needle tests is not defined. But even at very low rates of shear, the viscosity of dilute gelatin soles is probably not over 3 or 4 times that of as equal to that of glycerine, which has a viscosity approximately 1,000 times that of water. A method which even occasionally permits of an error of 20% can scarcely be regarded as a reliable method. As early as 1880, it was known that the starch grains of plant cells moved under the influence of gravity and became accumulated in the lower part of the cell, see Denicky 80, Hein 85. Niemiola noticed that the speed of fall of such grains was much slower at a temperature of 6 degrees Celsius, and he suggested that this was due to an increase in the viscosity of the protoplasm. Heilbrunn 12:14 first proposed to use the speed of fall. Of matter of fact, the speed of fall of spheres through fluids is frequently used by physicists and physical chemists as a measure of the viscosity. And not a few viscosimeters have been constructed for the purpose of determining viscosity of inanimate fluids in this manner. The principle governing all these measurements finds mathematical expression in the well-known equation of Stokes. This equation will be considered in the next chapter, for in the determination of absolute viscosity, certain corrections must be applied to it. These corrections are much less important for the measurement of relative viscosity, and for the present we will assume, as Heilbrunn does, that the viscosity is strictly proportional to the time of fall of particles through a fluid. In order to measure the time of fall, Heilbrunn cut longitudinal sections through young seedlings and placed them on the stage of a microscope, tilted in such a fashion that the stage was vertical. The sections were cut in such a way that at least one layer of cells remained intact. Care had to be taken to study none but intact cells. Even in these, the protoplasm showed effects of the sectioning process. Immediately after sectioning, the viscosity was high due to the stimulation of the wound. After 10 or 15 minutes, the viscosity decreased rapidly, remaining low for perhaps 3 hours, after which there was a pronounced viscosity increase, which Heilbrunn interpreted as due to the death of the cells. Beyond any question, the gravity method of measuring protoplasmic viscosity is a very valuable one, it must nevertheless be admitted that the cells in thin sections of plant tissues are far from being in a normal state. The very fact that they die within 3 hours is an indication that they are not in the same condition that they were in before sectioning. Moreover, the effect of wounding may be a serious complicating factor, compares Zollicott 18. The use of the gravity method, it would seem far better practice to expose the intact plants or tissues to the action of gravity, and then to cut sections later. In this way, any effects of wounding would be avoided. As a matter of fact, Nemec as early as 1901 made a few viscosity tests in this way. Recently, Buonuno Toinea has also used the gravity method on intact masses of tissue. Certain types of plant material are more favorable for study by the gravity method than are others. In the opinion of Professor Weber, who has used the gravity method more than any other worker, starch sheath cells are not as favorable as cells richer in protoplasm. For example, root cap cells, or cells of the coleoptile of wheat seedlings, personal communication. The granules and inclusions of animal cells do not ordinarily move under the influence of gravity, and the movement when it occurs is usually very slow. It is advantageous, therefore, to subject these cells to a stronger force than that of gravity. With a small hand centrifuge or with an electric centrifuge, a centrifugal force can be developed, which may be thousands of times as great as gravity. Fortunately, living cells can be subjected to centrifugal force without injury. Many workers in the field of experimental embryology have used the centrifuge in the attempt to analyze the developmental potencies of various visible structures of marine ova. For literature, see McCoane 27. In these experiments, granules were shifted to one side or the other of the egg, so that later definite regions of the embryo contained certain grannies and not others. In most instances, in spite of the abnormal distribution of the granular material, the embryos develop perfectly normally even after prolonged and drastic centrifugal treatment. The embryologists who centrifuged eggs frequently noted that the granules moved more readily at one stage than at another. Generally, they referred these differences to differences in the viscosity of the protoplasm, but they made no systematic observations on the viscosity differences, and their published statements consist of a few scattered remarks. Only in relatively recent years has it been realized that the centrifuge method is an extremely important means of determining the viscosity of living protoplasm. As has already been mentioned, the speed with which a particle moves through a fluid varies inversely with the viscosity. For relative measurements of viscosity, it is only necessary to determine the speed with which the protoplasmic granules move under the influence of a given centrifugal force. In this way, for a particular type of protoplasm, one can discover how the viscosity changes from time to time, or how it changes when the protoplasm is treated with various reagents. In the next chapter, it will be shown that absolute viscosity can also be determined by the centrifuge method. Some further details regarding the theory of viscosity measurement by the centrifuge method will then be considered. The use of the centrifuge for viscosity Dag 1a centrifuged egg of Arachia after Lyon 7 and Morgan, and Schrunkir 9 of Cumingia after Morgan 2. Determination requires no technical skill. Measurements can be made in a relatively short time if one only wishes to know whether the viscosity is increased or decreased by a given reagent. It is merely necessary to centrifuge treated and untreated cells simultaneously in separate tubes for an appropriate time, and then to note the degree of granular movement under the microscope. On the other hand, if one wishes a more exact measure of the change in viscosity produced by the reagent, it is necessary to make a series of tests in which the cells are centrifuged for varying intervals of time. In this way, it is possible to determine how many seconds are required to move the granules a certain distance through the cell, and this gives the speed. A few practical hints with regard to the centrifuge method measuring protoplasmic viscosity are given in a recent paper, Heilbrunn 26a, in order to obtain an idea of what happens when cells are centrifuged. Reference may be made to pig. 1. This shows a centrifuged egg of the sea urchin Aracia and a centrifuged egg of the clam Cumingia. The use of the centrifuge method is not limited to animal cells. polio wing some older experiments in which the centrifuge WSEA is used for other perthrosis, DNBP80, MODIB99, and BUS3. In recent years, various observers have determined viscosity in eukaryote cells with the aid of centrifugal force. The first worker to employ the centrifuge for the measurement of the protoplasmic viscosity of plant cells was Sjost 13. One advantage of the centrifuge method lies in the fact that it can be used on relatively large masses of material. Whole animals can be centrifuged, and their tissues can then be fixed and sectioned in order to determine the speed of granular movement in the individual cells. In such tests, rapid fixation is essential, for the granules following centrifugal treatment slowly return to their original positions. It is doubtful if results can ever be obtained with small cells. For in such cells, the proximity of the cell wall exerts a retarding influence on the movement of the granules, and so does the nuclear membrane if the nucleus is of relatively large size. In large nuclei, it is sometimes possible to observe movements of nucleoli or chromatin under the influence of centrifugal force, so that it might be possible to determine the viscosity of the interior of the nucleus. Until now, no such determinations have been made. In using the centrifuge method to measure relative viscosity, it must be remembered that the speed with which the granules move depends not only on the viscosity, but on various other factors as well. Were the size of the granules to change, or their specific gravity, this would make a difference in their rate of travel. Various reagents might very well alter the size or the specific gravity of protoplasmic granules, and the observer should be on his guard against the possibility of such an effect. Changes in size, if appreciable, ought to be visible under the microscope. In those instances in which cells contain both heavy and light granules which move to opposite ends of the cell and being centrifuged, the possibility of error due to changes in the specific gravity of granules can usually be excluded. For if both types of granules show the same variations in speed under different conditions, one can be quite certain that the differences are due to viscosity and not to changes in specific gravity. Suppose, for instance, that a reagent caused an increase in the specific gravity of the heavier granules. It would be quite improbable that the same reagent would cause an exactly comparable decrease in the specific gravity of the lighter granules. And yet, unless such an equivalent decrease occurred, the movement of the lighter granules would be relatively slower. To some authors, e.g., Chambers 21-24 first imprint, but not second, it has seemed that centrifugal treatment of cells might very well alter the viscosity at the movement of granules through the protoplasm, might tend to injure or to HQFE it. An effect of this sort would not have as much importance for measurements of relative viscosity. If a given sort of protoplasm were centrifuged at 2 times, or under different conditions, if it were more viscous at one time than at another, it would offer more resistance to the movement of granules, even if this movement did gradually cause liquefaction. As a matter of fact, the only actual tests that have been made indicate that the movement of granules through the protoplasm has no liquefying action at all. It is certain that if such a liquefying action occurred, or if the centrifugal treatment broke down fibers or a network in the protoplasm, then centrifuge tests at a higher rate of speed should give lower viscosity value. Or if the shearing force of the granules is greater, then the viscosity should be less, on the assumption that the movement of the granules introduces a lowering of viscosity. But as a matter of fact, tests both with Aracia and Cumingia eggs show 7 that no matter how fast or how slow the centrifuge is turned, the same viscosity values are arrived at. The data for the Cumingia egg will be presented in a later chapter, CP94. The use of the centrifuge method for the cells of the alga Spiragira presents a crucial case, and in view of the fact that these cells have often been used in viscosity measurements, a few words regarding them will not be out of place. Spiragira cells, as everybody knows, contain large spiral chromatophores. It is the speed of displacement of these chromatophores that is used as an index of protoplasmic viscosity. The physical structure of the Spiragira cell is not well understood, but we can with justice assume that the chromatogora lies loosely embedded in a layer of protoplasm which completely lines the cell wall. Under the influence of centrifugal force, the chromatophore tends to move past the cell wall, from which it is separated by a layer of fluid. Viscosity is defined as the resistance offered by a fluid lying between two planes to the movement of one plane over the other. Obviously, the speed of movement of the chromatophore in the Essen-Rogira cell is an inverse measure of the viscosity of the protoplasm in which it is embedded. If the measurement is to be exact, it is of course essential that the chromatophore does not itself undergo a marked change in physical state. A very useful method for the measurement of protoplasmic viscosity is the determination of the speed of Brownian movement of granules in the cytoplasm. In comparatively recent years, this method has been used by Chiflot and Gaudepiep 5, by Leblanc 19a, B by Babis 20 and others, and there is apparently a general belief that the use of Brownian movement to measure viscosity is a 20th century development. As a matter of fact, soon after the middle of the 19th century, many biologists made observations on Brownian movement in living cells, and at least some of these workers regarded Plier observations as a source of information regarding the efficacity or viscosity of the protoplasm. Thus, Libby Eck in 1870 begins a long paper with the following sentence. In den nachfolgenden Mitteilungen wird vielfach die Brownsche Molekularerregung benutzt, Fieber die Anwesenheit leichtflüssiger Substanzen. In general, the observations that have been made on Brownian movement are not very exact. The movement is seen to be vigorous at one time and less vigorous or absent at another. There have been no two published papers on the actual speed of the granules, and measurements of such speed would usually be very difficult to make. However, in some cells it is possible to follow the movements of a single GX granule, and in this way to determine the speed. In other cells, the granules are so crowded together that etch more exactly according to Maxwell. The viscosity of a substance is measured by the tangential force on unit area of either of two horizontal planes at unit distance apart, one of which is fixed, while the other moves with the unit of velocity. The space between being filled with the viscous substance, no observation of the Brownian movement is possible. This is often the case in marine eggs. In such instances, the speed of Brownian movement may sometimes be determined by estimating the speed with which the granules return through the cell after they have been centrifuged into one end. An example of such a determination will be given in this chapter. In 1922, Heilbrunn described a new method of viscosity measurement, the magnetic method. Small iron particles were introduced into the protoplasm of slime molds, and the protoplasm was then placed in the magnetic field of an electromagnet. In his first experiments, Heilbrunn determined the distance from the magnet and hence the magnetic force necessary to cause a barely perceptible movement of a small iron sphere. Later, he studied the twisting of iron rods within the protoplasm. As Heilbrunn himself points out, these methods are only possible for large masses of protoplasm. Details regarding Heilbrunn's measurements will be found in the next chapter, CP 62. Series 24 modified Heilbrunn's method. He used nickel particles and injected them with the aid of a micropetta. In order to measure viscosity, Series determined the speed with which the nickel particle approached the electromagnet. For a further discussion of Series' experiments, CP 64, still another method of viscosity determination is the plasmolysis form method recently advocated by Weber, see Weber 21, A, 24A, CD 25A, B, CD. When plant cells are plasmolyzed, the border of the cell may be perfectly smooth, in which case the plasmolysis form is said to be convex, or it may be angular, echic. When the border of the cell is extremely uneven, the plasmolysis is said to be a Krampfplasmolyse. Sometimes the term concave is used as opposed to convex. For illustrations of various types of plasmolysis form, see Figure 2. The reason for the different types of plasmolysis under different conditions was at first uncertain. It seemed quite probable that surface conditions might play an important role. However, Weber has shown that in general there is a perfect correlation between the type of plasmolysis and the viscosity of the interior of the protoplasm, as determined by the centrifuge method. With low protoplasmic viscosity, convex plasmolysis occurs. As the viscosity increases, the plasmolysis becomes angular or echoic, and with still higher viscosity, cramped plasmolysis appears. It seems protoplasma monogrammi Heilbrunn 4. Fairly safe, therefore, to use the plasmolysis form as an indicator of the relative viscosity of the interior protoplasm. The plasmolysis Pig 2 diagrams of plasmolysis form. A. Spirogel semidiagrammatic chloroplast not shown. 1. Convex. 2. Echoic. 3. Concave, cramped plasmolysis. Epipleum, concave plasmolysis. After Pringsheim 54. C-dentaria, concave plasmolysis. After Hegeli 55. These diagrams were made under the direction of Professor Weber-Dohr. Form method of estimating protoplasmic viscosity has the advantage that it is very simple, and that it can be employed on some types of material that are not easily tested by other methods. In many plant cells, the protoplasm is more or less continually in a state of flow. Botanists have frequently measured the rate of such flow under various conditions, and the literature of plant physiology contains numerous valuable observations of this sort. It is beyond any question logical to assume that the speed of protoplasmic streaming decreases with increasing viscosity. But if, under a given set of conditions, it is found that the rate of protoplasmic streaming decreases, one can never be certain that the effect is due to an influence on the viscosity or to a direct influence on the force or forces which occasion the protoplasmic streaming. Until these forces are known, we can only regard the data on protoplasmic streaming as suggestive. In this connection, it is interesting to note that 14 measured both the rate of protoplasmic streaming and the viscosity of the protoplasm in the cells of longitudinal sections of the stem of Ida faba seedlings. In such cells, the viscosity decreased for 2 hours and 20 minutes, and then underwent a sharp increase for an hour and a half. The speed of protoplasmic streaming increased with the decrease in viscosity and at a corresponding rate. Then, however, the speed of protoplasmic streaming began to decrease, even though the viscosity of the protoplasm was still decreasing. It is thus obvious that the speed of protoplasmic streaming is not always a perfect measure of the viscosity. Belehradic 2425 has attempted to obtain a more accurate measure of the viscosity from the speed of protoplasmic streaming. It has long been known that in the moving protoplasm of a plant cell, the tiny granules move at a faster rate than the chloroplasts. Babik believes that the ratio of the speed of the colloid to that of the tiny granules is a measure of the viscosity. In support of this rather unusual idea, Babik states that with increasing viscosity, the protoplasm would tend to pull along the chloroplasts, and W would more and more prevent them from being retarded by the cell wall. In the standard definition of viscosity, WUQA conceive of two parallel planes, each of unit area as being of unit distance from each other, and separated by a given substance. The retardation which one plane suffers in moving over the other is a measure of the viscosity. Obviously, from this definition, with increasing viscosity of the protoplasmic fluid between cell wall and chloroplast, and between cell wall and granule, there will be an increasing retardation of the speed of the chloroplast or granule, and this retardation will be greater the greater the surface of the chloroplast or granule. This is evidently a very important factor, but it has been completely neglected by Belehradic, who seems to think that the greater the viscosity, the greater the speed of the chloroplast. As a matter of fact, Belehradic himself states that it is almost impossible to determine in any rigorous manner in what manner the viscosity of the protoplasm depends on the ratio of chloroplast speed to granny speed. Not being able to solve the problem, Belehradic is content simply to divide chloroplast speed by granule speed, and to regard this simple fraction as a measure of the viscosity. Brahm W. Hat has gone before. It appears somewhat doubtful if it would not be better to divide granny speed by chloroplast speed instead of chloroplast speed by granule speed. Even more unconvincing is the method of viscosity determination proposed by Belehradic in 1926. By measuring the rate of biological processes at two different temperatures, He hopes to estimate the viscosity of the protoplasm on the assumption that the speed of the process is governed by one chemical reaction, which obeys the formula he proposes. The various complications involved in the interpretation of temperature coefficients have to some extent been outlined in a short note. Heilbrunn at 25e, to ignore these complicating factors, to assume one reaction instead of many. To assume, moreover, that colloidal viscosity acts in the same fashion as the viscosity of true solutions on the rate of chemical reaction. All these assumptions may lead to a formulation which omits a viscosity measure. But for the present, at least, until the nature of the assumptions and their justification is more clearly understood, it seems probable that the more direct tjos of viscosity determination are to be preferred. The electrical properties of colloids are very important. When an electric current is sent through a colloidal solution, the colloidal particles typically wander either to one electrode or the other. This is the well-known phenomenon of cataphoresis. Many investigators have studied the cataphoresis of living cells. This can very conveniently be followed, or rather measured, under the microscope. For the technique of such measurement, see Macaulay's 21. Special apparatus has also been devised by Northrop and Coates, see Northrop 22, KX-3, Nortimer and Coates 25. In this type of apparatus, cells can be observed to migrate in a closed chamber bounded by parallel walls. The cataphoretic velocity is measured at a point halfway between the bottom and top of the chamber, and also at a point 1/6 of the distance between bottom and top, where these values can be obtained from a curve in which the cataphoretic velocity is plotted against various arbitrary distances from top or bottom, C.F. Bjundlich and Beemanson, 27th. The true cataphoretic velocity can then be calculated from the formula of Vogt, in which W is the true cataphoretic velocity, Y the cataphoretic velocity in the center of the chamber, and M the catanhoretic velocity at a point 1/6 of the distance between bottom and top. In studying the electric properties of protoplasm, it is advantageous to have electrodes which can be introduced directly into the cell. The problem of constructing non-polarizable electrodes which could serve this purpose was not a simple one. It has, however, been successfully met by Edish and Pietpe, 25, and by various other workers, Taylor, 25, Jalea, 26, by Tendick and Werdeman, 27. The technical details of all these types of microelectrodes cannot be considered here. Their construction involves no new theoretical development, and they represent modifications of various standard electrodes in which a long and very fine capillary pipette is attached to the main body of the electrode. The pipette is of the type used in microdissection and generally has a diameter of only 1 to 2 microns. In the Etch-Peteret, Taylor, and Jalene instruments, it is filled with agar. Because of their small bore, the microelectrodes have a very great electrical resistance. They are attached to an ordinary microdissection apparatus and are moved about in the same way that microdissection needles are. Chapter 6. The absolute viscosity of protoplasm. Microscopical study has shown that protoplasm is typically a concentrated suspension of visible granules. Are these granules suspended in a relatively viscous medium or in a fluid medium? Some authors would have us believe that the intergranular material of protoplasm is normally a gel of high viscosity. Others hold that protoplasm is relatively fluid and that the intergranular material must necessarily be fluid. As a matter of fact, this difference in opinion is by no means a new development. For many years, biologists have argued as to whether the living substance is fluid or solid. The question is in part a question of definition. We can define a fluid as without form or amorphous, flowing readily, and so adapting itself to the shape of the vessel which contains it. If we have a gelatin jelly in a test tube and invert it, the gelatin does not flow out when the tube is inverted. It is a gel. But if we now subject the gelatin to heat, it flows out of the tube readily. It has become a fluid sol. For the colloid chemist, this is the usual conception of sol or gel. Actually, the distinction we have made is rather an artificial one. With a certain size of test tube and under the influence of a certain force, gravity, the gel does not flow. But if we apply a greater force, it does flow. Our distinction breaks down. Theoretically, it is better to follow Maxwell and to define the fluid state as a state in which flow occurs even when the force exerted of a substance is infinitely small. For a true fluid, the rate of flow is proportional to the shearing force. That is to say, the force tending to produce distortion or movement. When the rate of flow is not proportional to the shearing force, a substance is elastic and can no longer be considered as a true fluid. For some authors, it is an elastic fluid. Others prefer to call it a plastic solid. For the present, such absolute distinctions between solid and fluid states need not concern us. In the next chapter, we will consider the question of elasticity. The problem to be discussed at this point is whether or not protoplasm is highly viscous. Is it sol or gel in the ordinary sense in which these terms are used? Actually, the problem is a twofold one. Inasmuch as protoplasm is a suspension, we can consider either the viscosity of the suspension as a whole, or the viscosity of the dispersion medium, that is to say, the intergranular material. A watery suspension can be so concentrated that its viscosity may reach a value many times that of water. As the concentration is increased, the viscosity may eventually become infinite. It is possible to determine both the viscosity of the entire protoplasm and the viscosity of the intergranular material. The earliest measurements of absolute protoplasmic viscosity were made by the German botanist Heilbrunn in 1914. In the starch sheath cells of Yichia faba, a leguminous plant, the starch grains can be observed to fail under the influence of gravity. By measuring the speed with which the grains fall and comparing it with the speed of fall in water, Heilbrunn was able to determine the absolute viscosity of the protoplasm of these cells on the assumption that the speed of fall was inversely proportional to the viscosity in the two cases. This assumption is on the whole justifiable, but it is not strictly true. The law governing the speed of movement of a spherical particle through a fluid was first stated by Stokes in 1850. According to this law, in which v is the velocity, g the gravity constant, and the radius of the specific gravity of the particle, g that of the medium, and all the viscosity. From this formula, it is obvious that if g, a, q, and a remain constant, then the viscosity is inversely proportional to the velocity of the particle, or directly proportional to the time of fall. If the particle is moving under the influence of gravity, it can safely be assumed that when one measures the speed of drop of starch grains inside a cell and outside, that the diameter of the starch grain will not change appreciably. Its specific gravity will also presumably remain constant, and of course g will not change. But it must be remembered that the specific gravity of the protoplasm is different from that of water. It is somewhat greater. In a liquid of greater specific gravity, the gravity of the protoplasm of the Vita faba cells is not known. Other types of protoplasm have a specific gravity of about 1.05 to 1.1. If one estimates the absolute viscosity of protoplasm by comparing the speed of drop of starch grains in water and in the cell, one should perhaps make allowance for the difference in specific gravity of the two fluids. However, if one assumes that the specific gravity of protoplasm is no greater than that of water, the error is not large. Probably the error in Heilbrunn's measurements from this factor is roughly about 10%. That is to say, his values are 10% too high. Such an error could certainly be disregarded, except for the fact that there are other corrections which must also be made to Heilbrunn's values. In order to understand these corrections, we must discuss the applicability of Stokes' law. And in view of the fact that later in the chapter it will be necessary to refer to Stokes' law agam, the discussion will be made fairly complete. The theoretical derivation of Stokes' law involves a number of assumptions. Arcoldi lists 5 of them as follows. 1. That the discontinuities of the fluid are small compared with the size of the sphere. 2. That the fluid is infinite in extent. 3. That the sphere is smooth and rigid. 4. That there are no slip at the surface between sphere and fluid. 5. That the velocity of the sphere is small. To these might be added 6. That the particle is truly spherical. That the particle is a single particle and not one of many. 8. that electrical forces may be disregarded. The first assumption is chiefly important for very tiny spheres falling through gases. For liquids, it may generally be disregarded. The second assumption was investigated theoretically the absolute viscosity of protoplasm, cali and experimentally by Ladenbuisch, who introduced the following form of Stokes' law for the case of a sphere falling through a fluid contained in a cylinder. In this formula, I is the radius of the containing cylinder, L is its length, the other symbols are as before. If we are to apply Ladenburg's correction to the cells of Vita faba, we must know the diameter of the starch grains and the length and width of the starch sheet cells, or rather the length and width of the layer of protoplasm through which the starch grains fall. Certainly within a cell, the speed of fall of a starch grain would be decidedly less than outside of a cell. If Ladenburg's correction is neglected, the values obtained for the absolute viscosity are too high, and perhaps as much as 20 or 30% too high, or even more. The third and fourth assumptions are probably not very important in the application of Stokes' law to protoplasm. Most protoplasmic inclusions are smooth insofar as they can be seen by microscopic observation, and in general they appear to be rigid. We are also safe in assuming that the slip between a particle enclosed in protoplasm and the protoplasm itself is zero or approximately zero. Since the time of Poisson, it has generally been supposed that when a substance is wetted by water, its coefficient of slip in relation to water is zero. Evidence for this view is given by Wetham, who found that there was no measurable slip when water flowed over various metals or even over a film of oil. In the derivation of Stokes' law, it is assumed that the velocity of the moving sphere or particle is so small that higher powers of the velocity may be neglected. For small particles falling under the influence of gravity, this is certainly true. In the derivation of Stokes' law, it is assumed that the moving body is spherical. Most of the particles found in protoplasm are spherical. Starch grains are often not perfect spheres, but they drew with them 1890 brachioids. 48 P225 are nearly non-spherical, so that Stokes' law probably applies to them. Moreover, in a comparison of the Fa ray of the same particles in different fluids, any factor due to the non-spherical shape would probably cancel out. Stokes' law was derived for the movement of a single sphere through a fluid, and not for the movement of many. When many spheres move together, the fluid between them must of course move in the opposite direction. The presence of many spheres interferes with such a return flow. The nearer the spheres are to each other, the greater the resistance to this return flow, and the more slowly do the spheres move under the application of a given force. Thus, to return to the case under consideration in a liquid of given viscosity, many starch grains falling together would fall much less rapidly than a single starch grain falling alone. Sittenraj has attempted to calculate a correction for Stokes' law, which would apply to the case of many particles moving through a fluid. According to Cunningham, in this expression, A is again the radius of the particle, B is half the distance between the centers of two adjacent particles. But in making the computation, Cunningham prefers to substitute quantity B, which is equal to B of Cunningham, is admittedly on a rough approximation to the truth. One can be certain, however, that when many particles fall through a fluid, the speed of drop of any one particle is greatly retarded. Cunningham gives the following calculated values of Q for various values of B and B. Finally, it should be pointed out that Stokes' law does not consider a possible electric charge on the moving sphere. Practically all small Ki-A3 articles suspended in water do have an electric charge, and such a charge would tend to retard their movement. But the effect of an electric charge on the speed of movement only becomes important when the particles are of submicron or amicron size, that is to say, when they are so small as to be invisible with the highest powers of the microscope. Let us now return to the case of the Vita cells studied by Heilbrunn. It will be remembered that he determined the time it took starch grains within the cell to drop a certain distance, and then compared it with the time of drop of similar grains outside the cell. The ratio between the two times was taken as the viscosity of the protoplasm of the cell. From the discussion of Stokes' law, it is obvious that starch grains within a living cell would tend to drop more slowly, not only because of the greater viscosity of the protoplasm, but also because of three other factors. These three factors are, first, the fact that protoplasm has a greater specific gravity than water. Second, the presence of cell walls, which would tend to retard the fall of the cell are numerous and in fairly close proximity to each other. The first of these factors is relatively unimportant. The second might make a difference of 20 or 30% at most, whereas the third factor might produce a very great effect. All of them would tend to make the viscosity values of Heilbrunn for Vita cells too high. In addition, we should perhaps also consider the fact that Heilbrunn studied cells in thin sections. Under these conditions, there is an injury effect, which may produce viscosity increase. See chap. 8. What Heilbrunn actually found was that the time of fall of the starch grains within the cell was in some cases only 8 times as great as the time of fall outside the cell. In other cells, the ratio was somewhat greater, and he occasionally found cells in which the starch grains did not fall at all, and in which he assumed the viscosity to be infinity. These cells with infinite viscosity were apparently in a pathological state and were probably dead. The interesting thing about Heilbrunn's measurements is that he showed for the first time that protoplasm, in some cases at least, was not the extremely viscous material that many authors had supposed. Books on cytology often speak of protoplasm as a very viscous fluid, and the student of elementary biology is always impressed with the high viscosity of the living substance. The early writers on protoplasm often referred to it as a slime, that is to say, a fluid of very high viscosity. Of course, after all, to say that a fluid is very viscous means but little. It may be very viscous in comparison with ether, and at the same time very fluid in comparison with a heavy oil. For purposes of Orient compiled, for the most part, from the physical tables of Landolt-Bobensteev, viscosity of various fluids at 20 EC. In absolute units, Heilbrunn's measurements shelved that the viscosity of protoplasm might be very much less than the viscosity of glycerin or olive oil, that it might even be less than that of sulfuric acid. At the time of Heilbronn's experiments, this was rather an unusual idea, although Ewart III had concluded from observations on the effect of gravity on rate of streaming that the viscosity of the protoplasm must be low. Ewart believed that the viscosity of streaming protoplasm must lie between the limits 0.4 and 0.20. That is to say, he thought it could not be less than 4 more than 20 times as viscous as water. It is certainly less than 8 times that of water, and it is probably not more than 4 times that of water. It should be pointed out that Heilbronn's values were for the entire protoplasm rather than for the intergranular material. Heilbronn's first measurements of protoplasmic viscosity were made in 1914. In 1920, Seebeis published some estimates of the viscosity of various types of protoplasm based on his impressions with the microdissection needle. Seebeis's values are given in terms of the scale already mentioned in Chapter 4, CP 42. The obvious discrepancies of this scale have already been discussed, and no great amount of significance can be attached to these values. Seebeis believes the endosarc of a flowing amoeba to have a viscosity value of 4, that of a quiescent amoeba, a viscosity value of 5 to 6. He regards active myxomycete protoplasm as liquid, that is to say with a viscosity value of 3, whereas just before fruiting he believes it to be as viscous as Brito or Vaseline. This does not agree at all with the later measurements of Heilbrunn on various SJ-species of myxomycetes. For Heilbrunn found the protoplasm just before fruiting to have a viscosity only 9 to 18.5 times that of water. Depending on the species, see below. In estimating the viscosity of marine ova, Seebeis is impressed with an earlier observation of Chambeb 17, whom he quotes as follows. The internal cytoplasm of a marine egg is a viscous fluid. The viscosity is high enough to prevent any Brownian movement of the enclosed granules. Seebeis then cites an old observation of Exneb, made in 1867, to the effect that the Brownian movement of particles is stopped when the viscosity of the dispersion medium reaches a value as great as that of ordinary laboratory glycerin. Seebeis concludes that protoplasm in which no Brownian movement is present must possess a consistency of about this value, Accordingly, he ascribes to Echinoderm ova a viscosity value between 6 and 7. As a matter of fact, this train of argument is not very conclusive. Many authors have described Brownian movement in marmots. A very good description is given by Lilly 6. Chamebebs himself in later work, 24, mentions Brownian movement in the sea urchin egg. As a matter of fact, in the egg of Arbacia, the movement is not very vigorous, and for a very obvious reason. In a recent paper, Heilbrunn 26C, it was shown that the average distance between adjacent granules of the Arbacia egg was approximately equal to the radius of the granules, that is to say about 0.6. On the average, therefore, a granule in Brownian movement in this egg cannot travel farther than 0.16, and this distance is so small that it is almost at the limit of microscopic visibility. Under the usual conditions, therefore, Brownian movement in the Arbacia egg is not very easy to see. But one has only to centrifuge the eggs, and then the Brownian movement of the granules at the edge of the granule-free area is very easy to observe and quite rapid. Indeed, it has been possible to estimate the speed of this movement, as will be shown you later. In view of the fact that Brownian movement does occur in sea urchin eggs and in other marine eggs, there is no justification for Siefers's opinion that the viscosity of the protoplasm of these eggs must equal that of glycerin. Heilbrunn 22 used the magnetic method in order to measure are very interesting and will be considered in detail. The follow absolute viscosity of slime mold protoplasm, Badhamia utricularis, Plasmodia before fruiting 0.095 to 0.105 Physarenone scenarium. Plasmodia before fruiting 0.09 to 0.15 Traclicia fat. Plasmodia before fruiting 0.1 to 0.16. The above values were obtained with slime molds on glass, surrounded by a ring of moist cardboard. If the slime molds were placed on moist filter paper and were also surrounded by a ring of moist cardboard, the viscosity decreased until it was in McBenney viscosity tests. Heilbrunn determined the number of amperes which had to be passed through an electromagnet in order to twist a small iron rod through the protoplasm. The rod was 220 microns long and 50 microns diameter. The number of amperes necessary to twist the rod in protoplasm was divided by the number of amperes necessary to twist it in a drop of water, and this was taken as the absolute viscosity. Heilbrunn was very careful to test the accuracy of his method for measurements of absolute viscosity. He found that the viscosity in small drops was always greater than that in large drops. In Physaria and Badhamia, the amount of protoplasm is small, and for this reason the viscosity values obtained for these forms is relatively high. As Heilbrunn points out, the ease with which the iron rod is twisted is not only a function of the viscosity of the fluid which surrounds it, but also a function of the surface or adhesive forces which tend to keep the rod in contact with the glass or other substratum on which it rests. The higher the viscosity, the smaller the part played by these surface or adhesive forces. Hence, when Heilbrunn studied the twisting of iron rods in viscous liquids of known viscosity, the values obtained for these liquids by the magnetic method were found to be too low. On the other hand, for liquids less viscous than solution of glycerin, Heilbrunn obtained a value 25% too low, and for ether, a value 300% too high. From these considerations, in view of the fact that protoplasm is more viscous than water, we would tend to conclude that Heilbrunn's values for slime mold protoplasm were somewhat too low. But there is really another factor to be considered. An iron rod in protoplasm presumably rests on a differentiated film of protoplasm, which is between the main mass of the living substance and the glass of the slide. Protoplasm in general and slime molds in particular form films at their boundaries, see Chap 13. These films are very little to have more adhesive properties than glass. If this were the case, an iron rod resting on such a film would tend to have its movement seriously retarded, and too high values for viscosity would be obtained. Perhaps these two sources of error neutralize each other, but they may very possibly be of a different order of magnitude. With his electrical method, Heilbrunn found that the viscosity value obtained for a concentrated solution of glycerin was 25% too low. Perhaps in the case of protoplasm, there is a similar error of about 25%, although this seems more or less hypothetical. It seems quite possible that the presence of a protoplasmic film beneath the iron rod might cause an error of 100% or more. Obviously, we are more or less in the dark. Heilbrunn's values with the magnetic method are not very accurate. However, they represent results of a greater degree of precision or a lesser degree of uncertainty than any previously obtained. From Heilbrunn's observations, it seems safe to conclude that the viscosity of the protoplasm of slime molds before fruiting is not over 15 to 25 times that of water. And it may be and probably is much less than that. These values are only for the endoplasm. Toward the ectoplasm, Heilbruck found great variation. In some instances, the viscosity of the ectoplasm approached infinity. Such variable values are to be expected if the ectoplasm is a gel, for the viscosity of a gel varies with the amount of the shearing force exerted upon it. With relatively low shearing force, the viscosity of a gel is infinity. It has already been pointed out that cells are typically surrounded by membranes, and these membranes are certainly gels. Perhaps in slime molds, the ectoplasm corresponds to the membranes of ordinary cells. In Heilbronn's experiments with the magnetic method, he determined viscosity either by measuring the magnetic force necessary to turn an iron rod, or in some preliminary tests by measuring the magnetic force necessary to cause a barely perceptible movement of a small iron sphere. In 1924, Biz made 1 or 2 tests of absolute protoplasmic viscosity with the magnetic method. With the aid of a micropipette, he introduced nickel balls into the eggs of the sand dollar Echinirachnius, and then observed the speed with which they were drawn toward an electromagnet. Apparently, he made but few observations, and he states only that the speed of movement of a nickel ball in an Echinirachnius egg is of about the same order of magnitude as the speed of movement of the nickel ball in glycerin. Biz does not give a very complete description of his experiment, and it is hard to see how he could be certain that the nickel ball when in the glycerin was at exactly the same distance from the magnet as it was when in the egg. There are also a number of other objections that could be made to Biz's experiment, but it is hardly worthwhile to consider all of them, for there is one source of error which in itself would make it impossible to draw any conclusions from Spieris's observations. In recent years, it has become clearly recognized that the entrance of a needle or other object into a cell is frequently accompanied by injury. Thus, by 27 describes various injury phenomena which lollop the introduction of a fine needle into blood cells. Chambebs and Pollack 27 describe injuries and so-called cytolytic changes as the result of the presence of a micropet in starfish eggs. These workers use needles or pipettes only a micron or 2 in diameter. The nickel hull used by Cepheus measured 16 microns in diameter, and the pipette which introduced it into the egg must have been appreciably larger than this. Certainly, it is fair to assume that the introduction of such a large foreign object would cause marked injury in any cell, and such injury might alter the viscosity in any conceivable way. By coagulating the protoplasm, it might very well raise the viscosity to infinity. It would have been a simple matter for Cepheus to test the extent of the injury provoked by his micropet. It would only have been necessary for him to fertilize the eggs he had operated on, and to observe then whether or not they developed normally. There is no record of such tests in his paper. The centrifuge method has recently been used to determine description of this method has already been given, but so far little has been said regarding the physical principles on which it is based. We have seen that the movement of a granule through a fluid obeys Stokes' law. When the force acting on the granule is centrifugal force rather than gravity, the law takes the following, in which, as before, F is the speed of movement of the granule, G the gravity constant, the specific gravity of the granule, Q the specific gravity of the fluid through which the granule travels, the radius of the granule, and I the viscosity. In addition, C is the centrifugal force in terms of gravity. In the above formula, G is of course known, A, the radius of the granule, can be determined by direct measurement, the centrifugal force, C, can be calculated, if the number of turns per second and the radius of turn of the centrifuge are remote. As shown in the last chapter, V can be determined from a series of centrifuge tests. There remain then only A and Q. If it were possible to know A, the specific gravity of the granule, and Q, the specific gravity of the intergranular fluid, we would be able to use Stokes' law for the determination of the absolute viscosity of protoplasm by the centrifuge method. As a matter of fact, it is possible to determine the specific gravity of the granules of cells, and from this value it is then possible to calculate protoplasma monogramming. Heilbrunn 5, the specific gravity of the intergranular fluid. We are thus in a position to use the centrifuge method for the measurement of absolute viscosity. It must be remembered that the derivation of Stokes' law involves a number of assumptions. 8 of these assumptions have already been listed and more or less adequately discussed in relation to Heilbrunn's viscosity determinations on the protoplasm of starch sheath. I see pages. In general, the principles involved in the centrifuge method are much the same as those involved in the gravity method. The previous discussion of the applicability of Stokes' law is therefore in the main sufficient, and only a few additional remarks are necessary at this point. Assumption 2, that the fluid through which the granule travels is infinite in extent, has more importance for starch sheath cells in which the starch grains move than for cells in which one follows the movement of granules. In sea urchin eggs, Arbacea, the diameter of the smaller granules is only a 200th of the diameter of the cell. Ladenburg's correction may therefore be neglected in this case, for it only becomes appreciable when the moving particle is much larger in comparison with the size of the containing vessel. The fifth assumption, that the velocity of the moving sphere is small, might be thought not to hold for centrifuge experiments. However, the highest speed that has ever been noted for the movement of granules through a cell under the influence of centrifugal force is less than 2 × 10 centimeters per second. Such a speed is certainly small enough so that its higher powers may be neglected, and this is the condition required for the validity of Stokes' law. Moreover, in the cell in question, the Gumingia egg, centrifuge tests with a relatively small amount of centrifugal force give the same viscosity values as when the granules move at top speed with higher centrifugal force. Finally, attention should be directed again to assumption 7, that there is only one sphere or granule moving through the fluid. In living cells, there are vast numbers of granules, and all of them move when the cells are centrifuged. Fortunately, we can apply the correction factor of Cunningham. In order to use this correction, we need merely know the radius of the granules and the distance between them. However, Cunningham's factor considers only the case in W each granules of the same size are moving in the same direction. Many cells contain granules of differing size, or they contain granules which move in opposite directions on the application of centrifugal force. Granules of different sizes would move at different rates of speed. They would thus tend to collide. Collisions would also occur between granules moving in opposite directions. The effect of all these collisions would be to decrease the speed of the moving granules, and this would make the values for the viscosity obtained by the centrifuge method higher than they really should be. Because of the varying conditions found in different cells, it would probably be best to consider the effect of collisions for each type of protoplasm separately. In those cases in which practically all of the granules are of one size and move in the same direction, the collision factor may be neglected. We are now in a position to consider the actual measurements. In the Aracia egg, the viscosity was determined both from the speed of the small heavy granules and from the speed of the larger pigment granules, which are also heavier than the rest of the protoplasm. Let us consider first the case of the smaller granules, inasmuch as these granules are very much more abundant than the other granules in the egg. We may neglect the influence of collisions, especially in view of the fact that collisions with the larger granules would tend to increase the speed of movement, whereas collisions with the fat or lipoid granules would tend to decrease it. In order to apply Stokes' law, we must find values for F, A, Q, and Q. The determination of the velocity, F, is a relatively simple matter. In a series of tests, one finds the number of seconds just sufficient to move the small heavy granules into one half of the egg. When nearly all of the granules are in one half of the egg, we can assume that the granules originally at the pole farthest distant from this half are now at the equator. The distance traveled is thus half the diameter of the egg, and when this is divided by the time in seconds required for the movement, we obtain F. As a matter of fact, the distance traveled is slightly greater than the radius of the egg, for during centrifugal treatment, the egg becomes somewhat elongated. If this elongation is taken into account, the speed of the granules is found to be 0.00011 centimeters per second when the centrifugal force is 4,968 times gravity. The radius of the granules, AA, is obtained by direct measurement with a movable scale ocular. For very small granules, such measurement is certainly inaccurate, and the inaccuracy is especially important in view of the fact that the value for A is squared in the formula. The radius of the small heavy granules in the Heilbrunn egg was found to be 0.160 LC, or 0.0000. In order to determine the specific gravity of the granules, the eggs were broken up by shocking them with splintered glass, and the resultant mass was then centrifuged in various concentrations of sugar solution. In concentrated solutions of sugar, the granules move to the inner part of the centrifuge tube. In dilute solutions, they go to the bottom, that is to say, to the outer part of the tube. Those solutions of sugar, in which the granules move neither the one way or the other, have the same specific gravity as the granules. Inasmuch as the specific gravity of various concentrations of sugar solution is known, it is possible to determine the specific gravity of the granules. But this determination rests on the assumption that the granules do not change their specific gravity after emerging from the egg. In all the end determinations, the specific gravity of the granules was tested as rapidly as possible after their removal from the egg. However, if the granules were allowed to remain out of the egg for a time, the exospecific gravity did not change from the value obtained immediately after removal. This is evidence that the specific gravity of the granules outside of the egg is not markedly different from their specific gravity within the egg. But it is far from conclusive evidence, for it is quite possible that as the granules emerge, there is a very rapid change in their specific gravity. Here then is a source of error, and apparently we have no means of knowing how great an error may be involved. But fortunately, there is a method of calculating the maximum value which the specific gravity of the granules may have, so that we can at least limit the extent of the possible error in one direction. As we shall see later, there is no need of limiting it in the other direction. For the present, we will be content with the value obtained in the Mamer acuity outlined. It is possible to determine the specific gravity of entire eggs in the same way that the specific gravity of the granules was determined. That is to say, by centrifuging them in various strengths of sugar. Knowing the specific gravity, both of the eggs and of the granules, it is possible to calculate, the specific gravity of the intergranular fluid, provided that one knows the relative volume of the granules as compared to the volume of the entire egg. The relative volume of the granules can be computed from the fact that after the most vigorous centrifugal treatment, the granules occupy a spherical segment, the altitude of which is about a third of the diameter of the egg. From simple geometrical considerations, it can then be shown that the granules occupy of the total egg volume. Now, in this fraction, it is easy to derive a simple equation for Q in terms of on, the specific gravity of the granules, and S, the specific gravity of the entire eggs. By calculating Q, it is possible to arrive at a value for CR for further details. See Heilbrunn 26c. It only remains to find Q, the Cunningham correction for Stokes' law. This correction, as shown previously, involves both the radius of the granules and the distance between the centers of adjacent granules. The radius is known. The distance between the centers of adjacent granules can either be measured, or it can be computed from the proportion of the total volume of the egg that the granules occupy. Such computation is certainly more accurate than DC-Kett observation could be. The distance between adjacent granules is calculated in the following fashion. We assume that the granules are surrounded by spheres so that the entire volume of the egg is filled. Then, if the radius of the granules is and the radius of the spheres is 6, and the total number of granules or spheres, the volume of the egg, is netty shown. That's because we have already shown that the volume of the granules is of that of the entire egg. The volume of the egg is also equal to nx. The volume of the spheres must be multiplied by, since spheres when closely packed occupy only 74.05% of a given volume. Equating the Uyo equations for the volume of the egg and solving for B, we equation given on P58. We now have O, the terms necessary. Substituting them into the modified form of Stokes' law, the viscosity is found to be 0.0183. This is 1.8 times the viscosity of water. The viscosity of the Arachia egg protoplasm may also be calculated from the speed of movement of the large pigment granules. These granules have a radius of 0.46 microns, or 0.00046 cms. With a centrifugal force 468 times gravity, their speed is about 0.00 CO24 centimeters per second. Dit. Ordinarily, the pigment granules break down when they emerge from the egg, and the measurement of their specific gravity would involve difficulties. The pigment granules do not break down in the absence of calcium, see chap 13, and perhaps by smashing the eggs in calcium-free seawater, a value for the specific gravity of the pigment granules might be obtained. This was not done, and in the absence of a measured value, we may assume that the pigment granules have the same specific gravity as the colorless granules. Q and Q are thus the same for the pigment granules as for the colorless granules. Substituting into the modified form of Stokes' law, we now obtain a value of 0.069 for the viscosity. This value is too high because the effect of collisions has been neglected. The large pigment granules, as they move through the egg, are continually colliding with the smaller granules, which move at a much slower rate. The retarding effect of these collisions can be calculated. For details, see Heilbrunn 26c. When correction is made for the effect of collisions, the value for the viscosity, as determined from the speed of the pigment granules, is 0.25 or 2.5 times that of water. This value agrees fairly well with the one previously found from the speed of movement of the small granules. We therefore conclude that the viscosity of the intergranular material of the Arbacia egg is approximately 2 times that of water. There are a number of possible errors involved in the measurement of the viscosity of Arbacia protoplasm. The most important one: the error due to the inexact determination of the size of the small granules. 2. The possible error involved in a change of specific gravity of the granules which may occur as they emerge from the egg. 3. The error involved in the calculation of Q4. The possible error due to an influence of the moving granules on the viscosity. The last of these sources of error really does not exist. It is true that in many gels, pressure or mechanical force can produce a lowering of the viscosity. But in the Arbacia egg, no such effect is present, for when measurements of viscosity are made with widely different amounts of centrifugal force, the same values are obtained. If the movement of the granules through the protoplasm produced a lowering of the viscosity, then the greater the centrifugal force and the greater the speed of the granules, the lower the viscosity would be. The first source of error, that due to the inexactitude with which the size of the granules is measured, may be considerable. The pigment granules of the Arachia egg are large enough so that their diameter may be determined with some degree of accuracy, but the colorless granules are too small for accurate measurement. One way of estimating the size of the colorless granules is to compare them with the pigment granules. Their diameter appears to be about 1/3 as great, and this estimate of their size agrees very well with the value obtained by direct measurement with a movable scale ocular. If instead of being 1/3, the diameter of the small granules were as much as 1/2 of the diameter of the pigment granules, then the viscosity value given above would be only 1/2 as Genji as it should be. However, it does not seem possible that the diameter of the small granules could possibly be as great as 1/2 that of the pigment granules, so that this is the maximum error possible. The second possible error appears in the determination of the specific gravity of the granules. Perhaps they undergo a change in specific gravity as they emerge from the egg. If the specific gravity of the granules decreased on emergence, then the value obtained for the viscosity would be too low. It is hardly necessary to consider the opposite possibility, that the granules undergo an increase in specific gravity, for no one could assume that the value obtained for the viscosity is much too high. It is too close to the viscosity of water. In computing the viscosity, the value taken for, or the difference in specific gravity between the granule and the intergranular fluid, was taken as 0.76. Now, it can be shown by a simple calculation that this cannot be far from the true value. We know the specific gravity of the entire egg. It is approximately 1.05 times. We know further that the colorless heavy granules occupy about a total volume of the egg. If then we knew the specific gravity of the intergranular fluid, we could calculate the specific gravity of the colorless heavy granules. The specific gravity of the intergranular fluid cannot vary within very wide limits. It must be greater than the specific gravity of seawater, for it contains at least as much salt as seawater and probably more, compare P. 25. And in addition, it contains dissolved proteins. Now, if we assume that the specific gravity of the intergranular fluid is equal to that of seawater, that is to say, 1.03, then the specific gravity of the granules is found by simple algebra to be 1.14. And a Q is LL. This is certainly a maximum value. And yet, it is not very much greater than the value 0.76 used in computing the viscosity. If we were to assume that the specific gravity of the intergranular fluid were only slightly greater than that of seawater, then decidedly lower values of OQ would be found by computation. Thus, if the specific gravity of the intergranular fluid were taken as 1.04, then OO would be only 0.5. In these calculations, we have neglected the pigment granules and the fatty granules. This involves no serious error, for their total volume is very small compared to the volume of the colorless heavy granules. Our calculations show beyond any question that the value used for a BC is not far from the correct one. The second of the possible errors is therefore not great. Finally, there is the error involved in the calculation of Q. Cunningham's mathematical treatment involves a number of assumptions, and his formula for Q is not exact. How great an error may be involved, it is at present impossible to say. The question of the possible errors involved in the determination of the absolute viscosity of the protoplasm of the Sorichin egg has been taken up in some detail. This has been done in part because it has seemed impossible to some commentators that any protoplasm could be so fluid. One reviewer has indeed pointed out the contradiction between the value obtained by the centrifuge and that arrived at by C-meat. It will be remembered that C-Phys was of the opinion that the viscosity of the Sorichin egg protoplasm was like that of glycerin, and he thinks of it as being about 800 times that of water. The value obtained by the centrifuge method is of a totally different order of magnitude, even if we allow for every possible source of error and go so far as to neglect the CT-Ch-NX-CAM correction completely. Because of the sharp contrast between the values of Heilbrunn and Sears, it is of interest to consider the determination of the viscosity of Arbaceae protoplasm by another method. When one centrifuges Arbaceae eggs, the heavy granules pass into one hemisphere of the egg. They do not remain in this position indefinitely, and after a time they again become scattered to the entire egg. The return of the granules is due to Brownian movement. According to E. Stein, the distance traveled by a granule in Brownian movement in the time t is given by the following formula. In this formula, dx is the distance traveled by the granule in any one plane, t is the absolute temperature of the radius of the granule, and rj the viscosity. In the case we are considering, we already know A and T, may be taken as the radius of the egg, that is to say approximately 38 microns, if now we can determine T. The time required for a granule to travel across half of the egg, we could calculate the viscosity. No very exact determination of T is possible, but it is a simple matter to find the general order of magnitude of T, and from this the general been calculated for various assumed values of Jj. For the sake of simplicity, we have used the viscosity in terms of water plasm where 8 or 900 times water, as Sayat Biz believes it to be. It would require 150 hours for the granules to return through the egg after being centrifuged into one hemisphere. Ordinarily, the Arbacia egg only remains alive for 24 hours. And surely, within 2 hours after centrifugal treatment, the granules have become completely scattered throughout the egg. If one tries to estimate the time required for the first granules to reach the pole of the egg farthest from the hemisphere to which they have been centrifuged, one finds that no exact measurement is possible. The time seems to be more than half an hour and less than an hour. This would indicate a viscosity between 2.87 and 5.73 times that of water, or roughly about 4 times that of water. It must be remembered that the Einstein formula is not exact, and that it would probably give too high values for the viscosity when the granules are as numerous as they are in sea urchin eggs. With a high concentration of granules, there is surely a retardation of speed of Brownian movement, although this might not be noticeable if the granules are moving toward a granule-free area. The study of Brownian movement in Arbacia eggs has shown that Seabiz's value for the viscosity of the protoplasm is certainly wrong. This was to be expected, for Seabiz's opinion was primarily based on the erroneous observation of Chambiz, to the effect that there is no Brownian movement in marine eggs, see previous discussion. The determination of the protoplasmic viscosity of the Arbacia egg, both by the centrifuge method and by the study of Brownian movement, depends on the measurement of the small granules. These cannot be very accurately measured, as has already been pointed out. Fortunately, any error in the measurement of the granules would have opposite effects in the two determinations. Thus, if the value that we have used for the radius of the granules is too low, this would result in too low values for the viscosity as determined by the centrifuge method and in too high values for the viscosity as determined by the Brownian movement method. In view of this fact, it seems wise to average the results obtained by the two methods. In this way, we find the most probable value for the absolute viscosity of the Arbacia protoplasm to be 0.03, or 3 times the viscosity of water. It seems certain that the viscosity of the Arbacia protoplasm, or rather of the non-granular part of the protoplasm, is only several times as great as that of water. To biologists used to thinking of protoplasm as a very viscous fluid, this may seem surprising, but from a physical standpoint, it is readily understandable. The non-granular part of protoplasm is essentially a protein solution. At any rate, the constituents which would have the greatest effect in increasing viscosity are protein. Now, most protein solutions either have a viscosity 2 or 3 times that of water, or they are gels with a viscosity approaching infinity. The viscosity measurements indicate certainly that the protoplasm of the Arbacia egg is not a gel, and in the next chapter, additional evidence will be presented which will bear out this conclusion. It is therefore not at all surprising that the non-granular protoplasm of the Arbacia egg should have a viscosity of the same order of magnitude as a protein cell. The determination of the absolute viscosity of Arbacia egg protoplasm by the centrifuge method was to some extent uncertain, because of the possible error involved in Q, the correction factor of Kaneko in the egg of the clam Gomingia. This factor is of less importance. This is due to the fact that in the Gomingia egg there are almost as many light granules as heavy granules. See figure 1p45? Hence, when the egg is centrifuged and the heavy granules move in one direction, it is comparatively easy for a return flow to accompany the lighter granules in the opposite direction. In the determination of the protoplasmic viscosity of Gomingia egg protoplasm, the Cottingham factor was neglected. The results obtained were thus certainly too high. They are also too high for another reason. The Gomingia egg contains 2 sets of granules which move in opposite directions when the egg is centrifuged. These granules would certainly tend to collide, and the collisions when they occurred would slow the speed of the granules and thus tend to increase the viscosity values. The effect of such collisions was also neglected. The specific gravity of the heavy granules of the Gomingia egg was determined by the same method employed for the similar granules of the Arbacia egg. The value obtained was 1.18677. The calculation of the specific gravity of the intergranular fluid is relatively simple. The Gomingia egg contains approximately equal numbers of light and heavy granules, and these are not only of the same approximate size, but they differ in specific gravity from the intergranular fluid by approximately the same amount. For when the egg is centrifuged, the light granules travel about as fast in one direction as the heavy granules travel in the other. From these considerations, it is apparent that the specific gravity of the entire egg must be approximately equal to that of the intergranular fluid. The specific gravity of the entire egg was found to be 1.04853. P is therefore 1.18677.1.04853, or 0.13824. Here again, there is a possible error. As in the ease of the Aracia egg, for the granules may change their specific gravity upon emergence from the egg. Fortunately, for the Gomingia egg, it is possible to show that errors in the determination of the SX3 specific gravity of granules cannot be of such a nature as to make the values found for the viscosity much lower than they should be. It is certain that the specific gravity of the intergranular fluid is 1.04853. Now, the light granules cannot have a specific C gravity much lower than 0.9. For there are no substances which could possibly be present as fluid or solid material in protoplasm which could have a specific gravity more than a few hundredths lower than this. If we assume the specific gravity of the light granules to be 0.9, then a Q for the light granules is 1.04853 to 0.9, or 0.14853. This value is only slightly greater than that already found for the heavy granules. As stated previously, Q must be the same for light granules and heavy granules. Hence, the value found for the heavy granules cannot possibly be more than a few percent too low. The granules of the Gomingia egg move very rapidly G under the influence of centrifugal force. For details, see Heilbrunn, 26C. From the speed of this movement, one can calculate the viscosity of the protoplasm. It is found to be 0.043 or 4.3 times that of dead water. This is really maximum value. As stated previously, no allowance was made for Cunningham's correction or for the effect of collisions between granules. Both of these factors would tend to slow the speed of the granules, and both would make the values determined for the viscosity higher than they should be. All that WA can say with certainty is that the viscosity of the intergranular Gomingia protoplasm lies between 1 and 4.3 times that of water. The determination of the viscosity of the Gomingia protoplasm is especially important in that it seems that all possible errors which would tend to markedly increase the value have been excluded. Colloid's factor has been completely neglected. The specific gravity of the granules is certainly not appreciably greater than we have found it to be, and even if the size of the granules is somewhat larger than our measurements indicate, this cannot be a very important source of error. Apparently beyond any question, the viscosity of the protoplasm of the Gomingia egg is of the same order of magnitude as that of a protein sol, both in the case of the Gomingia egg and the Aracia egg. Our determinations of protoplasmic viscosity concern only the material between the granules, that is to say, the dispersion medium of the protoplasmic suspension. It is of high interest now to consider the viscosity of the entire suspension, granules plus dispersion medium. The viscosity of a suspension may be calculated according to the formula derived by Ebstein and later by Hatchek. This formula may be written as follows, in which 9EnC is the viscosity of the suspension, Regio, the viscosity of the dispersion medium, the ratio of the volume of the suspended particles to the total volume of the suspension, and Intersterning gives the value of H as 2.5. Hatchek considers it to be 4.5 in the protoplasm both of the Aracia and of the Gomingia eggs. It is about the formula therefore shows that the viscosity of the entire protoplasmic suspension is 1 1/2 to 2 times the viscosity of the intergranular fluid, depending on whether one uses Einstein's value for Hatchek's. It is now generally recognized that the formula of Einstein and Hatchek does not give very accurate results, especially for more concentrated suspensions. Various other formulae have been suggested. Thus, for example, Bingham has proposed the following empirical formula. 7C and Yo are as before is the volume concentration of the solid, and D is the particular value at which the concentration of the granules is so high that the viscosity becomes infinite. Bingham, Fluidity and Plasticity, New York 1922. Perhaps Bingham's formula is better than that of Einstein and Hatchett. As the concentration of suspended matter becomes higher and higher, the viscosity of the suspension certainly must approach infinity. And this could not be predicted from the Einstein-Hatchett formula. But in order to use Bingham's formula, one must know 7, the concentration of suspended material, at which the viscosity becomes infinite. Or inanimate suspensions, CLL can readily determined by varying the concentration of suspended matter until there is no longer any flow possible. At first sight, it would not seem feasible to apply the Bingham formula to protoplasm, for one cannot add or take away granules from the cell. And yet it is really a very simple matter to change the granular concentration of the protoplasmic suspension. Osmotically, one can extract water from the cell, and of course, this has the effect of increasing the concentration of granules. When Arbacia eggs are placed in a hypertonic solution, which reduces the volume to 3/4 of what it is normally, the viscosity of the protoplasm is increased about 6-fold. When the volume is decreased to 2/3 of the normal, the viscosity is greatly increased and approaches infuity. Similar results were also obtained for Gumingia eggs. For both types of material, therefore, the value for JJD in Bingham's formula is approximately GG plasmic suspension in both Arbacia and Gumingia eggs as 3 times the viscosity in a granular fluid. The application of Bingham's formula is not without error. The increase in protoplasmic viscosity in hypertonic solutions is apparently not due entirely to the greater proximity of the granules. For when the eggs are returned to seawater, the viscosity does not completely return to its original low value. There is thus an error due to the specific action of hypertonic solutions in increasing the viscosity. The effect of this error would be to make the viscosity as calculated from Bingham's formula somewhat too high. If we average the results from the Einstein-Hatchett formula and from the Bingham formula, we can conclude that the viscosity of the protoplasmic suspensions in Arbacia and Gumingia eggs is 2% times that of the intergranular fluid. In the Arbacia egg, there is also a direct method of estimating the viscosity of the entire protoplasm. In this egg, it will be remembered that the heavy granules are of 2 sorts, small colorless ones, and pigment granules with a diameter approximately 3 times as great. The pigment granules are very few in number as compared with the colorless granules. When the eggs are centrifuged, the pigment granules, because of their larger size, tend to move much more rapidly than the colorless granules. But the more rapid movement of the pigment granules is continually retarded by collisions with the colorless granules in their path. We have already considered the effect of these collisions in calculating the viscosity of the intergranular fluid from the speed of the pigment granules. Viewed from another angle, the speed of movement of the pigment granules through the rest, the protoplasm is a measure of the viscosity of the rest of the protoplasm, and inasmuch as the pigment granules are few, the viscosity of the rest of the protoplasm cannot differ very appreciably from the viscosity of the entire protoplasm. From this point of view, if we neglect the effect of collisions, the viscosity, as calculated from the speed of movement of the pigment granules, should give a rough measure of the viscosity of the entire protoplasm. It has already been pointed out, CP 79, that the viscosity calculated in this manner is 6.9 times that of water. The viscosity of the intergranular protoplasm of the Arbacia egg was found to be 3 times that of water. From the speed of the pigment granules, one would conclude that the viscosity of the entire protoplasm was 2.5 times that of the intergranular material. The value obtained by direct determination thus agrees perfectly with the value obtained by calculation from the Einstein-Hatchett and Bingham formulae. The entire protoplasm of the Arbacia egg is thus about 7 times as viscous as water, and the entire protoplasm of the Gumingia egg is something less than 11 times as viscous as of these marine eggs is of the same order of magnitude as that of the plant cell, studied by Heilbrunn. The viscosity of the protoplasm of these cells is less than that of an oil, less than that of sulfuric acid. Ordinarily, as the biologist examines protoplasm under the microscope, he gets the impression that it is very viscous. TT is probable that such an impression, doubtless a universal one, is due in part to an illusion. When we examine protoplasm under the microscope, we see granules much larger than they really are. They may be magnified 100, 500, 1,000 times. Often as the cell is subjected to outside forces, we follow the movement of these granules from one part of the cell to another. Now the movement of a particle through a fluid under a given force is faster the larger the particle. Toward small particles, the speed varies as the square of the diameter. We see that the granule we are observing appears to move with some difficulty. We think of it as a large granule, failing to realize that it is much magnified, and subconsciously we associate the difficulty of motion with a high viscosity. We do not take account of the fact that the granule is really very tiny. Of course, it is true that the speed of the granule is magnified as well as its size. But it must be remembered that the ease with which a granule moves varies as the square of the diameter. A granule, which is only 1/100th of its apposite size, would move at a speed 1/10,000th of what we might expect if it were as large as it looked, or 1/100th of what we might expect if not only the granule size, but the speed as well were magnified by the microscope. In our minds, we naturally correlate the slow movement of the seemingly large granules with a viscosity much greater than it really is. It'll be 126C again when we see protoplasm flow through narrow tubes. Both the speed of flow and the diameter of the tube are magnified equally, but by Poiseuille's law, the speed of flow increases with the fourth power of the diameter of the tube. If we saw the tube as small as it really is, instead of magnified hundreds of times, we should doubtless subconsciously associate any flow through such a narrow tube with a very low viscosity. Actually, we are concerned not so much with the explanation of impressions or illusions as with actual facts. And at the present time, the best measurements available indicate that the protoplasm of starch sheath cells, of slime molds, and of marine eggs is only a few times greater than that of water. It must not be supposed that all living cells have such a low protoplasmic viscosity. Certainly, this is not true when the entire protoplasm is considered. Some muscle cells are composed for the most part of fibrils, definite formed elements which iractically thew up the cell. The presence of these fibrils must result in a certain degree of rigidity. The contents of the muscle cell, considered as a whole, must have a very high viscosity, although the fluid between the fibrils may or may not be very viscous. Probably many types of specialized cells contain fibrillar strands of one sort or another. Thus, for example, in the common protozoan paramecium, there is a neuromuscular apparatus composed of strands, which extend through the ectoplasm and also ramify into the endoplasm. Also, in this protozoan, there are strands in the endoplasm along which the food vacuole has traveled. The presence of these strands would have an effect on the viscosity of the protoplasm as a whole, and they might offer some resistance to the passage of granules or food vacuoles through the cell. It is not surprising, therefore, that Petit B25 found high viscosity values for the protoplasm of paramecium. In her experiments, she fed the paramecia powdered iron and then centrifuged them. On the basis of the speed of the food vacuoles filled with the powdered iron, she calculated the viscosity of the protoplasm of the paramecium. In making this calculation, she determined the specific gravity of moist iron particles, and she assumed that the specific gravity of the fluid through which the iron vacuoles passed was approximately equal to the specific gravity of the entire paramecium. As a result of the calculation, it was found that the viscosity of the paramecium protoplasm, considered as a whole, was 87.26 or 8,026 times that of water. In another experiment, Fetter fed the paramecia starch and then determined the speed of movement of the food vacuoles filled with starch grains. In this test, the viscosity was found to be 8,027 times that of water. It is very probable that the viscosity of the paramecium protoplasm varies with different rates of shear. It would be interesting to make determinations of absolute viscosity at different centrifugal speeds. It has been intimated that the presence of strands in the paramecium cell would make the viscosity of the protoplasm as a whole high. This is a logical explanation of the high viscosity values obtained by Fetter, but it is of course not the only explanation, and as a matter of fact, it does not seem to be the most probable one. If a paramecium cell is centrifuged even for long times and at high speeds, the small granules which are scattered throughout the protoplasm do not move at all. This may be protoplasma moiographian or E6 due to Dish-Gillish, one of three causes. Perhaps the intergranular medium is too viscous to permit of a movement of the granules, or perhaps the granules do not differ very markedly in specific gravity from this medium. Or perhaps finally the cell is so full of granules that there is no possibility of their moving into a limited portion of the cell. This last explanation seems the most probable one. Apparently, in many cells, the granules are so numerous that they fill up almost the entire volume. This is apparently true in the case of the egg of the starfish Asterias. After long centrifugal treatment, only a very small region of the egg becomes free of granules, and longer centrifugal treatment does not seem to increase the extent of this granule-free region. This was probably first noted by Lyonset. Similarly, in the case of the egg of the sea urchin Strongylia centritus, it was found impossible to centrifuge the granules into a small fraction of the egg volume. It is not surprising, therefore, that GXEXDWH5 found these eggs very resistant to centrifugal force. After long centrifuging, there is indeed a small region which becomes free of granules, but the volume of this region is very small. It may sometimes be seen as a little hyaline projection at one end of the egg. In cells in which the granules are so numerous that they occupy practically all of this space, one must conclude that the viscosity of such a concentrated protoplasmic suspension is high. It would certainly be very surprising if it were not. As to the viscosity of the intergranular medium of such cells, we have no precise information. But the fact that the granules are so closely crowded and yet in living cells must retain the power of moving over each other is a strong indication that the viscosity of the intergranular medium must be low. In this charter, it has been shown that in certain cells, at least, the viscosity of the granule-free protoplasm is only several times as great as that of water. Even when the granules are considered along with the rest of the protoplasm, the viscosity of the entire suspension is often not more than 8 or 10 times that of water. These values are much lower than those usually assumed. Thus, in a recent book, May 21st, the author assumes, as a matter of course, that the viscosity of the streaming protoplasm of plant cells is well over that of castor oil, that is to say, over 1,000 times that of water. Other authors assume that protoplasm is typically a gel with high viscosity. It is hoped that, in the future, the viscosity of the protoplasm of man, other types of cells, will be measured. In many instances, the determination of the absolute viscosity of the protoplasm is not a very difficult matter. Often, the specific gravity of the granules of a cell can be calculated in the manner outlined for the Arbacia egg. The difference between the specific gravity of the granules and that of the rest of the protoplasm can then be set within fairly narrow limits. And when this is done, merely a few centrifuge tests together with a few simple size measurements are necessary to complete the tests. The discussion so far has been concerned only with the viscosity of the cytoplasm. In view of the fact that the nucleus is such an essential part of the cell, it is very important to know what the physical properties of its interior are. Unfortunately, there is very little definite information. In the literature of the present time, one finds many opinions as to whether the interior of the nucleus is a sol or gel, but there is not much evidence one way or the other. LBPBSLKIC24 believes that the interior of the nucleus is a sol, so does Shade B26, although he had previously held, 25, that it might sometimes be a gel. Kite 13 studied the nuclei of a number of types of cells with the microdissection method. He found the nuclei tough, with the exception of the nucleus of the immature starfish egg. Chambers 24 believes that Kite's results are due to coagulation or gelatinization following the entrance of the needle, and he cites his previous observations and the unpublished observations of Chambers and Schmidt. To show that the nucleus has a fluid interior, According to Chamber, that has definitely been shown, however, that the interkinetic nucleus in every living metazoan cell so far studied is fluid and possesses no visible structure except for one or several nucleoli and a delicate investing membrane. Taylor 20 found by microdissectionary that the macronucleus and micronucleus of the protozoan JIC uploads were both gels. He states that the micronucleus could be pulled into threads. Similar observations were made by Berthold 86, CP 48 for the nuclei of the pollen mother cells of various monocotyledonous plants. These also could be pulled into long threads. In view of the many objections to the microdissection method, CP 40, it is doubtful if the results obtained by microdissection are of any value. They have unfortunately drawn attention away from definite exact observations on uninjured cells, which are of course far more valuable. These observations are of two sorts: those on Brownian movement of particles within the nucleus, and those on the movement of the nucleolus through the nucleus under the influence of gravity or of centrifugal force. Before discussing the observations on Brownian movement, it should be clearly pointed out that Chidieb's statement that there are no visible structures in the interior of the living nucleus other than the nucleoli is certainly not true generally, for it is contradicted by the observations of dozens, probably hundreds of observers. The first books on the cell, for example, those of Schwaken 39, Shelby-Doe 49, and Homesty 67, contain descriptions of granules visible within living nuclei, and they have been described by numerous observers since then. The granules within living nuclei may show Brownian movement. Busso 10 barely mentions movements of granules within nuclei. The most complete study of the Brownian movement of granules within living nuclei has been made by Giossa 16. He observed chiefly four types of cells. The nuclei of the cells of the salivary gland of the snail limna stagnalis have many granules of each are in very active Brownian movement. As is typical for Brownian movement, the amplitude of the displacements decreases as the granular diameter increases. For granules with a diameter over 40, Brownian movement is no longer visible. Granules 1I in diameter suffer displacements of Giossa states definitely that the displacement may occur in any plane in space, an important point. Giossa also describes Brownian movement of visible granules in the nuclei of clam eggs and in the nuclei of the epithelial cells of salamander larvae. Giossa describes threads or filaments in the nuclei of the Malpian tubes of Corethra larvae. He does not mention any movement of these filaments, and they are probably motionless. However, Chihuago has described modal filaments in the resting nucleus of frog leukocytes. Leposokin 24b describes Brownian movement in the nuclei of various Jalant cells. From Giossa's observations, it is possible to get a general idea as to the viscosity of the intergranular fluid of the nucleus. For the nuclei of the salivary gland cells of the snail, Giossa states that granules 1 micron in diameter suffered displacements of 1I. Unfortunately, Giossa does not state how great a time was required for such a displacement. If he had recorded this time, it would have been possible the use of the formula given on page 73 to calculate the viscosity. Thus, if the time were 1 second, the viscosity would be 0.07. If the time were 10 seconds, the viscosity would be 0.7. It is very probable that Giossa made Ill's observations quickly, and thus it seems certain that the viscosity of the nuclear fluid of snail cells is not much greater than the viscosity of water. Various botanists have noted that when ional cells are centrifuged, the nucleolus behaves as if it were heavier than the rest of the nuclear material and is thrown to the centrifugal end of the nucleus. Andrews 3 M5. Often the nucleus is so forcibly moved that it breaks through the nuclear membrane. In animal cells, the nucleolus is also moved by centrifugal force. At any rate, this is true in the case of immature eggs with large germinal vesicle, CL Ashley 6. In oocytes, the nucleoli move under the influence of gravity. Thus, H. Buryak 95, in studying sections of the ovary of the lobster, noted that the nucleoli were always orientated toward the lower side of the nucleus. In the immature egg of the sea urchin Echinus esculentus, Gray 27 has measured the speed with which the nucleolus falls under the influence of gravity. It travels at the rate of 0.4 per second. Gray does not state the size of the nucleolus, but from his figures it can be seen to be about ingenious. If now we make the assumption that the difference between the specific gravity of the nucleolus and the nuclear fluid is 0.1, the viscosity of the nuclear fluid, as calculated from Stokes' law, is 0.02 or 2 times the viscosity of water. This value must be very near the correct one. Obviously, it cannot be much too high, and it cannot be much too low. For the only value about which there is any doubt is the value we have taken for the difference in specific gravity between nucleolus and nuclear fluid. The specific gravity of the nuclear fluid must be about 1.04. That of seawater is 1.03. It would hardly be possible for the specific gravity of the nucleolus to be very much greater than 1.14, for there are no substances in protoplasm which have a much higher specific gravity. The specific gravity of proteins typically does not exceed 1.3 Di. Chapter 7. The elasticity of protoplasm. When a solid body is distorted, it tends to regain its original shape, i.e., it is elastic. Liquids and gases, when compressed into a smaller volume, expand to their original volumes when the pressure is released. They thus exhibit volume elasticity, but they do not ordinarily possess elasticity of form. However, thin films of liquid do regain their form following distortion. A soap bubble subjected to expressure returns to its original shape when the pressure is released. As is well known, a surface tension film can behave like an elastic membrane. Moreover, even in mass solutions, which we ordinarily regard as fluid, may occasionally show form elasticity. Thus, a gelatin sol may have elastic properties in spite of the fact that it flows fairly easily. In the case of the elastic gelatin sol, its fluidity or viscosity is a function of the pressure or shearing force to which it is subjected. If the pressure is low enough, there is no flow at all. Only when a certain initial resistance is overcome does the sol exhibit the properties of a fluid. This is a characteristic of many types of substances. Probably it is more exact to speak of these substances as plastic solids rather than as liquids. At any rate, this practice is followed by many recent authors. A plastic solid or an elastic sol owes its elasticity to the presence of structural elements, which are often in the shape of fibers. The interlacing of fibrillar micelli provides a structure which offers resistance to distortion, and the result is a certain degree of elasticity. Under pressure, the structure tends to be broken down, and as the pressure is increased, there is an increase in fluidity, or in other words, a decrease in viscosity. In the case of some colloidal solutions, mere shaking is sufficient to produce a marked change in fluidity. Thus, vanadium pentoxide in concentrated colloidal solution is a pasty gel. When the gel is shaken, it becomes quite fluid. Various other colloidal solutions behave in the same fashion. They are then said to be thixotropic, and the phenomenon is called thixotropy. See Friction on 27. When a colloidal solution has a definite form independent of a containing vessel, it is always elastic. If, on the other hand, a colloidal solution is capable of flow, it may or may not have elastic properties. Freundlich and Seyfried describe a method for demonstrating the elasticity of colloidal solutions. A tiny sphere of some magnetic metal is suspended in the solution. It is then drawn to one side by a magnet. If the metallic particle S-returns back to its original position after the magnetic field is removed, this is certain evidence that the solution is elastic. A more usual method of demonstrating the elasticity of colloidal solutions is to measure their elasticity at various rates of shear. True fluids have the same viscosity no matter what the rate of shear. On the other hand, elastic fluids or plastic solids have a lower viscosity as the rate of shear increases. Thus, to choose only a single example of many such observations. Freundlich and Abramson, 27, in measuring the viscosity of an 89% gelatin solution in a Hess viscosimeter, found that the viscosity went through a whole series of diminishing values as the pressure on the solution was increased. Concerning the elastic properties of protoplasm, there is very little precise information. Probably every worker who has exoppermeared with living animal cells knows that they regain their original form following distortion. Plant cells are usually surrounded by a stiff cell wall, so that this property is not so evident. But with animal cells, sea urchin eggs for example, one could hardly escape knowing that the cells flatten under the pressure of a cover slip, only to regain their shape again when the pressure is removed. This ability to resist distortion must depend in part on the surface tension of the film surrounding the egg. It must also depend on the elasticity of the membrane of the egg, which we have shown previously to be solid and therefore elastic, cp. 13. Whether the interior of the egg is elastic or not is a problem which requires further study than the mere huge observation of the elastic properties of the egg as a whole. That cells are elastic, that is to say that they regain their form following distortion, has been known since the time of Leeuwenhoek. Practically all the literature concerns blood cells. Leeuwenhoek noted that red blood cells were frequently distorted when they passed through narrow vessels, but that they regained their original shape following distortion. This observation has been repeated by numerous subsequent workers. As early as 1822, J.C. Schmidt cites 13 authorities, all of whom had noted the elasticity of circulating red blood cells. To quote from Schmidt, die Blutkörner beim Durchgehen durch die kleinsten Gefabe oder Gauge gedrückt und damit verlangert werden, dass sie aber nachher wieder ihre runde Gestalt annehmen. Dieses sahen und glauben Leeuwenhoek, W. Kaupeck, FW Kauf, H. Bakes, H. Miles, J. Keel, Senac, Fontana, FRB, Albin, El Koldani, G. Beich, Spollanzani, Hunter, Blumenbach, Poli, J. Dollinger. Most of these references are from the 18th century. For complete citations, see Schmidt's paper. Following Leeuwenhoek's observations, many authors experimented on the elasticity of red blood cells. Such experiments were not uncommon in the 18th century and in the early part of the 19th. The large corpuscles of salamander blood were soon recognized to be aesgetially favorable material. In some experiments, the circulating blood of the salamander lung was compressed between glass or mica plates, and it was found that the size of the corpuscles might increase 3 or 4 or even 5-fold. Upon the removal of pressure, the corpuscles regained their original form. To quote Schmidt, 1822 again, Delator will auch von dem Drucke zweier Glimmbratschen, zwischen welche man das Blut presst, die Gestalt der Blutkörner vertodert und nach aufgehobenem Drucke wiederhergestellt gesehen haben. Auch F. Fontana W.D.R. sie zwischen zwei Glimmerblättchen so zusammengedrückt haben, da sie 5-mal breiter geworden seien nach aufgehobenem Drucke, aber ihre wahre Gestalt wieder angenommen hatten, for later observations of the same sort. C. Asherson 38, Wagner 38, Rollet 71. Rollet 71 also cites several other references on the same subject. Histas 42, Seep 93, and Voyager 66 may also be consulted. Stricker, this is probably a reference to H. Miles 167, studied the elasticity of white blood cells and showed that they might be stretched to 3 or 4 times their original length. In recent years, there has been frequent reference to the elasticity of protoplasm, and it has often been stated that the living substance is highly elastic. See, for example, CFWIS 26A actual data have been viewed. CFWIS 26B inserted microdissection needles into an amphibian red blood cell and found that it could be stretched to 3 times its original length, and would then return to its original size if the tension were removed. Sierres is apparently unaware of the older literature in which the same observation was made without any possibility of an injury effect from the insertion of the needles into the cell. Granting that blood cells can be stretched and will then regain their original form, one does not know how much of this effect is due to surface tension, how much to the elastic properties of the cell membrane, and how much, if any, to the elastic properties of the interior protoplasm. Safeas himself is inclined to regard only the cell membrane as elastic. He calls it the cell wall. On the basis of some interesting reasoning, Pondy 24 had already pointed out that the elasticity of the red blood cell was due entirely to its membrane. So with 27 experimented with the protoplasmic strands of various plant cells. He states that he moved a needle back and forth in the axis of a strand of a Trade-Scantius cell, the respective portions of the latter, i.e. the strand, lengthened or shortened elastically with little or no slip of the needle. Again, in Spirogyra, the nucleus may be pushed from end to end of the cell and immediately recoils to its original position when released. These observations of Soeth are of doubtful value. The mere fact that there is a strand in a cell visibly limited from the vacuole is an indication that the strand must have a physical surface. The apparent elasticity of the strand as a whole might be due, i.e., to the surface tension of this surface. Concerning the actual structure of the protoplasmic strands of plant cells, we know peculiarly little. Is the flowing protoplasm of these strands surrounded by the delicate tube-like membrane, which prevents it from diffusing into the surrounding vacuole? Or do the protoplasmic granules move along a slender filament W each occupies the center of the strand? Until we know more concerning the true morphology of the protoplasmic strands of plant cells, it is rather difficult to interpret observations concerning their elasticity. It is rather interesting to compare recent statements regarding elasticity of protoplasm with the statements of older workers, Istioeli and Schwendehi, 67 CP 403. After pointing out that when a Spirogyra cell is treated with a sugar solution, it forms long threads between the cell body and the cell wall. State that if these threads are cut, the protoplasm of the thread is pulled back to the main mass of the cell. But they indicate clearly that this is not necessary evidence of elasticity. Apparently, in 1867, one was well informed regarding surface tension phenomena. From what has gone before, it must be obvious that the question of the elasticity or non-elasticity of the cell as a double yaw is an idle one anyone at all conversant with. The properties of matter must realize that the surface tension of the cell in itself makes it resist distortion, and further that the presence of a rigid membrane, such as is typical for animal cells, likewise bestows elasticity on the cell as a whole. A much more important question is the problem of the elasticity of the protoplasm of the cell interior. In the folio wing discussion, we shall consider first the exertissable elasticity of the protoplasm of the interior of plant and animal cells, and second, the elasticity of the cell membrane of animal cells. Very few workers have undertaken a measurement of the elasticity of the protoplasm, which is contained in cells. In 1924, Siebes published a paper with the title, An Elastic Value of Protoplasm, and this paper has often been quoted as indicating that the protoplasm of echinoderm eggs and protoplasm in general is elastic. In view of the fact that Siebes' paper constitutes one of the few attempts to measure the elasticity of protoplasm, his data will be considered in detail. Siebes cites only a single experiment. This was made with an Echinarachnius, sand dollar egg. Into this egg, Siebes injected a nickel ball 16 microns in diameter. It has already been pointed out that such a procedure would be certain to produce severe injury. CP 64. But for the purpose of discussion, Y will assume that the egg in question remained uninjured. The Echinarachnius egg measures 140 Y, and according to Siebes, there is a distinct cortical zone whose depth is somewhat less than 1/10 of the diameter of the egg. Into this cortical zone, somewhat less than 14 UJL in thickness, the 16 nickel particle was embedded. From these figures, it is quite obvious that the particle was in direct contact with the outer membrane of the egg. When the current of an electromagnet was turned on and the egg was held Bg2 needles, the nickel particle moved approximately 9 Siebes does not definitely state whether or not the nickel particle returned to its original position after the magnetic field WA is removed. Apparently, it did return, for he made no correction in his stretching value for such a failure to return. How it could have failed to return is difficult to understand. When the nickel particle moved, it must have stretched the egg membrane, and the elasticity of this membrane was certainly sufficient to restore the nickel particle to its original position. It is thus obvious that Siebes' experiment gives us no real information regarding the elasticity of the interior protoplasm of the Echinarachnius egg. We do not even know whether or not this protoplasm is elastic. In the same paper in W. he makes the elasticity test in a short section on viscosity. Series describes one or two tests in which a nickel ball moved through the center of the egg and apparently did not return after the magnetic field was removed. This may indicate either absence of elasticity or adhesion to the outer cortex or membrane of the egg. An elastic fluid is a fluid WWD, an internal structure. If it is subjected to pressure or distortion, the structure tends to break down, and this is reflected in a more or less pronounced decrease in viscosity. Here, then, is a means of determining whether or not the interior protoplasm of living cells is elastic. Some very SDG observations are recorded in a paper of Heilbrunn, 22. It will be remembered that this worker introduced tiny iron rods into the plasmodia of various myxomycetes. These rods were then made to rotate as a result of their being exposed to a magnetic field from an electromagnet. The strength lie of the magnetic field, as determined by the amperage of the current WH passed through it, was then taken as a measure of the viscosity. It is interesting that Heilbrunn never makes any mention of the return of the iron rods to their original geodesition, and from his method of describing his experiments, it is quite certain that no such return occurred. It is thus apparent that the interior protoplasm of slime mold plasmodia is not highly elastic. But the most important experiments of Heilbrunn from the standpoint of the elasticity question are the experiments in which he repeatedly twisted the non-rods in the attempt to discover whether such repeated twistings had an effect on the viscosity of the protoplasm. These experiments V2 sitter shall now proceed to consider. The results obtained by Heilbrunn were of two sorts. In the first place, he found that for reticidaria, which has relatively large plasmodia, the viscosity showed no decrease following frequent twistings of an iron rod. The experiment was carried out in the following way. An iron rod within a reticularia plasmodium was placed at right angles to the lines of force of the magnetic field. The number of amperes current necessary to turn it parallel to the lines of force was measured. Then the stage of the microscope was turned through 90 U and the measurement repeated. The only experiment cited by Heilbrunn, the measurements of viscosity were repeated at minute intervals. It was found that the viscosity, as determined by the amperage of the current passed through the electromagnet, remained constant for 18 successive determinations at a value of 3.5 amperes. Then the viscosity increased to a value of 3.6 amperes and remained at this value for 7 more determinations, following which the experiment was discontinued. It is thus apparent that the movement of an iron rod through the protoplasm, if reticularia plasmodia, does not produce a viscosity decrease. This is strong evidence that the protoplasm of this type of plasmodium is inelastic. On the other hand, when the smaller plasmodia Badahamia utricular and Physarum cinereum were studied, the viscosity EVA is found to drop following frequent determinations. In the case of Badahamia utricularis, viscosity determinations could only be made at intervals of several minutes. The plasmodium is so small that the iron rod had to be oriented with a needle following each measurement in order to make certain that it had room to turn. Heilbrunn is of the opinion that the insertion of the needle may very well have had an effect on the viscosity value, and he cites an experiment in support of this view. Following 12 successive determinations on a Badahamia plasmodium, the cerium plasmodia resemble those of Badahamia, in that there is barely room enough for the iron rod to turn. In this form also, Heilbrunn found the viscosity to decrease following successive measurements. Thus, after 11 successive measurements, the viscosity value was 1.9, as compared with an initial value of 2.2. 9 subsequent measurements resulted in no further decrease in viscosity. There are 2 possible interpretations of these experiments of Heilbrunn's. At first sight, we would conclude that the protoplasm of meticularia is inelastic, whereas that of Badahamia and Fusarium is elastic. But it must be remembered that although the plasmodia of reticularia are large, those of Badahamia and Physarum are small. In the latter 2 forms, the turning of the iron rod tends to be prevented by the presence of the ectodasm. As has been shown previously, CP64, the ectoplasm of Myxomaceti plasmodia is certainly a gel, and it is doubtless elastic. Hence, in Batomia and Fusarium, the observed elastic effect is due not to the internal protoplasm, but to the outer ectoplasm. If we accept this second interpretation, we are no longer faced with the necessity of assuming that two closely similar types of living substance differ in a fundamental progeny. We conclude, therefore, that as far as the evidence goes, the internal protoplasm of slime mold plasmodia is inelastic. There is an interesting experiment of Rumbler 2T4, which should be cited in this connection. Rumbler studied the effect of pressure on the speed of rotation of O'Hara protoplasm. The cells of the alga O'Hara were mounted on a slide, and with the aid of a special apparatus, pressure was applied to the cover slip from above. Pressures up to 4.6 atmospheres had no effect on the rate of hydrotoplasmic streaming. The hydrotoplasm flowed neither more slowly nor more rapidly. A similar result had, as a matter of fact, previously been obtained by Harmax in '98. This author subjected cells of Nutella in a glass tube to a pressure of 2 atmospheres. In these experiments, the tube containing the cells was connected with a manometer tube filled with mercury. Hormak found that a pressure of 2 atmospheres had no effect on the rate of streaming. See also Laxterturbach 21. At first sight, it would seem that the experiments of Harminer and Rumbler offer proof of the inelasticity of the streaming protoplasm. For if the protoplasm were an elastic fluid or a plastic solid, we would expect a decrease in viscosity and an increase in the rate of flow with increase in pressure. But it should be pointed out that the pressure applied to an entire cell of cham or Nutella is hardly comparable to the pressure which is applied to gelatine as it is forced to flow through a capillary tube. We are probably not justified, therefore, in regarding the Homerite Jimbin experiment as a demonstration of the inelasticity of the protoplasm of the cells of O'Hara or Nutella. As mentioned previously, a standard method of determining whether or not a fluid is elastic is to make viscosity measurements at varying rates of shear. If the viscosity remains constant, the fluid is elastic. Thus, for example, if we measure the rate of flow through a capillary tube, in the case of an inelastic or true fluid, the speed is proportional to the pressure. The quid obeys Poiseuille's law for the speed of flow of liquids through tubes, and the viscosity remains constant as the pressure is varied. On the other hand, if the material we are studying is elastic, there is no longer a strict proportionality between the speed of flow and the pressure, for the viscosity becomes less and less as the pressure is increased. In general, no matter what taijo of viscosity measurement is employed, for elastic materials, the viscosity becomes less as the shearing force increases, whereas for inelastic materials, the viscosity remains constant as the shearing force changes. In using the centrifugal method to measure protoplasmic viscosity, it is a simple matter to vary the shearing force. One needs only turn the centrifuge at different rates of speed. The centrifugal force varies as the square of the number of turns per second. In a series of tests on the protoplasm of Gumingia eggs, Hurled 1026A, the centrifuge handle of a small hand centrifuge was turned once per second, once every 2 seconds, once every 3 seconds, and once every 4 seconds. The speed of movement of the protoplasmic granules was determined for each of these rates of turning. In the Gumingia egg, the fat granules move a little more rapidly than do the pigment granules. For the fat granules to move far enough so as to give the appearance of a definite zone at one end of the egg is noted in the second column, under T. The time in seconds necessary for the pigment granules to move far enough so as to give the appearance of a definite zone is shown in the fourth column, under T. In the first column, under S, are shown the number of seconds required for a single turn of the centrifuge handle. If the protoplasmic fluid, through which the grannies move as a true fluid, their speed of movement should vary directly with the centrifugal force, that is to say, with the square of the number of turns the centrifuge tubes make per second. From any one value for granule speed, it is easy to calculate what the speed would be for other amounts of centrifugal force, on the assumption that the granules really for TNT were obtained from the second series of values. The calculated values for T and T are given in the third and fifth columns respectively, are close to the speeds calculated on the assumption that the protoplasm through which the granules travel is a true fluid. The agreement is particularly satisfactory for the last two values in each case. When the centrifuge is turned at top speed, at the rate of 1 turn of the handle in 1 second, the observed value for the time of granular movement is too low. This is readily understandable from the conditions of the experiment. When the centrifuge handle is turned fast, it is not possible to stop it immediately following the exoneration of a given number of turns. Thus, when the handle is turned 2 times in 2 seconds, it continues to move for a good fraction of a second before it can be stowed peat. The percentage of such error is also greater when the total number of turns is small. If one calculates the protoplasmic viscosity for different amounts, which however happen to be close to the actual values, in terms of the viscosity of water, centrifugal force in terms of gravity, viscosity of Kimingia egg protoplasm in arbitrary, obviously, the viscosity remains constant as the centrifugal force varies. As pointed out previously, the value obtained with very high centrifugal force is not accurate, for it is impossible to stop the centrifuge quickly enough. Such an error would tend to make the viscosity value too low, and this is apparently the explanation of the low value obtained with a force 4968 times gravity. Attention should be called to the fact that the variation of shearing force in the above tests is greater than that usually found in similar experiments of colloid chemists on inanimate material. It should also be noted that the shearing force is not excessively great, for the speed of the moving granules is really very slow. Thus, at slower centrifugal speeds, the granules only travel 15 or 20 microns in a minute. The data that we have presented are not very extensive as far as they go. They show that the protoplasm of the Kimingia egg is not elastic. Other unpublished results tend to bear out this conclusion not only for the Kimingia egg, but for the Arbacia egg as well. From the A33 Casseting discussion, it is apparent that in spite of the various papers that have been written on the elasticity of protoplasm, there are in the entire literature only 2 experiments which really give any information concerning the elasticity of the internal protoplasm of cells. At any rate, only 2 such experiments have been discovered in a search of the literature. Heilbrunn's experiment in Beticularia plasmodia shows the internal protoplasm in this species to be inelastic. The centrifuge experiment of Heilbrunn also indicates an inelasticity of the protoplasm of the Kimingia egg. One would hardly be able to conclude much from these 2 experiments if it were not for the fact that there is some excellent negative evidence which supports the view that undifferentiated protoplasm is typically inelastic. When elastic materials are subjected to a one-sided pressure, they are always double-eye refractive. Now, in spite of the fact that many observers have studied cells under cross-nickels, no one has ever described any type of undifferentiated protoplasm, which is doubly refractive. When a biologist studies cells, he usually subjects them to the pressure of a cover slip, and as the preparation dries, this pressure may be considerable. The conditions of observation would therefore be favorable for the demonstration of anisotropy, if it were present. As a matter of fact, the cell membranes can be seen to be doubly refractive. But in spite of the fact that the polarization microscope was in very common use in the middle of the last century, there is apparently no record of any description of undifferentiated protoplasm as doubly refractive. Valentin 61 states specifically that the protoplasm of various egg cells is isotropic. And Dippel 72 P324 states categorically, sämtliche nur aus Protoplasma aufgebaute, nicht kristallisierte Inhaltskörper ohne Starkey-Einschluss modifizieren das polarisierte Licht in keiner Weise und geben sich somit als einfach brechende zu erkennen. An especially interesting observation is that of Valentin 64. He studied streaming plant protoplasm under a polarization microscope. From his careful description of the anisotropy of an occasional crystal and his failure to remark on the anisotropy of any other elements, it seems certain that the main mass of the flowing protoplasm was isotropic. An elastic fluid could never appear as isotropic when it was in a state of flow. It must not be thought that all protoplasm in cell interiors is inelastic. On the contrary, there are certain indications that in many cells there are at least elastic elements in the protoplasm. We have already had occasion to refer to the presence of definite fibrillar elements in some types of cells, CP17 and P80. Such fibrils are beyond any question elastic, as the very fact that they possess form indicates. Even in marine eggs, in which the main mass of the protoplasm is inelastic, elastic fibers appear during cell division. When unfertilized Gomingia eggs are centrifuged, although most of the protoplasmic granules move through the cell readily enough. Here and there, occasional granules can be seen to be entangled in the astral rays of the mitotic spindle of the first maturation division. Compare Heilbrunn 2060, P273. These astral rays are surely elastic. This is very evident in the Arbacia egg, in which the astral radiations can be seen to exert a pull or tension on the enclosing membrane of the cell to which they are attached. This membrane, the so-called hyaline layer, is at first perfectly smooth in contour, but with the appearance of the amphiaster it becomes indented, so that it is often very obviously crenate, see Heilbrunn 28, p. 222. Almost certainly these indentations are the result of a pull of the astral ray fibers, and so Heilbrunn interpreted them. The uneven appearance of the egg contour becomes exaggerated when the eggs are centrifuged. Then the astral ray fibers exert a still stronger pull on the periphery of the cell, so that the cell is often very much distorted. The astral ray fibers are gels, and they are apparently thixotropic gels. This follows from the fact that shaking results in a disappearance of the astral radiations. Wilson once shook Sorchin eggs violently just as they began to divide. The division was suppressed, and the astral rays disappeared. See Wilson's of the sea urchin egg could be prevented by pressure. Seapiz 24 A also describes the disappearance of the mitotic spindle when eggs of Exnemochnius are subjected to pressure. There can be no question but that the visible fibrils or fibers contained in protoplasm or forming a part of protoplasm are elastic. Such a statement hardly requires proof. It also seems probable that the dispersion medium of protoplasm, that is to say the medium in which granules and fibrils are suspended, is without elasticity in many, if not in all types of cells. This follows both from the experimental evidence of Heilbrunn and Heilbrunn, and from the fact that undifferentiated protoplasm is AOT doubly refractive. Further tests of the elasticity of protoplasm seem desirable. Whenever a cell is capable of having its viscosity tested by the 3-centrifuge method, it is possible to decide whether or not the protoplasm is elastic. One has only to make a series of tests at different centrifugal speeds. If in these tests the viscosity remains constant, then it seems certain that the protoplasm is inelastic. The second phase of our subject deals with the elasticity of the cell membranes of animal cells. These membranes are certainly rigid, and there is thus no doubt but that they are elastic. It therefore becomes of interest to Attini. A measurement of this elasticity. The deformation or flattening of sea urchin eggs when they came to rest on a flat surface. In Volb's terminology, if the horizontal diameter of the eggs is at A, and the vertical diameter, by then the eccentricity of the eggs can be expressed by the fraction for normal eggs, surrounded by jelly valves, found this eccentricity to average 0.12 when the eggs came to rest on a glass plate. If the jelly was washed off with 3% KCN, the eccentricity was found to be 0.026. From these values, Vallès calculates the surface forces of the egg on the basis of an empirical formula, which he obtains from an experimental study of small droplets of various liquids. According to Vallès, the surface forces include the following. Test forces de tension superficielle au sens capillaire de séparation. Forces hydrofaciales exercises à la limite entre les liquides extérieurs et les substances de phases différentes constituant la périphérie de peau. Les forces élastiques développées par des membranes enveloppantes. Et enfin, et surtout, les composantes superficielles des forces de rigidité des gels intérieurs. Ces y is partout des formations mécaniques dont peuvent être logiques. From the previous discussion, it is apparent that the last mentioned forces do not exist. The surface forces of VLs can then be taken to include surface tension forces and forces due to the elasticity of the egg membrane. For these combined forces, Vles calculates a total force of 10 to 2 or 5 dynes per centimeter. There is also another possible way in which one can study the elastic and surface tension forces of the membrane of a cell. When a spherical cell, such as for example a sea urchin egg, is placed in a hypotonic solution, the cell does not enlarge to the volume it would occupy if no elastic membrane were present. Thus, if Arbacia eggs are placed in a solution of 75% seawater and 25% distilled water, we would expect that the volume of the cell would increase until the concentrations of electrolytes inside and outside were the same. In other words, we would expect the cell to increase in volume until the final volume would be to the original volume as 75, or 4 to 3 actually when a Sorchin egg is placed in 75% seawater. It does not become 1.33 times as large, but only 1.28 times as large as it was before exposure to the hypotonic solution. Unpublished experiments. The difference is due both to the elastic properties of the membrane and to surface tension forces. It is interesting to note that when a Gomingia egg is placed in a 75% solution of seawater, it increases to only 125 times its original volume. This lesser increase is to be associated with the greater thickness and rigidity of the membrane surrounding the Gomingia egg. In a study of the volume changes of spherical cells in hypotonic solutions, it is also possible, to some extent at least, to separate the effects due to surface tension and those due to elasticity or rigidity of the membrane. When a surface tension film is stretched, the force which tends to diminish its surface suffers no increase. On the other hand, when a rigid membrane is stretched, the force tending to decrease the surface becomes ever larger. If we place cells like sea urchin eggs in a series of hypotonic solutions of decreasing strength, the cells increase more and more in volume. There is no change in the surface tension, force tending to prevent enlargement of the cell. But there is a very pronounced change in the force due to the elasticity of the rigid membrane surrounding the cell. When Arbaceous cells are placed in 50% seawater, the increase in volume of the egg is very much less than we would expect if the egg were free from the constraint of a surrounding membrane. The surface tension effect is no greater for the 50% solution than for the 75% solution. And making measurements of increase in volume of eggs in hypotonic solutions, too great a time must not be allowed to elapse before THC measurement is made. As water enters the egg, it produces changes through the protoplasm, which probably result in the freeing of electrolytes. It will be remembered that the sea urchin egg contains a large excess of bound electrolytes, CP25. Hence, in proportion to the greatly increased pressure of the elastically stretched membrane, the surface tension force becomes negligible. We are thus in a position to make a direct study of the elastic properties of the rigid membrane surrounding the sea urchin egg. We might, for example, study the changes in elasticity which the membrane undergoes when it is placed in contact with various reagents, provided that these reagents have no very great effect on the permeability and hence on the osmotic forces acting on the membrane. Or it might be possible to determine the modulus of elasticity of the membrane. It is not a very difficult matter to derive an equation for the case of an elastic membrane stretched by a pressure pushing it out from within. Such an equation has indeed been published by F. Bank 1010, and if one is willing to make certain assumptions regarding the osmotic forces acting on the membrane, one might perhaps use the equation to calculate absolute values for the modulus of elasticity. It must not be forgotten that as the volume of the cell increases, there is probably a release of some of the bound electrolytes in the protoplasm. We've already seen that these are present in abundance, CpH25. If bound electrolytes are set free as the cell enlarges, they would tend to make the cell expand even further, and this would have the effect of making the elasticity values too low. There is also a difficulty owing to the presence of granules within the protoplasm. It is possible that the granular volume does not increase at all when the eggs are ex-delaced in hypotonic solutions. If this were the case, it would in itself account for the fact that sea urchin eggs increase to only 1/28 times their original volume in 75% seawater. But it would not account for the results obtained with 50% seawater. The granular volume of eggs in different solutions can be roughly measured in centrifuged eggs, so that the influence of changes in granular volume can be determined. It is thus probable that in spite of difficulties of one sort or another, experiments with hypotonic solutions may furnish valuable information regarding the elastic properties of the cell membrane. Further investigation is therefore likely to yield results of considerable interest. Chapter 8. The action of temperature. For the most part, life is only possible within very narrow limits of heat and cold. The natural temperatures of our planet do not cover a very wide range, and yet many plants and animals are quite unable to withstand the extremes of the temperatures they are subjected to. Both in the tropics and in the Arctic regions, living organisms are very frequently killed by the heat or the cold of their ordinary environment. And even in the temperate zone, such heat or cold death is by no means uncommon. In many instances, the distribution of animals and plants is governed by their ability to withstand high or low temperatures a priori. It seems very likely that the action of heat and cold on protoplasm finds expression in colloidal changes. Experiment shows this prediction to be verified, but the action of temperature is not entirely lethal. Between the limits of heat and cold in which protoplasm can remain alive, there is a range of temperatures which can produce marked effects on the life and activity of the cell. Some of these effects are doubtless due to a direct influence of the temperature on the rate of chemical reaction within the cell. Many authors have stressed this aspect of the subject, and some of them have completely neglected the physical effects which temperature change might have. From the standpoint of physical chemistry, it is evident that the rate of chemical reaction in a heterogeneous system is dependent on the physical properties of the various phases of the system, especially on the degree of dispersion, if the system be a colloidal. It is thus of interest to inquire whether the ordinary temperatures of the environment can produce recognizable changes in the physical properties of protoplasm, and whether such changes, if observable, can be correlated with known differentiation of temperature rhesus in the behavior of protoplasm at one temperature or another. There is, however, a more direct interest in the study of the action of temperature on protogelasm. The physical changes which occur in protoplasm as the temperature is raised or lowered may very well throw light on some of the fundamental properties of the living colloid. Of all the independent variables of the cell, temperature is perhaps easiest to control. If we change the chemical constituents of the environment, we have no certain knowledge of how far such a change is passed on to the cell interior. But when we place cells at a given temperature, we can feel certain that we know the temperature of the interior protoplasm. In discussing the action of temperature, we shall consider the following topics. 1. The changes produced by moderate temperatures. 2. The heat death of the cell. 3. Death due to low temperatures. Most fluids undergo a progressive decrease in viscosity as the temperature is raised. That such a decrease in viscosity also occurs in protogelasm is indicated by the results of PACH-TB IA Edmite 13. He measured the viscosity of the protoplasm of Ascaris megalocephala eggs by noting the length of time necessary for the displacement of mitochondria granules when the eggs were centrifuged. PACH-B evented results are given in the following to throw the mitochondria from one end of the cell to the other. Fobe Fbmia used a centrifugal speed of 2,500 turns per minute, but the radius of turn is not given in his description of his experiments. Ratchers, according to Fbmia, T3 time, that is relative viscosity, 4 minutes. Fobemia's data show that the viscosity of Ascaris egg protoplasm is decreased 4 or 5-fold by a rise in temperature of 10 degrees. An older observation of Hogho also indicates a decrease in the viscosity of Ascaris egg protoplasm as the temperature is raised. She found that the Brownian movement of granules in centrifuged Ascaris eggs is more rapid at higher temperatures. It should be noted that the optimal temperature for the development of Ascaris megalocephala eggs is 37 C-bond 25. Thus, the temperatures studied by Fawi are all below that optimal or normal for development. If one compares the behavior of various types of protoplasm in relation to temperature, one often forgets that the optimal temperature is very different for different organisms. A temperature of 37 is normal for Ascaris, but sea urchin eggs exposed to this temperature are killed in a minute or two. For the eggs of the Annelid nereis diversicolor, Pannon 24 also found that the viscosity of the protoplasm decreased as the temperature was raised. Pachton determined the viscosity by centrifuging the eggs. He made measurements at only 4, which the viscosity at 10 is taken as unit 3. Relative viscosity Panton's results agree very well with the older results of F and G Weber 17 for plant cells. These authors measured the viscosity of the starch sheath cells of the stem of Phaseolus multiflorus, a leguminous plant, at different temperatures. The viscosity was determined by measuring the speed of fall of starch grains through cells and section slices of the stem. Apparently, the experimental error was not great, for when a series of tests was made, the same value for the viscosity was always obtained. The AOCO of temperature, it made no difference whether the protoplasm was heated to a given temperature or cooled to a given temperature. The viscosity value remained the same. This is an important point, and one not considered by other authors who have studied the effects of temperature on protoplasmic viscosity. The results of F and G Weber are given below in tabular form. The time of fall is in seconds and is a measure of the viscosity. Comparisons of viscosity at various temperatures should only be made for a given cell. Each row of figures represents the data for such a cell. Is to say, the ratio of the viscosity at a given temperature to the viscosity at a temperature 10 degrees higher. The values for this temperature coefficient decrease for higher temperatures. This is typical for water and watery solutions, and the Webers point out that the values for QUTQ obtained by them are very similar to the QO values which have been found for protein solutions and for blood plasma. In a later paper, Webeb and a co-worker made another study of the effect of temperature on the protoplasmic viscosity of the Xeob 23. In this work, they studied only the action of low temperatures. In order to avoid any possible pathological effects due to sectioning, the stems were centrifuged whole and were then sectioned just prior to examination. It was found that when the temperature was lowered to 2 ohm, the viscosity increased at least 4-fold and probably more. This viscosity increase occurred after exposure of only a few minutes, 5 minutes in one experiment. When the plants were again brought to room temperature, the viscosity returned to its original value, so that the effect of cold was reversible. The highest temperature for which they give data is one of 4 tomb temperature. They record a viscosity increase as compared with room temperature of at least 3-fold. It is clear that the viscosity increases described by Webe and Hohenio-Geb are somewhat greater than those previously thus in 2 experiments, F and G. Webe found the viscosity at 0 APG are approximately twice as great as that at 20 UA, whereas Webb and Hoheksegi found the viscosity to increase at least 4-fold when the plants were cooled to about 0. Perhaps, as Webby and Hohenegi suggest, there may be an error involved in sectioning the plants, as was done by F and G Webby, see also P43, Fawe Femate F and G. Webby and Pans have all obtained results which indicate that the protoplasmic viscosity in the cells studied by them decreases regularly as the temperature is raised. That this is not the case in all cells was shown by Bill Bankson 20. He found that in Arbacia eggs the viscosity was lower in the neighborhood of the freezing point than at room temperatures. In his description of his experiments, Bill Bankson records the temperature as 3 times. Probably it was not as low as this, for he measured the temperature of the freezing mixture surrounding the tube containing the eggs, and not the temperature of the fluid immediately in contact with the eggs. This may involve an error, and from subsequent tests, it seems certain that the temperature of Gilbertrin's experiments was not as low as 3 times. In 1922, with the aid of his magnetic method, Heilbrunn determined the viscosity of the protoplasm of the slime mold Beticularia at different temperatures. Heilbrun's data appear to be very accurate, but his figures are given in terms of a relative measurement, which varies from experiment to experiment. For the convenience of the reader, Heilbrun's figures have been relative viscosity no longer measurable, that at 26 the average of 5 tests. Heilbrunn interprets the variable value at 35 E as indicating that the protoplasm is injured and has become separated into phases of high and low viscosity. If the measurements were made in different regions, as Heilbrunn's explanation indicates, it seems just as logical to assume that some parts of the protoplasm coagulate more quickly than others. Whether we accept this interpretation or not, it is obvious that the viscosity of the protoplasm drops to a minimum value before coagulation occurs. This is apparently be particularly noted that the viscosity of the protoplasm goes through a maximum at 16 to 17.5, and it is lower both above and below this temperature. Were made on slime mold plasmodia, which were resting directly on a glass surface, as pointed out previously, CP62. The viscosity of slime mold protoplasm is less when the plasmodia are placed on moist filter paper. For such slime molds on moist filter paper, the viscosity varies hardly at all for different temperature. Thus, Heilbrunn placed pieces of Eticularia on moist filter paper and warmed them to various temperatures. After an hour in contact with moisture at a given temperature, the viscosity was measured with the magnetic method. The results Heilbrunn is retained. Slime mold plasmodia on moist filter paper, Heilbrunn 22. Viscosity on glass at room temperature viscosity on moist filter paper after Inlier plasmodia, saturated with water, remains constant. Apparently below 17, there is an increase in viscosity instead of a decrease as previously recorded for plasmodia on glass. But there is also another possible interpretation of this lower viscosity at 12. When plasmodia are placed on moist filter paper, their viscosity decreases slowly. In the only 3 experiments cited by Heilbrunn, the time required for the viscosity to reach a constant low value was 30 minutes in one case, and an hour, and 10 minutes in the other 2 cases. It seems likely that at lower temperatures a longer time is required for equilibrium. In view of the fact that the above experiments were made at the expiration of an hour, it is quite possible that at 12 enough time had not elapsed for the protoplasmic viscosity to have reached its final minimal value. The fact that the viscosity remains constant between 17 and 30 is surely difficult to understand. It has been shown that for Arbacea eggs and for Reticularia plasmodia on glass, the viscosity may decrease as the temperature is lowered. The same phenomenon occurs in the egg of the Chlamydia. For the protoplasm of this egg, Heilbrunn 24 was able to plot a fairly conjugate temperature viscosity curve. This curve is shown in Figure 3. The abscissae are degrees centigrade, the ordinates relative viscosity units. The viscosity was determined by centrifuging the eggs. In order to obtain greater precision, the eggs were centrifuged relatively slowly. The centrifugal force used was 311 times gravity. The ordinates in the curve represent the number of seconds required to form the zones typical for the Gemingia egg, see Figure 1, P45. For each point on the curve, a number of tests had to be made, for of course the eggs could not be examined while they were being centrifuged. Thus, at 23T, it was shown that 17 turns of the centrifuge for 68 seconds did not produce zones in the egg, whereas 18 turns 72 seconds did produce zones. The viscosity was therefore given as 72. For the limits of accuracy of the method did not make it advisable to differentiate any closer than this. The centrifuging was done at room temperatures, and this introduces an error, especially when the viscosity is high, and when the temperature of the experiment differs markedly from that of the room. For this reason, the determinations at 0 and 3P are not to be regarded as very exact. They are rather to be thought of as minima, although other experiments at higher centrifugal speeds showed that the tests at the low temperature are not very far wrong. The viscosity of the protoplasm of the Gemingia egg goes through a maximum at about 1510. This is essentially the same result that was obtained by Heilbrunn for slime mold plasmodia on glass. It is noteworthy also that in addition to the sharp viscosity interreese that occurs at 3101, there is also a sudden viscosity increase at 1 above the freezing point. In both cases, when either the temperature is raised or lowered, there is a minimum in viscosity just before the marked increase in viscosity takes place. From what has gone before, it seems apparent that there are two types of temperature viscosity curves which may be obtained for protoplasm. Some cells show a maximum viscosity at around 15, whereas in other cells, the viscosity decreases progressively as the temperature is raised. That there are really two types of curves is not absolutely certain. For fair Fermiot's results were all obtained at temperatures below the optimum. Panton's data for the Naras egg are too few. His 4 measurements might almost be superimposed on the curve for the Gumingia egg. And finally, the Webers worked with section tissues, and their results were not exactly confirmed by Weber and Hohenegger on intact plants of the same species. Nevertheless, it does seem likely that the behavior of protoplasm toward temperature does show differences for different types of material. Fly protoplasm of the egg of Nereus is highly viscous. At any rate, this is true for the form of Nereus found at Wood's Hole, whereas the protoplasm of the Gumingia egg is relatively very fluid. One would hardly expect them to show the same behavior toward temperature change. It might be expected that if some types of protoplasm show a maximum in viscosity at 15, that this would be reflected in the relation which some biological processes show to temperature. There are at least several examples in the literature in which processes of one sort or another have shown a maximum at around 15. Thus, for example, Plautz-Even found a maximum in crossing over and Drosophila at 13. Vach, Dilovai, and Jacob 24 found a maximum, all respond to the pedal muscles of the snail helix, at 2101, and Pereira 24 describes a maximum in the irritability of frog muscle at about the same temperature. Moreover, it has been known for a long time that the temperature coefficient of various biological processes may go through a sharp change at about 15. Many instances of such a change are recorded in Conitz 15. See also Crozier 24. Numerous authors have studied the effect of temperature on protoplasmic streaming. Generally, the rate of flow decreases steadily as the temperature is raised. Nagili found the increase in speed to follow a geometrical progression. That is to say, a logarithmic curve is obtained if his results are plotted. On the other hand, VTOL7G found an arithmetic progression and his results, when plotted, fall along a straight line. Later investigators have tended to confirm Velton rather than Nageli. Figure 4 shows the curve plotted by Lambers, 25, for a cell of Nutella macronifa. The curve is particularly interesting because it shows that the order of the observations, as indicated by the numbers on the curve, that is to say, the same value is obtained when the protoplasm is heated or cooled to a given temperature. In no case is it possible to say definitely that increased speed of protoxoplasmic streaming is due solely to viscosity change. But the results of Lambers suggest that the protoplasm of Nutella undergoes a progressive decrease in viscosity as the temperature is raised. This is the view favored by Lambers, Ewart 3. 61 thinks that both viscosity change and a change in the motive force play a role in the increased speed of streaming at higher temperatures. He points out that whereas viscosity typically decreases less and less for each degree rise in temperature, the increments of streaming velocity progressively increase between 10 and 30 d. Such a progressive increase in the increment of streaming velocity might be due to the fact that the viscosity-temperature relation is of the type shown in Figure 3. That is to say that there is a maximum in viscosity at around 15 or 20 e. If this were the case, and if we assume further that the propelling force increases uniformly as the temperature is raised, it would obviously follow that at lower temperatures there would be a pig. Speed of protoplasmic streaming in a cell of Nutella at different temperatures after Lambers 25. Smaller increment in streaming velocity than at Hegler temperatures. In favor of the opinion that the action of temperature on the rate of streaming is primarily on the propelling force, are the oldest experiments on the effect of temperature on streaming, those of Dupuhet 37. It may be interesting to quote Dutbochet's words, La circulation existe chez la cara flexilia mais lente. Il y confond lentement peau dans laquelle la plante est plongée. La circulation s'atténue graduellement. UA devient extrêmement rapide. À plus 18 degrés, the action of temperature is sent. La chaleur de taux extant portée lentement à plus 27 degrés. La circulation devient extrêmement lente. Elle augmente ensuite peine à peine de vitesse sous influence. Continue de cette même température de plus 27 degrés cent. Et au bout de 2 heures, elle est devenue d'une grande rapidité. According to these observations, the rate of streaming first decreases as the temperature is raised from 18 to 27, and only after some time has elapsed does it become swift. We would expect that the effect of temperature on protoplasmic viscosity would be quick. If this were the case, Dubochet's results would indicate a decrease in protoplasmic viscosity as the temperature was raised from 18 to 27. The increase in the rate of streaming that followed long exposure to the higher temperature would then be due to a response of the plant, which involved an increase in the force which caused the streaming. But of course, this is purely theoretical, and as a matter of fact, Dubochet's results do not seem to be in accord with the results of later experimenters. See, for example, Iowa 3P62. Why certain types of hydrocolloid should show a maximum in viscosity of 15 is an interesting problem from a physical standpoint. Various theoretical possibilities have been thought of. But it seems best to leave the question open. It is a remarkable fact that the protoplasm of most animals in lands is killed by temperatures only a few degrees above that of the natural environment. Often enough, organisms in nature are destroyed by a slight increase in the temperature of their surroundings. Dave Pop 97 gives a list of the temperatures weekly produce death in various animals and geology. This list is far from complete, and some of the observations on which it is based are not reliable. It seems safe to say that animals generally die at temperatures between 30 and 45 degrees, and that plants die at temperatures between 40 and 50 degrees. There are, of course, quite a few exceptions. Fishes in temperate latitudes usually die at about 25 degrees. Some animals die at even lower temperatures. Thus, Bobby 77 found the death point of 2 phyllopods, 2P18 and 19, respectively. Perfeb for Seavolves 2P294 states that some plants die at temperatures as low as 20. The alga Hydroiris jotids dies at 16 Klebs 96. There are also exceptional cases in which organisms can live at much higher temperatures. The optimum temperature for thermophile bacteria is 60 to 70 C, and they are only at dit led by temperatures above 75 CPF for P87 and literature P96. There are also records of animal and plant life in hot springs, although these records should be regarded with caution, for the temperature varies greatly in different parts of a hot spring. An organism may have spent most of its life in a relatively cool part of a hot spring, and may then have been displaced into a warmer ET just before the observations were made, Pista. If they are able to exist in a dry state, various organisms are able to withstand temperatures far above those which are normally fatal. This has been known since the time of Spallanzani. Seeds, mosses, lichens, fungus spores, and bacteria, when dried, are often capable of continuing their existence after an epiocia of an hour or hours to temperatures of 100 or even 20. For the older literature, see Pfeffi 4P elder literature, see Pfeffi 293, a few forms of animal life can also be dried, and these too can resist livy temperatures when water is absent. Thus, Ryan 22 found that rotifers and tardigrades, when dried, could withstand temperatures up to 150 IA. Not only is it true that certain animals and plants in a dry state can suffer exposure to leaguer temperatures, but there is also evidence that TGLA organisms and tissues, which cannot stand drying, also become increasingly resistant to heat as their water content is diminished. This is a point which will be considered in greater detail later. The cause of heat death in animals and plants has frequently interested zoologists and botanists. In our discussion, we shall consider only the typical case in which the organism dies at a moderate temperature. The few rare instances in which plants or bacteria can exist at relatively very high temperatures would require special treatment and special study. Nor is the capacity of dried organisms to resist high temperatures a general attribute of protoplasm. The organisms Weikla can be almost completely freed from a vapor, are hardly in a living state when they are thoroughly dry. The most obvious explanation of heat death, and the one most often gyroposed, is that the proteins of the living cell become coagulated by heat in the same manner that egg albumin becomes coagulated when it is heated in a test tube. Actually, there is very little experimental evidence in support of this assumption. Bobby and Richardson 99 isolated various proteins from muscle the action of temi-eriture, and claimed that the temji-eritures at which these proteins coagulated also produced definite shortening in the intact muscle. Later, Biodi and Halbutak 4 obtained similar results for nerve. The work of these authors hardly affects our main problem. For the most part, they were interested in temperature recs well above those which kill the tissues in question. And WKGR, or not, heat coagulation of Chi proteins occurs in cells already killed. Moreover, the above-mentioned work of Biodi and his collaborators has been sharply criticized by Myas 9, who has cast grave doubt both as to the accuracy of their experimental results and the logic of their interpretation among botanists. Lepeshkin has consistently regarded the Gia Dietli of protoplasm as primarily due to a heat coagulation of the cell proteins see Lepeshkivus 10, the authors will have cited above, and many others as well. Believe that the lead death of the cell is directly due to a coagulation of proteins. Of course, no one doubts that when a cell is killed thither by heat or by some other agency, its proteins are typically coagulate. The question is whether, in death at higher temperatures, the direct effect of the heat on the proteins is the Uchi primary factor. Ordinarily, proteins are not coagulated by high temperatures, what do cause death in animals and plants. For this reason, various authors have suggested other possible explanations for the action of heat, OE protoplasm, when TST5 proposed the view that heat caused Dietli by asphyxiation. Mayer Mena 7 suggested that the heat pyridized an accumulation of acid. Neither of these authors has offered very direct evidence in support of his theory. It is certain that IVN protoplasm is subjected to temperatures just warm enough to cause death. It suffers a gelation or coagulation. Sachs 64 described a warm mistar de protoplasma, which he deduced from the cessation of J. unratuelasmio streaming. DeVays 71 describes the formation of new granules in heat-treated plant tissues, and this is good evidence of coagulation. The same phenomenon is noted by Lepeshkin 2,4 Espirogem cells. With the centrifuge, it is possible to show that a great increase in viscosity when cells are subjected to temperatures just high enough to cause death. This has already been indicated on the temperature viscosity curve for the protoplasm of the Gimarija egg. Fig. 3. Bilbamon 22 found a similar sharp increase in viscosity for the protoplasm of slime molds. The coagulation or increased viscosity produced by warmth precedes death. Sachs states that the warm mistar is reversible if the heat treatment is not too prolonged. Lepeshkin also finds a certain degree of reversibility. In his study of the heat coagulation of plasmolized Spherogyra cells 23, he divides the process into 4 stages. The first stage is an invisible one, which involves an increase in permeability. The second stage involves a visible change in the surface layers of the protoplasm. Many new granules appear, and the protoplasm no longer tends to present a regular, smooth, rounded surface. The third stage involves a complete coagulation of chloroplasts, and the fourth, a complete coagulation of the protoplasm. If one interrupts the heat coagulation after the second stage and returns the alga to room temperature, the granulation produced by high temperature disappears gradually, and there is no lasting injury. Finally, in marine eggs, it can be shown that the coagulation caused by heat occurs before death and is reversible. Thus, in one experiment, when Arbacia eggs were kept at a temperature of 32.3 Ki for 20 minutes, the protoplasmic viscosity increased greatly, but it returned again to its normal value following removal of the eggs to seawater. At room temperature, see Heilbrunn 24b. Thus, in many types of living cells, heat produces first a coagulation and then death. In marine eggs, the coagulation produced by heat precedes any visible death changes and is very probably the direct cause of death. Is the heat coagulation of living cells simply a protein heat coagulation? Some facts speak in favor of this view. If one plots a curve for the time it takes for the protoplasm to coagulate at different temperatures, the general shape of the curve is very similar to that found for inanimate protein. Figure 5 shows the coagulation time at different temperatures of Arbacia and Gumingia egg protoplasm. The time of coagulation was determined by making centrifuge tests at frequent intervals. If one compares the curves in Figure 5 with the curve obtained by Boulia for the heat coagulation of serum albumin, the resemblance is obvious. Moreover, both in protein heat coagulation and in the heat coagulation of protoplasm, the temperature coefficient is of the same order of magnitude. Kiak and Martust found that the temperature coefficient of the rate of coagulation per degree of temperature was 1 for hemoglobin and 1.91 for egg albumin. TF1 examines the curves in Figure 5. It is easy to see that for each degree rise in temperature, the increase in rate of coagulation, decrease in time, is generally between these two numbers. For the temperature coefficient of heat death in plants and animals, similar high values have been obtained. The temperature coefficient for 10 degrees, i.e., D is 13.8 for hemoglobin and about 635 for egg albumin. Colloid 24 finds the GQ for heat death in cells of the following plants: TM discantia, discolor, beta vulgaris, brassica oleracea, Elodia densa, Troparioldia glomerata, Pisum sativum, to be 26, 71, 80, 31, 43, 118. Colloid also cites older authors who have found similar values for the Kichio. Justly discards the somewhat lower values of Lepishine T3 as being due to faulty technique. In animal cells, high values for the death have also been found. These range from 240 to 1,000. See Kaitzmuff. In spite of the similarity of the temperature coefficient, there are serious objections to the view that the heat coagulation of protoplasm is due to a direct effect on the protein. The protoplasm of marine eggs is coagulated by temperatures of 30 to 35 E, and as has already been shown, heat death may occur at even lower temperatures. That proteins undergo heat coagulation at temperatures as low as 30 or 35 E is extremely doubtful. It is true that in the literature there are 1 or 2 descriptions of heat coagulation at these temperatures, but these observations were made on tissue extracts, and in these extracts, salts and other chemical compounds probably play a larger part in the coagulation than does the temperature. In pure preparations of proteins, heat coagulation occurs typically at temperatures in the second place, heat coagulation of protoplasm, as it occurs in marine eggs, differs from the heat coagulation of pure ungulate. It is true that under ordinary conditions, the heat coagulation of proteins is irreversible. Under certain special conditions, it may be reversible. Thus, if the heat coagulated protein is boiled in the presence of certain electrolytes, it may go into solution again, see Will. The heat coagulation of the erotoplasm in Arbacia and Gumingia eggs. The protoplasm becomes fluid again on return to normal temperatures. In these marine eggs, actual reversal of coagulation is certain. It seems probable that the return of protoplasmic streaming in plant cells heated to the point where movement ceases and then return to normal temperature is also an indication of reversible coagulation. See Saxes 64. So too, we would interpret the reversal of heat rigor in frog muscle, as described by Pyokford 51, Gottschalk 93. We conclude, therefore, that the heat coagulation of protoplasm is essentially different from the heat coagulation of proteins, in that it is reversible. Whereas protein coagulation is irreversible, the proteins are returned to room temperature. The evidence that protein heat coagulation and the heat coagulation of protoplasm have similar temperature coefficients is no proof that the two processes are fundamentally similar. Many other physical processes also have very high temperature coefficients, for example, melting or solution. There is thus no good argument in favor of a direct protein effect as a cause of heat death, and there is much evidence against this interpretation. What other types of explanation are possible? In a later chapter, chapter 12, it will be shown that fat solvents generally cause a coagulation of protoplasm when present in sufficient concentration. Thus, a dilute solution of ether always produces a decrease in viscosity, but with an increase in concentration of the ether, there is a sudden coagulation. What more natural than to assume that warmth has an effect similar to that of ether? High temperatures tend both to dissolve fats and to liquefy them. And as the temperature is raised, there is first a decrease in protoplasmic viscosity, and then a coagulation, just as is the case when the concentration of ether is increased. We know, moreover, that fats may be profoundly modified by the very temperatures which cause heat death in protoplasm. Indeed, it is possible to show that the fats or lipoids of living cells are actually affected by the temperatures which cause death. When sea urchin eggs are treated with temperatures just high enough to cause a slow death, even before coagulation takes 1927, Biochem, p. 231, this does not affect our argument. Place the eggs when centrifuged, show a marked alteration in the appearance of the fatty materials. Ordinarily, these are compressed into a very small cap at one pole, but in heat-treated eggs, the fat particles occupy a much larger zone, and with longer heat treatment, they disappear completely. Heilbrunn 24. Apparently, the fat particles are dissolved at these lethal temperatures. According to Horstadius 23, the lipoids in the protoplasm of the eggs of the annelid Pomatoceras undergo visible changes at a temperature of 40 if the heat coagulation of protoplasm is really initiated by a solution or liquefaction of lipids. Then fat solvents should hasten the process. This was actually found to be the case in Arbacia eggs exposed to a temperature of 32°, Heilbrunn 24b found that 1 to 1% ether decreased the time necessary for coagulation from 24 to 12 minutes. Such low concentrations of ether have a liquefying effect at room temperature, but at 32 they hasten the coagulation of the protoplasm. The action of acids in depressing the heat coagulation temperature of plant cells, Cajo 26b, may also involve fats. Concerning the mechanism of the heat coagulation of protoplasm, we shall have more to say later. It will be shown in a subsequent chapter, chapter 14, that all agents which dissolve fats produce a characteristic coagulative reaction in the protoplasm. The solution of fats is only the first of a complicated series of reactions. If we accept the view that the heat coagulation of protoplasm depends primarily on an alteration of the fats or lipoids of the cell, it is easy to understand why an increase in water content would favor heat death and a decrease in water concentration would retard it. The higher the percentage of water in the cell, the more readily would fats and lipoids tend to dissolve. For Hanstein-Kroner 22 has shown that distilled water can cause a solution of protoplasmic lipoids in various plant tissues. In sea urchin eggs, it is possible to show from a study of centrifuged eggs that distilled water causes a solution of the lipoids of the cell. Various authors have shown that with a decrease in the SAX 64, found that in air, plants were able to withstand higher temperatures than in water. However, in Sachs's experiment, it is probable that the plants in the water actually reached a higher temperature than those in the air. A more dependable experiment is that of De Vries 7D1. He found that when various types of plant tissues were placed in 10% sodium chloride solution, they were able to resist higher temperatures than controlled tissues in water. Similar results were obtained by Werner 99 on frog muscle. He found that in hypotonic solutions, heat rigor of the muscle occurred at lower, whereas in hypertonic solutions, it occurred at higher temperatures than when the muscle was heated in isotonic solutions. The results of Weber 26 also indicate that the heat resistance of stoma guard cells of various plants varies with the amount of water they contain. The higher the considerable evidence has been presented for the view that heat death involves a solution or liquefaction of fats or lipids. It might therefore be expected that animals and plants which suffer heat death at relatively low temperatures would have in their cells fats of relatively low melting point. In 1924, Heilbrunn 24b attempted to show that this was the case. He pointed out that fishes have fats which are typically fluid at room temperature, whereas mammals and birds have fats which are commonly solid. And he suggested a correlation between this fact and the fact that fishes die at much lower temperatures than do the warm-blooded animals. Heilbrunn also quoted Zapek 1321 to show that the oils of tropical seeds have higher melting points than those of temperate climates. This is apparently a very general truth. Thus, palm oil, coconut oil, cocoa butter are much more saturated and have higher melting points than linseed when Heilbrunn's paper was published. It was very difficult to obtain data regarding the melting points of the fats found in the tissues of lower animals, where in general these fats have no commercial importance. The whole subject has been put on a much surer basis by the recent paper of Terras, Bonnet, Kopp, and Visha, 2017. These authors published many determinations of the iodine number of the fats found in the organs of lower animals. Their results show clearly that in cold-blooded animals the fats have a higher iodine number and are thus more fluid than those of warm-blooded animals. But when cold-blooded animals are accustomed to living at relatively high temperatures, they have fats of higher melting point. T. Bruyn, Bonnet, Kopp, and Visha report experiments in which they grew a fungus, Sterigmatocystis nigra, equals Aspergillus niger, and a Bacillus, Bacilla de la Fleule, at high and low temperatures. At the higher temperatures, the fats had a much lower iodine number, that is to say, a higher melting point. T. Bruyn and his co-workers also refer to the experiments of Hendricks and Hansen 1. In which it was shown that the cutaneous fat of pigs reared in the cold had a markedly lower melting point than the cutaneous fat of pigs reared at warmer temperatures. Moreover, it is a well-known fact that the fat in the interior of the body in mammals has a higher melting point than the subcutaneous fat, and this is to be associated with the higher temperature of the interior. See Hendricks and Hansen 1. Leeds and Babb PIO believe that the fats are formed in living systems by a series of reactions in which the terminal points vary according to the temperature of the medium. At higher temperatures, more saturated fats would be produced. These W would have a higher melting point. If in organisms the fats of the cells obey this rule, that is to say, if fats of higher melting point are produced when animals and plants live at higher temperatures. And H, as we have heat death depends on the liquefaction of fats. We have an excellent interpretation of the acclimatization of living organisms to higher temperatures. Organisms are killed by cold as well as by heat, with the exception of some bacteria which can resist temperatures close to the absolute zero. Animals and plants generally, when in an active state, are killed by temperatures in the neighborhood of the freezing point of water. In the discussion of heat death, it was pointed out that those forms of life which could exist in a dried state were far more resistant to heat when water was removed from them. So too seeds, mosses, rotifers, etc., when in a quiescent dry condition, can withstand much lower temperatures than when water is present. Baum, 21, found that dried tardigrades and nematodes. And rotifers, we are not killed by a temperature if we exclude the above-mentioned cases of unusual resistance to cold. There are in the main two types of phenomena which we shall have to consider. Some animals and plants die at temperatures above 0 centigrade. Others die only when their temperature falls below 0. Warm-blooded animals, mammals, for example, die when the body temperature falls to about 15 0. Whether death in these animals is due to a direct effect on the protoplasm of the cells is very doubtful. Tropical plants die at temperatures above 0. Sachs 64 cites various observations of this sort, but these are not very trustworthy. Often the temperature of a plant is below that of its environment due to the cooling effect of evaporation from its surface. In the experiments of Molish 97, any possibility of error of this sort is excluded. It is certain, therefore, that some plants do actually die at temperatures above 0 centigrade. It was shown previously that in the Gomingia egg, a marked increase in viscosity occurred at 1 degree above freezing. It is quite possible that such a coagulative effect would be an important factor in causing the death of cells at temperatures just above the freezing point of water. Mammalian muscle contracts on cooling to 5 EC, Batazzi 25B. There is thus a cold rigor as well as a heat rigor of muscle. In the Gomingia egg, the coagulation due to cold is reversible, just as is the heat coagulation. But it is not at all improbable that after a lengthy exposure to 1C, the protoplasm may be permanently injured. Concerning the reasons for a sudden increase in viscosity at temperatures just above the freezing point, we have at present no real information. A few scattered observations indicate that in this case, too, there may be a change in the physical properties of the lipoids of the cell. In Gomingia egg centrifuge at PC, the lipoid particles present a very characteristic appearance, quite different from that found in similar cells at normal temperatures. Most animals and plants are killed by the cold only when their temperature drops below the freezing point of water, owing to various economic jihazes of the subject. There have been many more studies of the cold death of plants than of animals. In temperate or cold climates, the death of plants as a result of exposure to cold is an important agricultural problem. The older literature on the effect of freezing temperatures on plants has been well summarized by Fetfetfu. For later summaries, see Chandler 13, Rosa 21st, Newton 22, Doyle and Clinch 26, Aikman 27. Generally speaking, death in plant cells. At one and the same temperature, death occurs if crystals of ice are formed, whereas if no crystals appear, the cells live on, see verb, also Kylin 17. The crystals may be formed either within the cells or outside of them. Concerning the cause of death, the leading theory is that of Milibethuegai at 8086. This author holds that as ice forms, the salt concentration of the rest of the protoplasm increases until the death point is reached. Milibethuegai's theory is supported by Malish 97 and by Maximo 14. However, the latter author believes that in addition to the salt effect following ice formation, there is also a mechanical effect of the ice crystals on the colloids of the protoplasm. If the Mtiller-Thucau theory is correct, then cold death at freezing temperatures is really a special case of the toxic action of high concentrations of salt in the cell. Viachuahat and Mollard, 2 and Zalhaboa 26 have described the formation of numerous vacuoles in cells killed by cold. These vacuoles appear both in cytoplasm and nucleus. They may owe their origin to the formation of ice crystals, or perhaps they are the result of increased salt content. As will be shown later, the appearance of vacuoles is a logical result of the increase in the concentration of salts in a cell, CP249. Chapter 9. The action of various physical factors other than temperature. The action of various physical factors other than temperature not only is protoplasm sensitive to changes in temperature, it is also sensitive to other physical factors of its environment. In the present chapter, we shall endeavor to show how the protoplasmic colloid is affected HJ physical forces and agencies of one sort or another. It is not always easy to separate physical factors from chemical. When a cell is treated chemically, such treatment may involve physical effects, a change in surface tension, for example, or a difference in the osmotic pressure on the cell membrane. So, too, physical agencies may act primarily by the initiation of chemical reactions. It is impossible to draw any sharp line. For the purposes of classification, we will consider as a 2SEB, 3 physical factor anything that starts as such. We shall discuss the action of the following: 1. Mechanical factors, 2. Electric currents, 3. Light, 4. X-rays, 5. Beta and gamma rays of radium. Biologists have studied the effects of pressure on protoplasm and the effects of wounding. Practically all of the work has been done by botanists. Since the time of Dupuchet 37, it has been known that mechanical pressure exerted on an extant cell can cause the cessation of protoplasmic streaming. This observation has been repeated by various observers. A relatively recent paper which deals with the subject is that of Lauterbach 21. References to the older literature may be found there. Lauterbach observed that in younger cells and at higher temperatures, less pressure was necessary to cause cessation of movement. Sly found also that the response to pressure, that is to stoppage of movement, traveled from cell to cell. All Tliask, fact deets were known to a Watt 3. When streaming protoplasm is subjected to pressure, it is probable, but of course not certain, that the cessation of movement is due to a coagulation or a sharp velocity increase in the protoplasm. In 1910, Lepeshkus described what Lie called a eucluini, coagulation of the protoplasm. Filanoituvu of B. pyrogyra were placed under a cover slip and distorted several times by Eierescher from above. Following this treatment, according to Lepesoki, the outer protoplasmic layer coagulated and the inner protoplasm broke Ujo into a number of balls. Some knows the cells burst and the balls passed out into the cerulean field. After half an hour, all the balls were coagulated too. Lepeshkik's criterion of coagulation is wholly a neo-biological one, and depends in the main on the appearance of nuclearides. Many authors describe the appearance of vacuoles following mechanical injury. Thus, Heilbrunn 22 states that inguinal injury produces vacuolization in slime mold plasmodia. Huentuor 1 states that countless vacuoles appear in onion, 91s, following mechanical coagulation. These had previously Ichui described in fixed material by Nemec 1b, but this Noel LHG is very much older, as evident from the following quotation from a paper in 1876. Heilbrunn entstehen in den verschiedensten Zellen wie dies bereits bekannt die Equal. Wo die Einwirkung nicht zu stark, so verschwinden sie nach einer Ruhepause wieder und das Protoplasma kami in seinen normalen Zustand zurückkehren. The formation of numerous vacuoles within the gemotoplasmy of a cell would beyond IE question cause a marked increase in the protoplasmic viscosity. If enough vacuoles were present, the protoplasm would of curly be totally incapable of flow. It would be gelled or coagulate. HL. There are two different ways in which mechanical fasciitis can act on protoplasm. Cells may be subjected to pressure insufficient to cause any rupture of the cell membrane, or they may be compressed so as to force out the interior protoplasm until it comes back with the outer medium. Tear and tearing of cells also exposes the interior protoplasm, oath, or COPTRSL, so that its protoplasm comes into contact with the R medium, the action of various physical factors, etc. 127. The naked Jane rotoplasm often reacts by forming a film. It is not our purpose at this point to consider the process of film formation about emerging droplets of protoplasm. This is a subject that will be taken up in detail in a later chapter, see Chapter 13. There is an interesting possibility that the coagulative effects of pressure are in all cases due to an exposure of the cell interior to the outer medium. When a cell is violently compressed, we see the protoplasm emerge. But if a cell is subjected to a less violent gerasure, it may very well happen that the cell membrane is broken here and there and immediately mended. If these slight were small, and if the mending process were rapid, the rupture of the membrane would certainly escape our notice. Evidence in favor of the view that all mechanical coagulation involves a rupture of the membrane of the cell is found in the fact that the appearance of vacuoles in cells under pressure, as mentioned in the sepietation from Velton, is also a phenomenon which occurs in cells which have been broken or torn. See chaps 13 and 14. If this point of view is eventually shown to be correct, we may be in a better position to understand the remarkable effects which pressure has on living systems, effects quite unlike anything that is to be found in inanimate colloids. But for the present, we have nothing more than an untested hypothesis. Some enough thing has already been said regarding the effects on protojoasm of pressures not great enough to cause a visible emergence. We shall now discuss the results of mechanical injury, such as is produced by a cut or wound. When an animal or plant is cut, the effect is not limited to the cells actually injured, or even to the cells at the border of the cut. Cells at a considerable distance from the cut surface may be affected. It seems not at all unlikely that the transmission of the injury effect from cell to cell is due to the diffusion of chemical substances formed in the injured cells. Thus, Frank 72 found that the morphological changes produced by injury appeared sooner in cells in the neighborhood of the midrib in the case of leaves of Eladia. This would indicate that the changes are due to a substance which diffuses from the cut or injured region. There is also other evidence which points in the same direction. Some of this will be brought forward in later chapters. But for the present, we shall consider the mechanical coagulation high itself, and we shall not attempt to understand the mode of its transmission or to determine whether this transmission is more physical or chemical. When plant tissues are injured, both morphological and physical changes occur in the neighboring cells. Although it seems quite probable that the morphological changes depend on the physical, no one has attempted to show a relation between the two. The morphological changes have been studied by cytologists, the physical changes by physiologists. In plant tissues, various form changes can be seen in the neighborhood of an injured or cut surface. Frank 72 described movements in the chloroplasts of Elodia cells. Tangle 84 found that protoplasm and nuclei of onion cells moved in a direction toward any injured region. Tangle cut the scales of onions with a sharp knife. Istessel 98 repeated Tangle's observations for many types of plants. It is interesting to note that Nestlé used not only a knife and glass needle, but he also found similar changes following wounds, JN reduced by the heat of a burning glass. Movements of nuclei in cytoplasm toward cut or injured surfaces have also been described by Nemec 1b, by Calling 26, and by various others. For references, see Cabling 26. The physical changes that occur in protoplasm following an injury to cells in the neighborhood have also been studied by botanists. Heilbrunn 12 found that when sections were cubbed through bean seedlings, Pliasalis and Vita, the viscosity of the protoxtrelasm was abnormally high for 10 to 15 minutes after the sections were made. During this time, the starch grains in the starch sheath cells did not move at all under the influence of gravity, although they moved rapidly enough later. Similar observations have been made by various other workers who have used the gravity method of measuring protoplasmic viscosity. See, for example, F and G. Wilbur T7, also Zolico, year 18. Recently, Dwenning 26 A and B has made an interesting quantitative study of the increase in viscosity which follows wounding. Dwenning studied the viscosity changes with the gravity, centrifuge, and plasmolysis, form methods. In his epidurements, he cut the plant tissue with a razor, but he is careful to show that there is no chemical effect of the metal. For he states that controls in which there was no possibility of chemical action gave the same results. Compare also the experiments of the action of various physical factors, etc. 120 vodka, H40. In his first paper, Buvinal used seedlings of Sical cereal and Raphanus satavis and sprouts of T. Descinia viridis. The seedlings were decapitated, and then viscosity determinations of the protoplasm of cells near the cut were made both with the gravity and centrifuge methods. In these viscosity determinations, the starch grains or granules were made to move in intact masses of tissue, and then sections were cut to determine the extent of the movement, as pointed out in a previous chapter. 44. This method is preferable to the older method of cutting the sections first, and then watching the fall of starch grains through the cells of the section. Dwenning found that coagulation occurred in cells several layers distant from the cut surface. There is a sharp increase in viscosity. The time required for the attainment of maximum viscosity varies somewhat in the different species. In Sical, at least 6 minutes elapse after cutting before maximum viscosity is reached. In Tradescantia, the maximum viscosity appears to be reached within 1% minutes. The coagulation or increase in viscosity is reversible. Thus, in Tradescantia, centrifuge tests showed that 6 to 7 minutes after cutting, the viscosity had returned to normal. When, however, instead of a single cut across the stalk, thin sections were made, a longer time was necessary for recovery. Under these conditions, in Tradescantia, the viscosity only returns to normal within 10 minutes at the earliest. In a similar experiment in Raphanus, the return to normal required 30 minutes. Winnebig points out that a longer time is necessary for recovery than for the development of maximum viscosity. The increase in viscosity following a cut through the stalk is quite considerable. In the cells of Cicallo, 1 minute after the cut, the viscosity rose to 4 times its original value. In Tradescantia, after half a minute, it rose to 3% times its original value. And in Raphanus, after 2 minutes, the viscosity was at least 5 to 6 times as great as before cutting. In a later paper, 26b, Weinach studied the effects of wounds on the protoplasm of onion cells. He used the cells of the scales of the onion, and as before, he stimulated by cutting with a razor. In the onion cells, there are no starch grains or larger granules, and neither the gravity nor the centrifuge method could be used. Bigening, therefore, used the plasmolysis form method. The time required for coagulation is not more than 30 seconds. The increase in viscosity is followed by the clouding over of the protoplasm and the appearance of new granules both in the cytoplasm and in the nucleus. There are also many small vacuoles formed during the coagulation process. Electric currents have a marked effect on various types of living material. In muscle and nerve physiology, the electric current is the most common means of stimulation. Lower forms of animal life and various plant tissues also show a characteristic response to the passage of electricity expected. It is to be expected, therefore, that the protoplasmic colloid should undergo changes following explasmy to electric currents. There is an abundance of evidence to prove that this is the case. In 1862, Brucke showed that electric currents produced a cessation of Brownian movement in the salivary corpuscles or leukocytes of human saliva. Kiel R&D 64 found that the passage of an electric current from an induction coil caused a cessation of Brownian movement in the protoplasm of the stamen hair cells of Tradescantia. Choala and Gautier 5 also found that the passage of an electric current stopped Brownian movement in the cells of the alga Casmerium. Baylis 20 studied the effect of currents from an induction coil on Brownian movement in the protoplasm of amoeba. The amoebae were placed on a slide in a drop of water, and the electric current was led to the drop through strips of platinum foil. With this technique, there is, of course, the possibility that various chemical substances formed at the electrodes may have an effect on the protoplasm. But Baylis's observations were usually made very rapidly, so that this was probably not a factor. In studying the Brownian movement, Baylis used a Zeiss paraboloid condenser, and he focused his attention on the ectogelism of the pseudopodia. Here one can observe a very vigorous movement of granules invisible with bright field. On the passage of an electric current, this movement ceases almost instantly, as if the protoplasm had been frozen. Then when the current is stopped, the Brownian movement begins again. In order to obtain these results, it is necessary to use currents that are neither too weak nor too strong. The latter produce an irreversible coagulation of the protoplasm. The action of various physical factors, etc. 131 from these studies on Brownian movement, it is apparent that electric currents can produce a reversible gelation or coagulation of the protoplasm. A much more exact study of the effect of the electric current on the colloidal properties of erotoplasm was made by Biebsa and Weber 22. These workers were careful to exclude any possible error due to the formation of chemical decomposition products at the electrodes. The current passed from carbon electrodes through troughs containing tap paper. Water and filter paper were changed from time to time so that there was no possibility of the diffusion of chemicals from the electrodes to the cells. The material used was the stalk, Epicala, the bean plant, Phaceolus muiflorus, and the current was passed through pieces of the stalk about 1 to 2 centimeters long. The viscosity of the protoplasm was determined by the centrifuge method. The results of the experiments are very clearly summarized by Biebsa and Weber, and the account that follows is practically a translation of their words. In order to produce a viscosity increase in the protoplasm, an exposure of only percent to percent minute is necessary if the current is relatively strong. 5 to 10 milliamperes. With weaker currents, 0.150 to 5 milliamperes, an exposure of 1 to 4 minutes is necessary to produce the same effect. The increase in viscosity produced by these currents is considerable, for it is at least a threefold increase. The effect is reversible, and after a recovery period of 20 to 40 minutes, the viscosity becomes as low or nearly as low as it was before exposure to the electric current. The results of Bursa and Weber find confirmation in some experiments of Zeidler 25. It is obvious that the results obtained from observation of Brownian movement and those obtained by the centrifuge method are consistent. Moreover, as we shall now proceed to show, these results are in complete harmony with the older knowledge concerning the effect of electric currents on the rate of protoplasmic streaming. This knowledge goes back to the year 1837. In that year, Bickel found that the passage of Veitch-Velten 76C describes a case in which the passage of an electric current through Elodius cells caused an initiation of Brownian movement. This was apparently due to the fact that the granules of the protoplasm were dislodged into the vacuole where they had more room to move. An electric current through CS of Cham produced almost immediately either a retardation or a complete stoppage of the protoplasmic streaming. Following Bickel, the action of electric currents on protoplasmic streaming in plant cells has been studied by JTX RG Irsek 61, Brucke 62, Heidenreich 63, Kuhn 64, Velt BN 76C, Hormate 98, Evart 3 and Ikoketsid 23. These authors worked with a wide variety of plant material, and in general they found that electric currents cause the rotation of the protoplasm to become slower or to stop completely. There are one or two exceptions which might be worth considering. Occasionally, according to Velter 76, there is a slight acceleration of the rate of streaming in leaf cells of Vallisneria spiralis. This Velta attributes to a heat effect as a result of the passage of the current through material of high electrical resistance. There were indeed indications that the cells actually did become warmer. Koketschi 23 also describes one case in which there was an acceleration of the rate of streaming in a part of a CGR cell. He favors a different explanation from that of Velten, but in the great majority of experiments, and they are many, the passage of an electric current has a very marked retarding effect on the speed of streaming. An experiment of Koketschi's may be taken as illustrative. In this experiment, he stimulated a Cham cell 7 times with a current from an induction coil. The current was of various strengths, being less, of course, as the distance between the primary and secondary coils was increased. Following each stimulation, Koketschi measured the speed of protoplasmic streaming by determining the time it took for the J protoplasmic granules to travel a speck. The time was recorded in beats of a metronome. Each beat B28, the head of the columns, referred to the successive stimulations. The distances in centimeters being the distances between the primary and secondary coils in each case. The first column gives the time at which speed determinations were made. In the other columns are records of inverse speeds. These figures show the times in metronome beats required for the protoplasm to move a given distance. These times would, of course, vary directly with the viscosity. Beating down any one column, one finds a record of a single experiment. When a complete stoppage of streaming occurred, this is indicated by SB. The action of various physical factors, etc. 133 speed of protoplasmic streaming in O'Hara after electrical stimulation, Koketsu 23. Time of observation before stimulation. Directly after stimu, if one compares the observations of Koketsu with the viscosity measurements of Bebsa and Webb, one is struck with the similarity. Apparently, the action of electricity on the rate of protoplasmic streaming in plant cells is due primarily to an effect on the viscosity of the protoplasm, rather than to an effect on the motive force of the streaming. In animal cells, too, the flowing movements of the protoplasm may be stopped by the passage of electric currents. See, for example, TT64 in Ullmann 69. But here the phenomenon is complicated by changes in the form of the cells. Moreover, for animal cells, no quantitative studies have been made which are at all construable in exactness with those which have been made on plant material. For both plant and animal cells, it seems certain that the passage of an electric current very quickly produces a reversible gelation or coagulation of the protoplasm. This is a fact of great importance, and it deserves further study. It seems probable that all types of irritable protoplasm may be electrically coagulated. Concerning the possible mechanism of such a process, we know little or nothing. The fact that protoxolasm is so sensitive to electric currents seems to indicate that the electric charges of the living substance are very important from a colloid chemical standpoint. If protoplasm were simply a colloidal solution of exerted so powerful an influence. If we consider protoplasm as primarily a suspension, and we have abundant reason for so doing, see chaps 2 and 5, we are perhaps in better position to understand the peculiar coagulative effect of electric current, for the stability of a suspension depends to a greater extent on its charge than does the stability of a lyophilic colloid, such as a protein solution. When an electric current is sent through a suspension or a suspensoid, the colloidal particles wander to one electrode or the other, and on reaching the electrode, they give their charge and are coagulated. But the contents of a cell do not wander to the electrode unless we consider the bounding membrane of the cell as an electrode. This, in a sense, it is. If we pass an electric current through a suspension so that its particles move toward a membrane, as for example in electrolysis, the colloidal particles, when they reach the membrane, lose their charge and become precipitated. Perhaps a similar process occurs in protoplasm, but this seems doubtful, for the electrical coagulation may be practically instantaneous even in large cells. In a later chapter, we shall have occasion to consider the electric migration or cataphoresis of protoplasmic granules through the cell, see chapter 10. The mechanism of electrical coagulation of protoplasm may very well be related to the vacuole formation which frequently accompanies or follows the passage of an electric current through the cell. This is a complicated subject and one that cannot be adequately discussed at this point. It will be considered later, see chap 14. The action of various physical factors, etc. 136 in recent years. There has been great interest in the action of light on plants and animals, but up to the present, there has been very little study of the effect of the light on the colloidal progeries of the protoplasm. Many authors have shown that light rays, and especially ultraviolet rays, cause the death of bacteria exposed to them. For references, see Pincusset 21 Clark 22. This death, no doubt, sooner or later involves a coagulation of the protoplasm, but one has no means of knowing whether or not this coagulation is a direct result of the radiation. PBT and Scheim 81 exposed plant cells to concentrated sunlight, taking care to avoid heat effects. Pi found that the increased intensity of light caused a decrease in the rate of protoplasmic streaming in Nutella. In Spirogyra cells, the concentrated sunlight tended to stop the TTE, thought Ornchenbewegung, in the wall layer of the protoplasm. This Ornchenbewegung probably depends at least partly on the Brownian movement of the granules. Many authors have studied the action of light on protoplasmic streaming. In most instances, their results seem to depend more on an action of the light on the propelling force of the streaming than on the viscosity. Light rays have frequently been found to have an influence on the initiation of protoplasmic streaming. For references to literature, see Baikiro 25, Dart. Baker himself found that the stimulating effect of light on streaming consisted in an increase in the number of cells, which showed streaming rather than in any increase in the maximal speed with which the protoplasm moved. Ultraviolet rays seem to have a specific effect in slowing or stopping the streaming movement of protoplasm, and this effect may very well be on the viscosity of the protoplasm. Hertel 4 found that when Elodia cells were exposed to ultraviolet rays with a wavelength of 280 UU, the rotation of the protoplasm first became slower and then stopped completely. Similar results were obtained by Schultz 10 for the stamen hair cells of Tradescantia, the root hairs of Hydrocharis, and the leaf cells of Vallisneria. He also used rays with a wavelength of 280 I. Valk 12 studied the effect of ultraviolet rays from a mercury vapor lamp on the streaming of slime mold protoplasm. In most of his experiments, he exposed the plasmodia for from 1 to 5 minutes and found a cessation of movement, which was usually reversible. However, the longer exposures caused death. In one experiment, in which the exposure was only 30 seconds, the speed of the streaming was increased. Such an increase in streaming was also observed when the rays passed through glass, so that the acceleration may have been due to an action of the visible rays of the spectrum. In his experiments on the stamen hair cells of Tradescantia, Schultz noted that the rays of 282 wavelength caused the appearance of many small vacuoles. These could be noticed even after an exposure of 30 seconds. That ultraviolet rays cause coagulation of protoplasm seems certain. Sibanovidianu and Henne exposed paramecia and white blood cells to ultraviolet rays and studied the appearance of the protoplasm under dark field illumination. They found that the protoplasm became more granular and appeared brighter, plus brilliant. This is evidence of coagulation. Huppert 24 treated Ascaris eggs with ultraviolet rays, 280 at various stages of mitosis, and then centrifuged them immediately. Before treatment, the eggs were kept in an incubator at a temperature of 37 C. The centrifugal treatment was always the same. The eggs were centrifuged for 30 minutes at a speed of 3,200 revolutions per minute. The radius of turn was about 10.5 centimeters. This is rather a severe centrifugal treatment, compare P103, and it is not surprising that the eggs always showed a complete stratification. But there was a very noticeable difference between the eggs which had been exposed to ultraviolet rays and the control eggs. In the exposed eggs, there was often a much slower return of the granules after the centrifuging process was discontinued. Sometimes the granules did not return at all. A similar observation had previously been made by Schultz 23. Both Schleip and Buppert conclude that there is a marked increase in viscosity in the protoxylosni of the eggs exposed to ultraviolet rays, and this conclusion certainly appears to be justified. In one experiment, Buppert, instead of centrifuging the eggs immediately after they had been exposed to ultraviolet rays, kept them overnight at room temperature and then centrifuged them. In this experiment, the eggs treated 5 and 10 minutes showed no movement of granules on being centrifuged, so that the viscosity of their protoplasm had evidently been greatly increased. Gibbs 26 studied the effect of ultraviolet rays from a mercury vapor arc on the extrotoplasm of Spirogyra cells. He found that the action of various physical factors, etc., 137 radiation between 3126A and 2378, i.e. 312.6 to 237.8, just cause first a liquefaction, and then on longer exposure, a coagulation of the protoplasm. Viscosity measurements were made with the shows the decrease in protoplasmic viscosity produced by relatively short exposures to the mercury vapor arc. Effect of ultraviolet light on Spirogyra protoplasm, Gibbs D26. Cells exposed to arc, screened with noviola glass slight displacement, marked displacement, with ultraviolet radiation from a mercury vapor lamp. The seed beings were exposed for 10 to 15 minutes, and the root hairs were then examined in dark field cardioid condenser. From the morphological appearance of the protoplasm, Adams concludes that coagulation has occurred after 10 minutes. The seedlings are not injured by the treatment. Root hairs covered with glass show no coagulation. Very soon after the discovery of X-rays and of radium, experiments were made to find out what effect or effects the new radiations might have on living organisms. And as soon as it became apparent that both X-rays and radium might have clinical value, these experiments became much more frequent. So that already there is a vast literature in this field, a literature which is scattered through biological and medical journals and journals devoted especially to the study of the effects of radiation. Most of the work that has been done deals with gross effects on whole animals or plants or human beings. Plants grow more rapidly or more slowly. Animals are killed or men are cured. Biologists have not infrequently described effects on CAU division or on the course of inheritance. Truly physical studies on the action of X-rajas or on the protoplasm have been relatively few. Some recent authors incline to the view that the primary effect of radium radiations and of X-rays is on the medium surrounding the cells. For literature on this subject, see Sartaby, Sebtobi, and Anbib 27. It is very commonly believed that the nucleus of a cell is much more sensitive to radium and X-rays than is the cytoplasm, and that the nuclear material is particularly sensitive during the metaphase when the chromosomes are not surrounded by a nuclear membrane. Numerous papers have been written in support of this thesis. For references to some of them, see Shins 24, Seed 25. Rius 25 states that the nucleus of the sea urchin egg is 18 times as sensitive to radium as is the cytoplasm, and 3.8 times as sensitive to X-rays. There have been relatively few studies of the effect of radium and X-rays on protoplasmic streaming. The results as far as they go indicate that the action of X-rays and of beta and gamma rays of radium are similar to those of ultraviolet rays. Webb, too, found no effect after treating two types of plant protoplasm with X-rays. He radiated Spirogyra cells with 170 Holzknecht units and then centrifuged 5 minutes later. There was no observable difference between the radiated material and the controls. After 20 hours, an increase in viscosity did occur, but this Webb interprets as being due to a secondary effect. Webb also radiated seedlings of Pliasolus MDDT florals with 24 Holzknecht units. But again, he was unable to detect any change in the viscosity of the protoplasm. Apparently, some types of protoplasm are very resistant to X-rays. This is certainly true for paramecium. In this form, prolonged exposure to strengths of X-rays that would have a very serious effect on the life of higher types of animals have no marked effect either on the protoplasmic viscosity or on the activities of the animal. Unpublished experiments. However, Williams 23 was able to show very definite effects of X-ray treatment on the protoplasm of the stalk cells of Saxifraga umbrosa. Short exposures caused an increase in the rate of protoplasmic streaming and also an increase in the Brownian movement of the small particles of the protoplasm. Longer exposures caused a diminution of the rate of streaming and finally complete stopstage. Similar results are also recorded in an early paper of Lop BOB 98, but this worker studied only the rate of streaming and not the Brownian movement. He found that a half-hour treatment with X-rays caused an increase the action of various physical factors, etc. 139, in the rate of streaming in the protoplasm of the leaves of Vallisneria spiralis. Treatment for an hour caused the protoplasm to become yellow, granular, and coarsely vacuolated. SEC2 also found that short exposure to X-rays increased the rate of protoplasmic streaming in various plant cells, and that longer exposure slowed it. From all these results, it seems probable that X-rays first produce liquefaction and then coagulation of the protoplasm. Similar effects have been found to follow radium treatment. Thus, Forbes and Thakub 25 found a decrease in viscosity of the protoplasm of the eggs of Nereis and Annelid following treatment with radium. In their own words, Nereis eggs radiated with G-rays, fertilized, and then centrifuged from 7 to 65 minutes after fertilization, shows slightly sharper separation of resulting zones than is found in unradiated controls. The difference is most easily explained as due to decreased viscosity of the protoplasm in consequence of radiation. Williams 25 studied the effect of beta and gamma rays of radium on the protoplasm of Saxifraga umbrosa. Short exposure caused an increase in the amplitude of the Brownian movement of the small particles of the protoplasm, together with an increase in the rate of J-plasmic streaming. Evidently, a decrease in viscosity is produced. Longer exposure to radium caused coagulation and vacuolization of the girotoplasm. Comparable results were also obtained by Zwelzer and Filper 25 on amoeba. These authors found that short exposures to radium caused an increase in the speed of amoeboid movement. Longer exposures resulted.